Good morning, my dear children. Just give me a second. Let me see if at all I am audible as well as visible. Yeah. Yes, I'm perfectly audible and visible. Very good morning, Kishan. Time is over. And Mir Jafar, good morning. Sabira, Vimala, and Kishan, Ajay, Manya. Oh my God, Shama. And Hina, everyone, Minaz, good morning, good morning. Just give me a quick second. I'll share this particular video link with the children in the WhatsApp group as well, so that together we can attend this very important live. Okay, perfect. So, my dear children, I hope that I am clearly visible as well as ma'am, good morning, ready to tackle toughest questions you have for us today. So, you are actually thinking that I brought tough questions, is it? Good morning, everyone. So, the thing I'll tell you very honestly how it goes. So, there are 103 questions that I have taken. And it will be a mix of PYQs as well as the newer questions which are there. You might find that the newer questions which are there are kind of similar to the questions that have come, because obviously you have to choose from the repeated concepts. Now some questions would be tough, some questions would be easy, and we are going to attempt all of it even if we know or do not know it, okay? And for the questions that you do not know, what you have to do is mark it up that, okay, question number 1 is the one that I have no idea, probably I should be revisiting to that particular question and doing it later on. Also, if you have any big doubts while I explain, okay, you can wait for some time and then ask me. Yeah? Yes, Shweta, good morning, Chetan, good morning, Macha ma'am, Hemant, everyone, perfect. So, Baba, let's begin. If everyone, all the machas and machis are ready over here, give me a quick thumbs up. Let's begin up with a powerful session this morning. Yeah? Good morning, Sanket. Yo. Thumbs up, please. Everyone, thumbs up. Deadpool, hello. By the way, if at all I miss out any one of your name, please forgive me at that particular point itself. Yeah. Okay. Cool. So, yes, I have got the thumbs up, and here you go. The first question is from the chapter Principles of Inheritance and Variation, and the question goes like this. How many types of genetically different gametes will be produced by a heterozygous plant having the genotype this? So, this is a genotype, and you have the options over here. How do you have to give me the answer? 1A, 1B, 1C, whatever is your answer, you can give me accordingly. Okay? Mokshita, hello, Mokshita. Okay, you're looking nice, thank you. Okay, so guys, what is the answer for this particular question? Come on. MD Sulaiman says the answer for this question is C. Okay. What about others? A. Kishan says it is A. Guys, give me the answer like this, 1A. Otherwise, there are 100 questions, we will be confused, okay? It's for you guys only. Savita says it is D. Kavana says C. GS Suni says C. Venkatesh says A. Heman Gowda, Papa Paul is not there over here. Yeah. And D. Shweta, Hazira, Viresh. Okay. So most of you guys are saying the answer for this question is D. D is what? 4. D is 4. So Baba, how are you getting 4 over here? For children who are like, I do not know the answer, listen to me out very, very carefully. It goes like this. You have been given that the parent is like this. Capital A, capital B, small b, and capital C, small c. Right? So basically, if at all you want to find the number of gametes over here, there is a chutku formula, macha, and it goes like this. 2 raised to the power n. What is n over here? N is equals to the number of heterozygotes. What do you mean by heterozygotes? The one where the alleles are different. So over here, if at all you see, capital A is there. Both the alleles are same only, so I cannot count this. Okay. What about capital B, capital small c? So listen, this is capital, this is small. So this is an heterozygote over here. So I have got my first heterozygote. What about capital C, small c? This is also an heterozygote. So basically, in total, how many heterozygotes I have got? I have got 2. So basically, I am going to replace this n by 2. 2 raised to the power 2 is equals to what? It is equals to 4. 2 into 2 is equals to 4, which is why the answer for this question is going to be D. Is it okay? Yes, so all of you who gave me the answer as D. So there is a Korean name being mentioned, which obviously I cannot read out, and Sudarshan and Samarth, Rukmini, everyone, okay. Shweta Patil, Vimla, and Chetan, Deepa, okay. Shravan. So I'm seeing you guys, okay, and ma'am, is biology compulsory? Only for the courses where biology is needed, okay. For example, farm science courses, you have to write PCMB, okay. In certain courses, you just have to write PCB. So wherever you require biology, yes, you have to write it. Sorry, ma'am, it's D. It's totally fine. Vidya, very good morning, dear. From this video, you will be able to get the maximum marks out there. I have put in my efforts. Yesterday, I slept at 3 o'clock. Okay, yesterday I slept at 3 o'clock. I was just making the PPT. I tried to be, as specific as possible as how the exam question paper has been throughout these many years. So let's wish the best for each other, right? Yes. Okay. So let's go ahead with the next question. Question number 2. If yes, give me a thumbs up. Rukhaya, if at all you understand the concepts well, you will score more than 45. Okay? Good morning, Lakshman. Guys, give me a thumbs up if at all it is clear, and we can go ahead with the next option. Good morning, Dreams. Okay. Thank you, ma'am, for studying. It's okay. You're most welcome, Acha. Okay, perfect. So guys, the rule goes like this. If at all you are someone who is new to my lives, the rule goes like this. After every question, we have to give a thumbs up. Why? That's only when Jasmine ma'am gets an understanding that, okay, children are basically getting things well. Yes, yes. So let's go. Perfect. The next question that we have over here is like this. Select the incorrectly matched pair. Baba, why I have put this question is that every term that you see over here has given a question before. So you need to have an understanding of these terms. That is exactly why I have given you the question. So select the incorrectly matched pair. So here you have the options. Pleiotropy is equals to height in humans. Linkage is seen in Drosophila. Incomplete dominance is seen in Antirrhinum. And codominance is basically shown by ABO blood group. What do you think is the answer for this particular question, Macha? Adarsh, good morning. Guys, the reason I have not kept the poll is that it's quite distracting, because in the polls anyway I cannot see your name. Do you want the polls? Yes. Thank you so much, Mania. Okay, so BAAAA, Abdul, Savita, Jarufication is also there. Oh my God. Okay, so Hazira, Shweta, Hina Kausal, Kavana, Harish, MD Suleiman, Anvita, Shweta, Vimla, Vayappa. Oh my God, so many children are there. Hemant, I'm just reading from between. Tarun, hello dear. Okay, Dreams, Kumar, Shweta, everyone. Most of you guys are saying the answer for this question is A. A is what? Pleiotropy and height in human beings. Is it A? You have been asked to find the incorrectly matched pair. Yes. Why it is A? Because height in human beings is an example of polygeny, not pleiotropy, and most of us get confused with that. Polygeny is something different, pleiotropy is something different. Polygeny means multiple genes which are there. Okay, so suppose I am listening over here, you have this gene, this gene, this gene. All of these genes are coding for something that is going to determine my character. So one character over here is going to be determined by multiple genes, which is known as polygyny. What did Mendel Baba said? He said that capital T and small t. Capital T is there for tall height, and small t is for dwarf height. You will not find any other alleles anywhere else. But this is not the case. Here you will find multiple alleles are there coding for one particular character, and that is known as polygyny. And where do you see it apart from height? Any idea, guys? Come on. Where do you see it apart from height? Polygyny is seen in, it's an easy question. Yes, it is an easy question. Okay. Polygyny, where do you see it apart from height? Come on, come on, come on. Dinky chika. Skin, exactly. Akshita says the answer for this question is what? Skin. So skin color, okay, so I'll tell you one thing. Please understand, I am taking time over here, but you need to understand this particular portion is important, okay? You might get questions from anywhere from these topics. So over here, listen, Baba. Suppose your skin color and my skin color is it same? No. Our skin color is totally different. Why? You and your friends, do you think it will be same? No, that is also going to be different. Why? Because skin color is going to be coded by multiple genes like this. For example, capital A, capital A, capital B, capital B, capital C, capital C. These three, all of them together gives me a skin color, and this is going to be the darkest skin color. If at all it's a combination like this where all the dominant alleles are there, it is going to be darkest skin color. Now just imagine that if at all you have a small allele, will that make any change over here? If at all I have a small allele over here, will that make any change? So it becomes capital A, small a, capital B, capital B, capital C, capital C. The skin color will be lighter than the previous one. So which one will be the lightest skin color? Like this. If at all of them are recessive, it becomes the lightest skin color. And always remember, height and skin color is an example for what? Polygyny, as I'm explaining over here. What about pleiotropy, Baba? Pleiotropy is something different. Pleiotropy means one gene has multiple effects. Ulta of this. It's what? The ulta of this. So one gene is there. Okay, and it is having multiple effects. Here, multiple genes are there having one effect. So, for example, pleiotropy's example, anyone, can you tell me the example for pleiotropy? And everyone who gave the answer, Venkatesh, Bhavish, Hina Kausar, Nagaraj, and Varsha, and Jurification, Yashu, okay, and everyone, Vinay, Sinch, Hazira, perfect. So, you guys are going really, really, really great. Okay, guys, skin. Din, da, da. Yes, Hina Kausar, small a, small b, small b, small c, small c is equals to the lightest skin color. Phenylketonuria is an example for what? For pleiotropy. Remember, in phenylketonuria, what is happening? One enzyme is not being produced. What is the name of the enzyme? Phenylalanine hydroxylase. Yes, exactly, guys. When phenylalanine hydroxylase is not being produced, what's the problem with that? Always remember, when this particular enzyme is not being produced, first of all, the conversion does not happen. Conversion of what? Phenylalanine to tyrosine. This conversion does not happen. Ma'am, is that a problem? Yes, because this guy is going to accumulate in our body in the form of phenylpyruvic acid. And when it accumulates in the body in the form of phenylpyruvic acid, you know what it causes? Mental retardation. So, ma'am, only one effect is there. Now, what's the next effect? You are saying one gene, multiple effects. So, the multiple effect is that basically this is not getting converted, so it is accumulated, one thing. Second thing, when tyrosine is not being formed, Baba, it is going to hamper our pigmentation, our hair and skin pigmentation, Baba, it becomes reduced. Why? Because tyrosine is super important for the formation of some very important pigment of our body which gives us color, skin color and stuff like that. You get it? So two effects, okay, two effects are being seen by one gene. That is why it is an example of pleiotropy. So this and this is not going to be matching. Linkage is seen in the case of Drosophila. And incomplete dominance is seen in the case of antirrhinum, and the codominance is found in ABO blood group. So the only answer for this question is going to be A. Is it clear? Number of questions, how many? 103 questions are there. We have to do it, okay? Ma'am, when is chemistry class? Chemistry class just got over, Kanya. So you can go and watch the live which was there, previously been done. Okay? That works for everyone? Yes. So every one of you who gave me the answer, you guys are doing absolutely great. Yeah? And I need a quick thumbs up. I think the thing that I'm saying and basically the live which is going, it's a bit slow. So Shobharaj, good morning. Ma'am, can you go a bit faster? Yes, I can. Okay. So let's go with the next question, and the next question goes like this. In the following pedigree chart, the mutant trait is shaded black. Okay, the mutant trait is shaded black. The gene responsible for the trait is what? The gene responsible for the trait is what? Ma'am, is the conversion of phenyl and tyrosine blocked slowly by pH or can secondary mutation amplify the effect? It can. Secondary mutation can also amplify the effect. Okay, Viresh. Sagar, Pakka, you do it. Kritik, hello. Guys, I'm waiting for you guys to answer this particular question. This is the graph. Akshita says the answer for this question is C. Rukmani says it is D. Karaya says it is C. So two people with C. Okay, Kavita says it is B. Okay, hey, B is odd. Okay, 3B is autosomal dominant. Okay, guys, look at the question carefully. Okay. Most of you guys are saying the answer for this question is C. Some people are still stuck with D as well. Let me clear it out. So over here, The answer for this question is going to be, first of all, we have to find whether it's dominant or recessive. Listen, how do you understand that? If at all, in one generation, the trait is not there, but in the next generation it is there. That means there is skipping of generation. You say it is going to be recessive. Okay, over here, the parents, do you think anyone is affected? We cannot see it. Basically, they are not affected. They could be carriers, but they are not affected, right? Yes. But in the progenies, are they affected? Yes. So, do you find skipping of generation? Yes, ma'am, definitely I see that. So, what will be this? This is going to be recessive. If at all I see at least one parent to be affected or one person to be affected in every single generation, I will say it is dominant. But here that's not the case, so it has to be recessive. Now we do elimination method. So wherever you see dominant, cut this off. Dominant is gone. Now you have got option C and D. You have to choose from here. Now, ma'am, I do not understand whether it's sex-linked or it is autosomal. How do I understand that? It's easy, Macha. Listen to me carefully. So over here, if at all you see, okay, in the first generation, girl and a guy is being affected equally. In the second generation, one guy is being affected. But in the first generation, if at all you see, if you see that both of them are equally affected, it's not the males who are predominantly affected, or else probably there were 4 males. Okay, not 4 males. Basically, yeah, there are 4 males. Out of 4, only one is being affected, right? So we can say that it can be autosomal. It might not be sex-linked, because in sex-linked you will be seeing that males are the one which are being affected more. Over here also, if at all you see only one guy is affected, that's not something that by which we can say it is going to be sex-linked. So basically, the answer for this question is going to be autosomal. Why autosomal? Because we cannot clearly see that the males are the mostly affected. And also one very important thing. Usually, okay, in the case of sex-linked disorders, you will find that if the male is affected, the daughter will be affected. From the daughter, it will go to the son. So there's a crisscross pattern being observed, where in sex-linked, always remember, there is a crisscross pattern being observed. From the boy, okay, to the daughter are there, not from the son, from the father to the daughter, from the daughter to the son. This is how it goes. Or from the mother to the son and then back to the daughter. That is how it goes. Okay. Yes, so the answer for this question becomes what? D. If at all it is clear to everyone, can you give me a thumbs up? Ayo. Can you quickly give me a thumbs up? Oh my God. Guys. What is happening over here? Are we all studying? Are we all studying together? Yeah? So why do I see lots of talking over here? Guys, you know what? If at all I am the only one who is keeping blah, 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 I get bored, you will get bored. If you want to make sure that I have constant energy, you guys have to give me that energy. By how? By giving me the answer and things like that. Yeah? Yes? So I'm assuming you are going to give me that, right? Yes, Harsh, I will definitely try it out. I'm not Harsha, sorry. I'll definitely try it out, dear. I'm not sure if at all I can at this particular point of time, but I can give you the most important topics. Yeah? Perfect. Ma'am, but second generation to third generation is affected now. Second, see over here. What is second generation? This, right? These are the parents. Again you see skipping and again you find them. So that is how you have to look. These are the parents, these are the children. This is the parent, these are the children. So if at all you see skipping of generation, that is how we say that it is going to be recessive. Yeah? Perfect. So, yeah. So I am going ahead with the next question that is there. Ma'am, where are you from? Is that a KCET question? I am actually from, I'm born and brought up in Indore, Madhya Pradesh, but my roots are from Kerala. I am from Wayanad. Yes. Which of the following are true about males in honeybees? Come on. Guys, give me the answer for this question. Which of the following is true about the males in the honeybees? They have 16 chromosomes per cell. They produce sperms by meiosis. They can have grandfather but no father. They develop as a result of fertilization. What do you think is the answer for this? Any idea? Yes, Sagar says everyone liked this particular video. Yeah, everyone liked this video. Perfect. Okay. Nauman says the answer for this question is A. Vishwan says B. Deadpool says A. Kishan says A. Okay. Savita A. Hina, Bhuvan, Kavita, Deadpool, Shobharaj, Akshita, and everyone. Most of you guys are saying Dhanush is having a different answer. Okay. Sagaring, Vimal, La, Viresh, Kavana, Nauman, Kyun is also there. Hello. Vidya, Shobharaj, Bhavish. Okay. So most of you guys are saying the answer for this question is A. A is what you're saying. You have to find what is true about the males. Okay, let's see what is true about the males. For that, we need to have an understanding of honeybees. Are we ready? We are going to go inside the honeybee comb. So, Baba, always listen. A female honeybee has 32 number of chromosomes. Okay, so I'm just writing 32 here for the females. What about the males? Males have 16 number of chromosomes. Okay, ma'am, fine, cool. Now we have studied one thing. Whenever organisms make gametes, what do? They undergo a process that is known as meiosis. So here also same thing happens. So Baba, 32 number of chromosomes are there. It undergoes meiosis to produce half number of chromosomes, that is 16. In what? In the gametes. This is the case of female honeybee. But Papa, male guy is only having 16 chromosomes. What will he divide, bacha? There is nothing to divide. So what will it undergo? Will it undergo meiosis? No, there is no option, ma'am. It will only undergo mitosis. It will make cells. Okay, it will undergo mitosis. It will make cells, and all of will be having 16 number of chromosome only. So they have 16 chromosome per cell. Correct. Okay, they produce sperm by meiosis. Wrong. They produce sperm by mitosis. So this is going to be wrong statement. Okay, what do you have to find? You have to find what is true. Now they can have a grandfather but no father. Ma'am, what? So listen to me very carefully. Over here 16 chromosomes are there in the girl. Okay, in the female. Here 16 chromosomes are there in the male. When the 16 and 16 comes together, what do you get? You get 32. And tada, will it be a male or a female? It is going to be a female again. So ma'am, how is male produced? That's the logic. Over here what happens is that this 16 chromosomes which are there, the egg which is there, okay, over here, it basically is not going to fuse with any other sperm out there. Rather, it will say that I am my own king. So basically, it is going to undergo a process that is known as parthenogenesis. And basically, it will transform into what? A male. That is why males have only 60 number of chromosomes, because there is no fusion of gametes. Now you tell me, does this male have a father? No, Baba, because the chromosome came from the mother only. But does it have a grandfather? Yes. Why? Because the female who is there, the mother who is there, obviously is formed by the fusion of what? By the fusion of the sperm as well as the egg. So obviously the female would be having a father. All the females would be having a father, right? So the mother, if the mother has a father, then obviously this particular child has a grandfather. Yes, but does it have a father? No. So they can have a grandfather but no father. This is also correct. Now they have developed as a result of fertilization? No. No fertilization happened over here. Parthenogenesis happened over here. So basically 1 and 3 are the only answer, which is why A is the correct option. Guess A is the correct option. It will come from the grandfather. Okay, got it? I just started for 3 times. I am MPC student, okay, but I had to attend biology and do basic is enough for the exam. Which exam you're talking about? You're talking about KCET itself? Is it about KCET itself? Hiya. Ma'am, in male honey, all somatic cells are germ cells. No. Somatic cells are there, but yeah, germ cells are also formed in such a way only. Yeah, exactly. Cool. If they have grandfather but no father, how do they? Oh my God. Okay. So I'll restrict myself to not saying that. Varsha, say louder. Am I not audible? Let me check. In such a way only. Yeah. What, Macha? I am clearly audible. Please check your earphones. Okay. Yes, ma'am. It's clear. Ayo. Yeah. Perfect. Can we go ahead with the next question, guys? Can we go ahead with the next question? Yes. So most of you have given me a thumbs up, and some people are asking something else. Hi ma'am, I have not prepared for KCET. I have 4 days left. I'll try my best. Can you please suggest me any video to watch? Anu Kavya, if at all you are a biology student, you can watch this. And if at all you're not, basically the PCM top 100 videos which are there, please make sure you watch it in 2x speed. It could be really helpful for you. Yeah? Yes, perfect. Let's go ahead with the next question. Question number 7. In a dihybrid cross between a true breeding round and yellow seeded and true, this is a PYQ by the way, and true breeding wrinkled and green seeded plant, the ratio of segregation of round and wrinkled seed traits in F2 generation is what? Biru, your smile is too cute. Thanks, Macha. Okay. So guys, tell me the answer for this. Thank you so much, Shobhraj. Okay, Aarti says the answer for this question is going to be B. Okay, what about the others? Akshita says B. Deadpool, bacha, what happened? Sia. Kiran says it is B. Norman says B. Kyun and Ramya and Sagaring. And Savita, Panvit. Panvit is there. Hello, hello. Manya, Anand, Dhanush. Anne Maria is also there, Kishan, Tarun. Okay, so most of you guys, Ron, Alex. Okay, I see you, dear. Okay. So, guys, the answer for this question, most of you guys are saying it is B. B is what? 3:1. And you know what, Macha? You guys are absolutely correct. Okay, you are absolutely correct. Let's check it out. So, in a dihybrid cross between a true breeding round yellow seeded and true breeding wrinkled green seeded plant, the ratio of segregation of round and wrinkled seeded is what? So, basically, you have given what? Round and yellow is given to you. Jang-ta-dang. And what? Wrinkled and green is given to you. Wrinkled and green is given to you. This thing we have been studying from so long now, capital R, and small r, small y, small y. First of all, we have to find which one is the dominant trait. The dominant trait is what? Dominant trait is this. And basically you are being asked about, you are not asked about round and yellow organisms or wrinkled and green. You are being just asked about what? How much of them, or basically what is the ratio of segregation for round and wrinkled? Round and wrinkled. So just assume that yellow and green is gone away. Only round and wrinkled is there. That means only capital R and small r is there. This is just like capital T and small t. When we do the, when we do the mixing of this, what do we get at the end? We get 3:1 ratio. If at all you remember, it will be something like this. So capital R, small r, and small r. So three of them are going to be round. These one are round because you have one capital R. This is obviously going to be round. So three of them are going to be round, and one of them, Baba, is going to be wrinkled. 3:1 ratio is something that we can easily find. Now, if at all you have another way of solving this, that would be something like this. Baba, we know the dihybrid cross ratio. What is the dihybrid cross ratio? For 9:3:1. This is the dihybrid cross ratio. What is 9? 9 is going to the dominant character. What is dominant character? Round and yellow is the dominant character. So I'm writing, okay, round and yellow macha is the dominant character. One guy, Papa guy, is going to be the recessive character. What is it? Wrinkled and green. So I'm going to write wrinkled and green over here. Okay, now what about this 3 and 3? What are they? So listen to me. Here it was round and yellow. So this time it is going to be round and green. New combination, macha. Okay, similarly here it was wrinkled and green. So this time it is going to be wrinkled and yellow. So these characters which you see in the middle are non-parental. You do not see in the parents. These are new characters. Anyways, that is not our concern. Our concern is to find the segregation of round and wrinkled. So listen to me. 9 of them are round and yellow and 3 of them are round and green. Here you see round. So 9 plus 3 is equals to what? Is equals to 12. Okay. 3 of them are wrinkled. 1 of them is also wrinkled. So 3 plus 1 over here you get 4. 12:4 is equals to what? If at all you do the ratio, it is going to be 3:1, which is why the answer for this question is going to be B. Is it clear? Hiya. Is it clear? Children? Dun, 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 dun. Tarun, what happened? Thank you, ma'am. I'll give my best, and response made me glad, is it? Wishing you the best. Viresh, if at all you could, what do you say, if these genes were tightly linked with this, no, you will not. Okay, so, ma'am, bye ma'am, guys, please like the video. Ma'am is so beautiful, just enough to distract, oh my god. Okay, so, yes. Everyone says it is clear, which is why we'll go to the next question out there. And what is the next question like? Next question will be like question number 8. Which of the following statement is going to be correct? Change in the whole set of chromosome is called as aneuploidy. Sickle cell anemia is a quantitative problem. Female carrier for hemophilia may transmit the disease to the sons. Thalassemia is a qualitative problem. Okay, tell me. Yes, Norman, I did. Okay. Harshita, you can, dear, you can crack KCET, give your best, okay? Yes, this question came for examination. This is a PYQ. Come on. Which of the following statement is correct is being asked to you? Yo, love you, ma'am. Love you too, Bharti. Okay. Akshita says the answer for this question is B. Okay, what about others? Deadpool says it is C. Norman says C. Swati says C. Parvati C. Savita C. Kiran, Anand, Amulya, Sudarshan, Q. No. Sanjana is also there. Hello. Okay, so most of you guys are saying Varsha and Vimla and Maria, okay, are saying C. And what is C over here? C is female carrier for hemophilia may transmit the disease to the sons. You have been asked what is correct. Let's find it out. Change in the whole set of chromosome is called aneuploidy. Oh my god, it's not known as aneuploidy, that is known as polyploidy. You're talking about whole set, right? That is polyploidy, and poly, P for plant. That is usually seen in the case of plant. Aneuploidy means, Baba, only loss or gain of one of few chromosomes are there, that is known as aneuploidy. Okay, next thing, sickle cell anemia is a quantitative problem. Sickle cell anemia is a qualitative problem. No, why? Because a mutation happened over here in beta globin chain. Yeah, so quality of the hemoglobin that is being formed is problematic over there. It's not a quantitative, it's a qualitative problem. So this is also wrong. What is quantitative, Baba? It is going to be thalassemia. So if you see over here, thalassemia is qualitative problem. Ulta is written over here. No, it is going to be quantitative. This is quantitative because quantity of alpha and the beta chain is going to decide this particular disease. Okay, now what are we left with? We just left with one option, and that is what? Female carrier for hemophilia may transmit the disease to the sons. Yes, Baba. How is hemophilia? For example, this is a female and she is a carrier. Carrier means only one of her X chromosome is going to be affected. How is a male formed? Male is going to be formed when X and Y comes together. From here, the mother can only contribute X. Y is always contributed by the father only, right? So the mother can contribute this X also, right? In that particular case, the child will be having the disease. The son is going to have the disease. Yes. So female carrier for hemophilia may transmit the disease to the sons. This is absolutely correct. So all of you who gave me the answer for this question as C, you guys are correct. Yeah, got it. What is raid? What raid are you gonna do? Parvati, okay, dear, Pakka, I'll go slow. Some people are saying fast, some people are going say slow, so that's why I got confused. Ma'am, I'm selecting engineering for further studies. I have to write bio in KCET. No, dear. What engineering are you looking forward for? Tell me that also. Okay. So children, if at all this is clear, can you give me a thumbs up? Everyone. Afan, yes, I do remember you. Okay. Okay, somewhere in the middle. Okay. Guys, I also want to ask one question. Do you want polls, as in not the polls where your names are written? Obviously, that is not there. But do you want polls over here? For example, A, B, C, D, you have to choose from there. If you want it, you can let me know. Then I'll ask them to do it. I actually said no. Okay, so question number 9. XO type of sex determination and XY type of sex determination are the examples of. XO and XY type of sex determination are the examples of what, Baba? Male homogamity, male heterogamity, female homogamity, or female heterogamity. What do you think is the answer for this particular question? Okay, B. Akshita says B is the answer. Yashwan says B is the answer. And Lavanya says B. Anand says B. Viresh, Dhanush. Okay, most of you guys are saying B, 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 B. GSD. Okay, so the answer for this question, it is going to be B only. Why? You have been asked about XO type of sex determination and XY type of sex determination is an example of. Listen, XO is seen in the case of insects. XY type of determination is seen in the case of humans as well as in the case of Drosophila. What happens over here? The females are going to produce two type of gametes, X and X. Since the same type of chromosomes are being produced or same type of alleles are being put in the gametes, we say it is homogamity. It is what? Homogamety. Female homogamety. Females are going to produce same type of gametes. Okay. What about males over here? Male can produce either a gamete with X chromosome, or probably it can produce a chromosome with no chromosome at all. So two type of gametes are being produced. Yes, this is why it is known as male heterogamety. Males are producing two type of gametes. Okay. What about here, Baba? Here also the females are producing same type of gametes. What about the males? One X and one carries Y. So these two type of gametes are produced by the males, which is why again male heterogamety. The answer for this question is going to be B, as you all said. You all are absolutely correct. Okay. Yeah, example grasshopper. Now can you guys tell me, one of you at least, can you tell me which organism is the example for female heterogamety? Which of the example is for female heterogamety? Any idea? Any idea? XX, so that is kind of very rare, dear. Okay, it will be a syndrome if at all someone is having it. Guys, which organism has female heterogamety? Females are going to be one that produces two type of gametes. Birds, exactly, Aishwarya. Okay, Guk is also saying birds. Okay, Hitesh, Lavanya, perfectly perfect. So Savita, Uday. So in the case of birds, you see female heterogamety. Why? ZZZW is seen in the case of birds, where Baba Z and W. These are the two type of gametes being produced by females. This here females are going to be heterogamous in nature, and W for women. You can remember like that. He. Yes. So anyways, and this is our grasshopper. Say hi to grasshopper, everyone. Yeah. Yes, perfect. Now let's go ahead with question number 10, and that is, a man with blood group A marries a woman having blood group B. The maximum possible blood groups among their progenies are what? Exactly, Vimla, everyone. Dreams, edit, don't spam the chat, Baba. Please. Guys, what do you think is the answer for this? A man with blood group A marries a woman with blood group B. The maximum possible genotypes is being asked. Okay, Hitesh says the answer for this question is A. Lavanya says it is A. And Ramya says it is C. Dhanush says C. Okay. Akshita says C. Anvita, Amulya, most of you guys are saying A is the answer. Shivaji, Sraicharan. Okay. So most of you guys are saying A, which is why I need to look up at this particular question. Okay. And some people are saying, listen, a man with blood group A, okay, marries a woman with blood group B. So always think of the worst scenario, okay? This guy can be somewhere like this, IA, right? Or it can be somewhere like this also. In this case also, it will be A blood group because A is dominant over O. Yeah. Similarly, over here, it can be IB, and it can be IB, IO because B is also dominant over O. Right? Yes. Okay. Fine. Now what are you asked? The maximum possible blood groups are being asked. So maximum possible blood groups are going to come when you fuse this with this. When you fuse this with this, it will always be AB. That's all, because A and B are co-dominant. So if you want to find the maximum, you have to fuse both of them. So suppose over here I have IA, IO and IB, IO. And I'm fusing. This and this comes together. I get IA, IB. What is a blood group, bacha? It is going to be AB. Why? Because A and B, both of them are co-dominant. Okay. What about this and this? It is IO and A. Che. IA and then I have IO. So A is dominant over O, which is why it will be A blood group. Okay. What about this and this? I get IB, IO. B is dominant over O, which is why I get B blood group. Finally, you have IO and IO. IO gives rise to what? O, because there is no one to be dominating. So I get AB, I get A, I get B, and I also get O, which is why the answer for this question is going to be A. So those of you said the answer for this question is C and things like that, check it out. Hello, Veena. Okay, so the answer for this question is A, and if it is clear, give me a thumbs up. Lakshmi, I will be doing first PUC chapters also. Okay, don't worry. Guys, Macha, it's clear na. It's clear, children. Give me a thumbs up if it is clear. Yes. Answer is going to be A. It is not going to be C, okay? Dinky chicka, dinky chicka. Okay, perfect. So I think all of you got it. Guys, if you have any doubt, please stay back. I'll give you answers towards the end, yeah? Okay. Question number 11 goes like this. A pure-breeding pea plant with round and yellow seeds were crossed with pea plant having wrinkled and green seeds, okay? On selfing, F1 hybrid of this cross, 64 progenies were obtained in F2 generation. Find out the number of F2 progenies showing non-parental characters, something that was not there in the parent. What do you think is the answer for this particular question? Hello, Suresh. A pure breeding pea plant, okay, pure breeding, round and yellow were crossed with what? Wrinkled and green. And what did you find? F1 hybrid, you got F1. The F1 also basically you crossed them, and then finally you got the F2 generation. Now you are saying that 64 progenies were obtained. Okay, you are saying 64 progenies were obtained in the F2 generation. And you have been asked this question that how many of them shows non-parental character. So those who have been with me, who has solved the questions with me, knows the answer always. Okay, so A. Most of you guys are saying A is the answer. Is A the answer? Let's check it out together. Okay, it's 24. What you're saying? Let's check it out. So as I said, 9:3:3:1 ratio is for dihybrid. Yeah. Okay, 9 of them are going to be the dominant character, round and yellow, parental character only. Wrinkled and green over here, recessive character. Okay, now I told you these two are going to be the parental characters, but these two are going to be non-parental, which is do not see in the parents. So over here, round was with yellow, but this time round is going to be with green. What about that? Here, wrinkled was with always yellow, but this time it is going to be with was always with green. This time it is going to be with yellow, right? Yes. So out of 9 plus 3 plus 3 plus 1, that is equals to 16. Out of 16, we see that 6 of them, 3 plus 3 is equals to what? 6 of them are non-parental. And you have been asked, if out of 16, 6 of them are non-parental, what will be the case if 64 progenies are there? How many non-parental characters will be there? So what do? You multiply it with 64. This goes with this, you get 4. 6 into 4 is equals to what? 24. The answer for this question is going to be 24. Got it? Yes, if at all you have been asked about how many of them are going to show parental character, what do? This and this is parental, so 9 plus 1 is equals to what? 10, Baba, is equals to 10. 10 after 16 are parental, into 64 you do. So basically you will come to a conclusion that 40 of them are parental, right? 24 are non-parental and 40 will be parental. It's as simple as that. Got it, Macha? Or did not get it? If yes, give me a thumbs up. Anyway, you guys are talking. Sanjana. Ajay says, explain it once. Okay, I can explain. Listen to me carefully. You have been asked in dihybrid cross, how many of them are going to show non-parental character? If you are saying that after dihybrid cross, you are getting 64 progeny. So what do? The thing that we already know is this particular ratio. And we know that 9 of them are going to show the parental character, dominant character. 1 of them is going to show parental recessive character. Only these 2 guys are the ones which are going to be non-parental. Okay, so if at all we see 3 plus 3, 6 of them are non-parental. Out of what? Out of 16, 6 of them are non-parental. Now you are being asked about 64, so what do? You do 6 by 16 into, you multiply it with 64. You will get the number of them who are non-parental in 64 progenies out there. So your answer comes out to be 24. 24 are non-parental. Yeah? Yes? Yes. Ayyo. Let's go. Question number 12. This is going to be the last question of this particular chapter. Both male and female have normal vision, though their fathers were colorblind, and the mothers did not have any gene for colorblindness. The probability of their daughter being colorblind is what, Baba? Hello, Deepak. The probability of their daughter being colorblind. Both male and female have normal vision. Okay? We have to decode this. Male and female have normal vision. So whenever we say that females have normal vision, you have to think twice because they can be carriers. Males, if we say that they are normal vision, they are normal only. Okay, just like in reality also, no, we say that we are okay, but internally we might not be okay. Okay, we might show that later on. So anyways, so both, and we might torture you as well. So both male and female have normal vision, though their fathers were colorblind. Now this is the thing. So father was colorblind. Okay, let's make father is colorblind. For father being colorblind, one X chromosome should be affected, okay? And mothers did not have any gene. So mother is Papa, guys. She is normal over here. Okay, let's make the son and daughter first. Then we will be able to understand things better. So I am going to make that. This and this crosses. I get XC. Okay, she is a carrier. She's a female and she is a carrier out there. Okay, what about this? Again, I get this. She is going to be a female and she is going to be a carrier. Why? Because she has one chromosome. Okay. Now, what about this and this? The guys which are there, Baba, however you do, they are going to be normal only. And this is what it said. Both male and female have normal vision. You see, the female has normal vision, but she is what? She is a carrier. She is a carrier. Always remember. Okay, we have got whatever thing we wanted to have. Now, the probability of their daughter becoming colorblind is. So, you are being asked, if a woman with normal vision, but we know that she is not normal, she is a carrier out there, and a man with normal vision will be always like this. How much probability of their daughter, what is the probability of the daughter becoming colorblind? So let's check it out. So for daughter to be formed, X chromosome from here, X chromosome from here. This is a normal daughter, okay, female who is going to be normal. This and this comes together, basically X and XC. Over here, she is going to be a carrier. She's a female, but she is a carrier, okay. What about the males? Males are going to be normal. So if at all I see none, okay, if at all I see none of the daughters, Baba, are having the disease. None of the daughters are colorblind. Why? Because all of them over here, one of them is a carrier and one of them is normal. Right, so the answer for this question is going to be A. It is going to be 0%. Okay. Got it? So how many of you said A? Most of you were saying A only. Okay. Some people were a little bit distracted. Is this clear? You have to solve questions, okay? You have to solve questions like this. From color blindness, you may get question. Oof. Children, I'm asking you, listen, I feel very bad when I'm not able to interact. You know, you guys are my fuel. If at all I do not interact with you, all my energy goes up. So, because there are so many chats coming up, even I am not able to deduce who is saying what and a lot of things. And yes, explain once again. I can explain once again. Okay, let me explain once again. Everyone listen to me out carefully. I know it's a time-taking process, but please listen. This is an important question. First of all, whenever you get questions like this, you have to find what is the parents, what do you call, genotype out there. Then only you will be able to answer things, right? So, both male and female have normal vision, though their fathers were colorblind and mothers did not have any gene for colorblindness. That has been given. So these two are the parents out there, and talking about them, it is clearly mentioned that they have normal vision. But always remember, when you talk about males and when we say that they have normal vision, obviously they have to be like this. But in the case of females, when you say they have normal vision, they can be like this, or they can be a carrier as well. They can be like this also. Okay, they will be having a normal vision even though they are a carrier. So what do? We need to find whether the mother is like this or like this. First thing, that is what most important thing. How do we know that? Basically, we have to go and look what is there here, her mother and father like. So over here it is given that the father was colorblind. So father was colorblind, that means over here the chromosome was like this, and the mother did not have any gene for colorblindness. That means she was normal. Now over here, Baba, if at all you make the female, this and this crosses, you get the female. One X chromosome is affected. That means yes, she is a carrier. If you cross this and this again, since the father has only this to contribute, this female is also going to be colorblind. So we know that basically the mother is like this. The mother is going to be like this. And now we have to do what? We have to do the cross. So over here mother is like this, and father, it's already said that the male is having normal vision. And then when we do the crossing, if at all we want to find the daughter's probability of getting color blindness, we cross X and X, it is going to be a normal female. We cross this and this, it is going to be a carrier female. This is going to be what? A carrier female out there. But both of them, are they colorblind? No, none of them are colorblind. So can I say 0%? Yes, I can say that. Yeah? Yes. I hope now it's clear. Yeah? Yes. Guys, Are the questions clear from this particular chapter? This is an important chapter. And also one more very important thing, once this live ends, go through the PDF, go through each and every question, understand the concepts which are there. Okay, since you guys are somewhat saying fast, I might not be explaining the other part of the story which I meant to explain. Okay, so please, ma'am, if father is not normal, then obviously what is going to happen? If father is not normal, he has color blindness. So obviously he'll pass the gene, and the mother can also pass this particular gene. So there are chances that the female can have the disease, but the mother, if she's a carrier, then again, if it's something like this, when this and this comes together, it's like this. But when this and this comes together, she will be just a carrier. She will not have the disease. So 50% of the females can have the disease. Cool da. Okay. Perfect. So I shall go ahead with the next question out there. And ma'am, Y and XC are the male carriers. There is no male carrier. In sex-linked disorders, there are no male carriers. Males either have the disease or they are normal. Okay? Yes. Cool. So let's go ahead. Now guys, listen to me very carefully. The next chapter which is there is going to be of PU1. So what I have done is that in between I have put PU1 chapters also. So in that way, everything is going to be cleared. Okay, ma'am, in WhatsApp PDF is not coming. Baba, just roll down a bit. At the top it is there. That's why we are not putting it. Okay, we'll update it once more. Okay. If mother is not normal, again you will get the disease. She will pass the disease to everyone. Yeah. Yes, cool. So the question goes like this. Question number 13. Read the given statements and select the correct options out there. And it goes like this. Dash is followed by zygote of prophase 1. So over here, you have to check, okay, what is going to be filled up over here. 1 is followed by zygote of prophase 1. So what is 1 over here? Crossing over is an enzyme-mediated process that involves enzyme dash. So what is that particular enzyme? 2. Okay, meiosis 1 is dash division. Basically 3 should be meiosis 2. Meiosis 1 is dash division. You have to fill that. X-shaped structure called chiasmata are seen during dash stage of prophase 1. What do you think is the answer for this particular question? C. Okay, Panvit says C is the answer. Lavanya says C. Dhanush and third row, ma'am. Okay, Norman says third row. Deadpool says C and Sanjana says C. Parvati C. Dhanush says C. Okay, Amulya also says C. Okay, so let me check it out. Lakshmi, Komali, hello. So C is what? C is going to be this, and we need to check out whether the answer is correct or not, right? So let's do it. Listen, the most easiest one to find among them is what? Crossing over is done with the help of an enzyme 2. So crossing over is going to be always done with the help of enzyme recombinase. So basically this and this option is there, right? So I can exclude these two options which are there. I can just cut off these two options. Now I'm left with these two. Okay, fine. Now meiosis is what division? Meiosis 1 which is there, you know, when a normal cell is there, when it undergoes meiosis 1, we say it's reductional division. Why? Because the number of chromosome becomes reduced to half. It becomes 23 over here. So it's reductional, right? Now over here, if at all you see, this is the option that is having reductional. This is the option that is having recombinase. So which is why the answer for this question is going to be C itself. It is going to be C. This is a very important concept. Okay, it's a very important concept. Got it? Why answer is C? Ma'am, where is the first PU chapter link, video link? Dear, in the 15 days challenge, you can watch. I did one video for first PU. That would work. Otherwise, I am doing the questions over here. You can just check out from here. Guys, I have a question for you guys. Do you need an understanding of all the phases, as in just prophase 1, meiosis 1? Because this is where I have seen most of the questions have been coming from. This is a very important concept. Very important concept. If you need it, you let me know right now. Okay, I'll explain. So listen. Yes, I'll explain it to you. Do not miss out on this. Probably you can get that 1 mark from this particular chapter. Yeah. So listen, meiosis 1 is there. In meiosis 1, you would have prophase 1, metaphase 1, anaphase 1, and then telophase 1. And then you will have meiosis 2. That is how it goes, right? Yes. In prophase also, it has been divided. And this particular thing is something that is mostly being asked. Question is based on this. So it comes like this. Prophase 1 is divided into multiple phases. Lazy zebras play during day. Lazy zebras play during day. So those of you who have attended my live before, you know this. Lazy zebras play during day. L stands for what? Blah, 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 blah. Leptotene. Leptotene. This is what? Okay, just a second. This is leptotene. Leptotene. Zygotine. Picatinine. And diplotene, and finally we all die once, right? So dikinesis comes at the end. So these are the stages that you need to remember. Okay, now we go ahead with this. What happens in leptotene? Always remember, your chromosomes which are there, they are not initially chromosomes. They are found in the form of what? Chromatin fibers. They are found in the form of chromatin fibers. So basically these chromatin fibers have to be condensed to form what? To form the chromosome. So chromatin condensation to form the chromosomes happens in which stage? In the leptotene stage it happens. Now you remember one more very important thing. So when we say that basically I have got the characters from my mother and my father, what do I mean by that? That means that egg carries certain number of chromosomes, the sperm carries certain number of chromosomes. They are going to come together. What do you call these chromosomes which have same type of alleles at the same location? For example, I say I have an allele for height over here. I also have an allele for height over here in this particular chromosome in the same location. I call these chromosomes as what? Homologous chromosomes. They are going to pair up, okay? They are going to pair up because they have similar things. So I call them as homologous chromosomes. And I hope you know that before mitosis or meiosis starts up, what is going to happen? Preparation is going to happen and they are going to form copies of it. They make the copies of it. So basically, is it one chromosome now or two chromosomes? It's only one chromosome. These two things that you see over here, they are what? Chromatids. But since the centromere is only one, we call it as one chromosome only. And these two chromatids which are there, we call them sister chromatids. Why? Because they are joined together. Okay. What about this? What about this? If I talk about this chromatid and this chromatid, they are non-sister chromatids. They are what? Non-sister chromatids. Why am I talking about this? Because this is going to be explained up over here. So what happens over here, Baba? Chromatin got condensed to form chromosomes. Now we can visibly see chromosomes over here in leptotene. These stages, the unique features are important for you to remember. Now what happens in zygotine? In zygotine, Baba, these two guys come together. These two guys which are there, okay, the homologous chromosomes, they're going to come together and they are going to be glued up together. Say they're like, Macha, you and me are same. So basically they form a synaptonemal complex over here. They form a synaptonemal complex over here. We say that this process is known as synapsis. It is known as synapsis. And basically, what do you call the whole thing when all the chromosomes comes together over here? You say that this is the formation of bivalent. Bivalent means one chromosome, second chromosome. So bivalent chromosomes are being formed over here. So these features are important for zygotine. First is chromosome condition for leptotene. In zygotine, synapsis or synaptonemal complex formation happens and basically they form bivalents. What happens in picatinine? Now my dear Macha, just listen to me very carefully. If at all they are very close, they are going to exchange certain things. They will be exchanging certain things. So what happens over here? You have the chromosomes, yeah, and this chromosome, and in between that you have what? Synaptonemal complex. Basically crossing over is going to be taking place in picatinine. You will see what? Crossing over. And crossing over is being done with the help of what? Recombinase enzyme. And 8 questions, or basically I think 7 questions have come from the same concept. Recombinase crossing over. Recombinase crossing over. Where does it see? In picatinine stage, you are gonna see this. In picatinine, you are gonna see this. Now what happens in diplotene stage, which is the next stage out there? Diplotene stage, Baba, breakup happens. Breakup, sad things. Now you only think when breakup happens, still there will be some attachment here and there, right? So here also the same thing happens, Baba. What is the thing is that the chromosomes which are there, they're gonna separate it. Why? Because the synaptonemal complex which was there in between, it went. Okay, the synaptonemal complex, it got dissoluted. We say it got dissoluted. So ma'am, how are they still connected? As I said, at certain points they touch each each other. They are still joined. What do you call these points as? You say this is the formation of chiasmata. You say it is chiasmata. So after breakup, they are still joined at certain points. Now what happens, Baba? The last step, that is dikinesis, where you are going to see that basically they are going to be so now when the breakup happened and now you are moving away from each other, it leads to the movement of chiasmata to the terminal position. It will move towards the end. So we say terminalization. Okay, terminalization of what? Chiasmata happens in dikinesis. These are very, very important things. You can easily score 1 mark out of it if at all you remember it. This is very, very, very important. Yeah, got it? Got it? So over here, the answer for this question, dash is followed by zygotine of prophase 1. Dash is followed by zygotine. That means what comes before zygotine, Baba? Before zygotine, it is going to be L. L is for leptotene. Lazy zebras play, right? Leptotene. Crossing over is an enzyme-mediated process that involves enzyme what? Recombinase. Meiosis 1 is what? Reductional division. And meiosis 2 is kind of equational division. Finally, X-shaped chromosomes called chiasmata are seen during dash stage of prophase 1. X-shaped structure called chiasmata. Chiasmata is seen in where? In diplotin. Breakup stage, right? Diplotin. So which is why the answer is this. Concept is clear? Guys, is the concept super clear to you, everyone? Clear, ah? Can we go ahead? Thank you so much, Manya. Okay. Hello, Ajay. Guys, if it's clear, can you quickly give me a thumbs up? Jinay, we will be doing all the questions, Macha, don't worry. Okay. Yes, perfect. Thank you so much. Let's go ahead with the next question. Cells in the quiescent stage, this is a PYQ. Cell in the quiescent stage, that is G0 phase, always become cancerous, show indefinite proliferation, remain metabolically inactive, and remain metabolically active. What do you think is the answer for this particular question? What do you think is the answer for this particular question? Guys, don't fight amongst each other. Listen, we are not for that. That is not even our ultimate goal, right? We have to score good. Yeah? So, yes. Okay. Lavanya says the answer for this question is going to be D. Lachi. Hello. Lavanya says D. Panvit says 4. That is D. Okay. Sanjeevani says the answer for this question is D. Shreya says it is C. And. Karthik, thank you so much. Deadpool says D. Why is A, B, C, D option given to me? Okay. Okay. Raksu is also there, Chase Master is there, Ayush is there, Norman and Maria. D is what? Remain metabolically active. Yes, Baba. So the cells that enter the G0 phase, we say resting phase or quiescent stage. This is where the cells are not dividing. Okay, they are not going to divide. But always remember, they do not divide does not mean that they are not active. They are metabolically active. Okay, remember that. It's a very important thing. They are metabolically active. This is an easy question, very straightforward question. So D is the answer for this. Yeah. Yes, perfect, Vijay. Kyun and Kidzone is also there. Hello. Yeah, Afanullah, perfect. Let's go ahead with the next question. I'm not asking for the thumbs up now because I know it's too much. Question number 15. The complex formed by a pair of synapsed homologous chromosome is called synapsed homologous chromosome. The complex which is being formed with the pair of synapses. There are two homologous chromosomes. They are together. What do you call this as? Come on, guys, give me the answer as 15 ABCD. Listen, so here is the problem. Is PYQ repeated? Macha, the PYQ is not repeated. The concept could be repeated any time. So we have to be really, really careful, okay? B. We now have subscribed. What is happening over there? Subscribe here. Okay, so Lavanya says B, Renuka says B, Kishan says Amulya B, Lavanya, Sanjana, Kidzone, Sinch, and Samarpreet, okay, Samarpreet, and Min 17. Guys, you have so creative names such that I cannot read it. Monisha, Sassy, oh my god. Okay, so B is the answer for this particular question. B is what? Bivalent. We just discussed about it, no? So these two chromosomes which are coming out here, this is one chromosome with one centromere, this is another chromosome with another centromere. Basically, when they come together with synaptonormal complex in between, I will call this as bivalent. I will call this as bivalent, which is the answer for this particular question. Okay, got it? Yes. Now, give me a thumbs up, then only we'll move ahead with the next question because I think, oh, yeah. Give me a thumbs up and then only we'll move ahead with the next question. Where to find PDF? Baba, if at all you join the WhatsApp channel, no, you will be finding the PDF out there. Okay, after this class, I will only put the PDF. Yeah? Yes. Guys, much a thumbs up. Okay, perfect. Next question that is there. Thank you. This is from this chapter, Cell, the Unit of Life. So obviously we are not going to spend too much time in PU1 chapters. Here and there you will find 3 to 4 questions out there. That's all, yeah? Because you get 2 or 1 question, right? In the below diagram, identify the part which connects the peripheral microtubules to the central sheath. This is the PYQ. Okay, so your question goes like this. This is a diagram that is given. Where is the diagram? Yeah, this is a diagram that is given to you. It's of microtubule. You know, flagella, cilia is formed of microtubules. Now outside you are going to find what? Peripheral peripheral microtubules out there, and you are being asked that over here it is connected to the central sheath with the help of what? So these are microtubules, this is the central sheath. What is the thing that helps in the connection of it? And over here you have got the options. Some people are saying 2. Some people are saying B. Plasma membrane. Why did you say plasma membrane, macha? Look at the question carefully. Which connects the peripheral microtubules to the central sheath. Okay, so I think plasma membrane is something that everyone can relate to very easily, which is why you are saying that is the answer. Okay, the answer for this particular question is going to be radial spoke. And with how much confidence is everyone saying B? Radial spoke is what? So over here, if at all you see, okay, if at all you see, these are the tubules. How many of the tubules are there? You see, ma'am, many gold things are there, we do not know. So 9 of them are there. If at all you see, these things are counted to be 1. They are doublet actually. They counted 1. So 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of them are there. You see 9 plus 2 arrangement. You might have heard about it in flagella and cilia, right? So 9 of the microtubules are arranged outside. 2 of them are inside. Yeah, you have been asked this question that the outside one are connected to the inside one with the help of what? With the help of these things that you see, Baba. These things are what? These are the radial spokes. How many radial spoke will be there? Each doublet is having one spoke out there. So 9 of them will be there. That is the question. What is the one that connects? The answer for this question is going to be A. So with the confidence that you gave me the answer right now, if at all you have given there in KCET, you would have confidently lost 1 marks out there. Yeah, so please make sure you know the structure of this. This is very important. Yeah, axoneme usually has, axoneme is the structure that you call it, and axoneme has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules out there. This is known as 9 plus 2 arrangement. Yeah, kids zone, wishing you the best. So macha, got it? Concept is clear? If yes, can you give me a thumbs up? Guys, if at all you think that the concept is super clear to you, give me a thumbs up. Thank you, Norman. Norman is like, oh my god. Okay. Ma'am, you are too active. I doubt can you teach full day. I have never seen. What do you mean by that? For everyone who has studied from me from 2nd PUC, you guys know how much I have taught, yeah? So thank you so much for the thumbs up. And Uday is saying, please explain again. Can I give 1 minute to Uday, please? Okay? So it goes like this. Listen, flagella, cilia, if at all you cut it open and see it, it looks like this. It is made up of axoneme. And axoneme, Baba, is made up of microtubules. How many microtubules are outside? 9 of them are outside. You might see so many of them, but actually they count as pair. Doublet is what you call them. So 9 doublets are outside, and basically you have been asked what connects these doublets, peripheral doublets, to the internal microtubules. There are 2 microtubules inside and 9 of them outside, right? What is something that connects both of them? It is going to be the radial spoke. This structure that you see over here, these are the radial spoke that is going to be connecting the outer one to the inner one. That is a question. Yeah. Yes, that's all. Okay, wishing you the best, Hina Kausar. Perfect. Guys, next question, question number 18. Which of the following statement is correct with reference to vacuoles? Now, the reason that I put abrupt PYQs only over here is that, I'll tell you. Today, you're going to get a video on the most repeated concepts and topics, okay, in YouTube. So there I found that from this chapter every year from new places questions are coming in. So anyway, we cannot guess what is going to come, but still, yeah. Which of the following statement is correct is being asked about vacuoles. So first statement, it is membrane bound and contains storage proteins and lipids. Okay. The next one is saying it is membrane bound and contains water and excretory substance. C says it lacks membrane and contains air. D says it lacks membrane and contains water and excretory substance. What do you think is the answer for this question? You're most welcome, Uday. Okay. 18, the answer for this question is saying B. Okay. Panvit says it is 2. Hello, Sujata. Hello, hello. Hitesh, Mudik Nishak, okay, and Shravan, Veena, Amulya, Shreyas, everyone, most of you guys are saying the answer is B. B is what? B is this. It is membrane bound and contains water and excretory substance. Is it? Yes, the membrane of vacuole is what you call as tonoplast. So this big vacuole that you see in the case of plants, Baba, it is covered with tonoplast. And what does it store? It stores water and excretory substance. So ma'am, here also it is written membrane bound. Yes, but here it is written that it stores proteins and lipids. That is a function of plastids, no? Plastids, you remember, elioplast stores fat, okay? Aleuroplast stores protein and stuff like that. So obviously it is not going to be this. So only answer for this question is going to be B. Here it is saying it is lacking membrane. Obviously it cannot lack membrane, which is why other options would be wrong. Okay. Yes. Yes. Okay. So let's go ahead with the next question out there. Guys. Clear na? If yes, give me a thumbs up. If yes, give me a thumbs up, everyone. Duration, ma'am, until our 103 questions are being completed, it will be going on. So please do not ask me duration. Okay, I'm trying my best to be as explain as much as I can with too much speed as well. Okay, where are you teaching from? I'm teaching from Bangalore. Yes, love you, ma'am. Love you too, Chetan. Okay, guys, if it's clear, just give me thumbs up because we are gonna enter the next chapter, that is molecular basis of inheritance, one of the most important chapters out there. Yeah. Yes, perfect. Now, can you give me 1 minute? I will just go bring my bottle. I have not brought my bottle and my head is paining so much. If possible, just give me 2 minutes, not 1 minute, 2 minutes. I have to fill the bottle also. Okay, so 11:16 is the time right now. We will meet by 11:18. Yeah, I will just bring my bottle and we'll go with the next one. Yes. Wait, okay. Yes, hello. Guys, am I audible? Visible? Everything is clear? Breakfast talks are ongoing now. Ma'am, it's 11:20. Yes, I'm sorry. Okay, so welcome back and let's go ahead over here. Sorry. Next chapter that we have is molecular basis of inheritance, my dear children. So let's go ahead with this particular question. Which of the following statement are correct with reference to the prokaryotic genome? Which is the statement that is correct with reference to prokaryotic genome? This is a very important question, okay? Monocystronic structural genes, introns absent in structural genes, transcription and translation are coupled processes, primary transcript undergoes splicing, and only one RNA polymerase is present. This is 2025 PYQ out there, and very important as well. So 2. Panvit is saying 2 is the answer for this question. What about others? Lavanya says B, Norman says B for the answer. Okay. Malamma and Komali, sorry, Lavanya, Deadpool, most of you guys are saying B. B is what? B is what? Only B, C and E are correct. Let's see it. First of all, you're talking about prokaryotes, right? And the first statement is that they have monocystronic structural genes. Baba, prokaryotes will go down. Obviously, they do not have monocystronic structural genes. They have polycystronic. Ma'am, what do you mean by polycystronic? Listen to me carefully. When I talk about one gene, for example, you might have seen in lac operon. Okay, one gene has three things. Z, Y, A. Basically, they have three genes out there, and they code for 3 different proteins as well. And it is found in one structural gene itself. This is known as polycystronic arrangement, where multiple cistrons or multiple genes coding for some proteins are being put together. This is found in the case of prokaryotes. So P for polycystronic, P for prokaryotes. Always remember that. Now, do they have monocystronic? No. Monocystronic is found in the case of what? Eukaryotes. That is us. We are having only one gene codes for only one protein. That is how it goes. Okay. Introns absent in the structural genes. Introns which are there, do they have introns? What do you mean by introns? Baba, in the DNA which is there, no, there are introns which codes for nothing, and exons which codes for the amino acids out there. So in between the exons you have introns in the DNA. Now you know transcription happens, the DNA is going to form what? DNA is going to form H and RNA after transcription. So in that particular case, will H and RNA will have introns? Yes, everything is getting transcribed. Okay, so I and E, everything will be there. But now, when the H and RNA has to form something that is known as mRNA, which is further going to undergo the process of translation to form proteins out there, the mRNA says, Macha, you please keep your introns with you, just give me the exons. I do not need introns out there. So they undergo a process that is known as splicing and finally remove the introns, and you get only a strand of exons, because that is the only thing that you need for formation of proteins. Now this is in the case of us, eukaryotes. Okay, this is also eukaryotes. Prokaryotes do not have any introns, so they do not have to do all of this drama out there. Okay, they are very simple organisms. Okay, now so A and B statements, both of them are going to be okay. So over here it is written that introns are absent in structural gene. That is correct. Introns are absent in structural genes in the case of prokaryotes. Now, transcription and translation are coupled processes. What do you mean by that? They are coupled. That is what do you mean by that? No, Baba. What is the thing? That is, in the case of eukaryotes, you know, we have cytoplasm, we have nucleus and everything. Transcription happens in the nucleus. Translation happens in what? The cytoplasm. So it is not a coupled process. Not everything happens together in our case. But in the case of prokaryotes, there is no nucleus, no cytoplasm. So sorry, new nucleus is not there. There is nothing that is membrane bound. So everything happens together. Transcription, translation can happen simultaneously, which is why we say it is coupled processes. So this is also correct. Primary transcript undergoes splicing. What do you mean by splicing? Splicing is this only, removal of introns. If they do not have introns, why will they undergo splicing? So this statement is going to be wrong, right? Because they are saying it undergoes splicing. Now, only one RNA polymerase is present. Yes, in the case of prokaryotes, only one. As I said, they are not complicated. They are simple organisms. They do not have multiple polymerase. They just have one. So which is why this is also correct. So as you guys said, B is the answer for this question. Only B, C, E are the answers. Got it? Yes, Kavita, it takes place in the cytoplasm. Guys, what are you doing over here? What are you doing? Who is copycat? This, that. Don't do, guys, if at all you are here for some bakar only and not for study, okay, I do not entertain that. If you do bakar with me, okay, during studies, it's fine, but not amongst each other. Don't do that. Introns absent in structural genes. What do you mean by that? That is what you are asking. As I said, Baba, we have introns. The non-coding regions are there. In the case of prokaryotes, they do not have it. They do not have the non-coding regions. They only have the coding regions are there. So introns absent in structural genes, that is correct. They do not have it. So yes, it is absent. Yeah? Yes. Ma'am, you're cute today. So yesterday I was not cute. That is what you mean? Okay? So yes. Make video on scholarship, definitely will be trying for that. Yeah. So guys, if it's okay, give me a thumbs up. Suresh, love you too. Okay, give me a thumbs up. Let's go ahead. Thumbs up needed. We have to go ahead. Okay, perfect. So Malendra is saying D. Can you explain B? Dear, I explained B, right? B was this only. Introns absent in structural genes. In the case of prokaryotes, you do not have introns. You just have exons. There is no introns in their DNA, so obviously that is not going to be going ahead also, right? And what do you mean by mature RNA? This RNA, listen, everything is happening inside the nucleus in the case of eukaryotes, right? And then only mRNA moves outside the nucleus. Why? Because mRNA has all the important codes to form what? Proteins. Now, it should contain only the important things, not the unimportant things, which is why splicing has to be done. The hnRNA which is being formed over here, it undergoes splicing, introns are being removed, you form the mRNA and that mRNA goes outside. This is the case of eukaryotes. Yeah? Yes. Next question. Let's go with the next question. And over here, question number 21. The codon on the mRNA are CAU, CCU, AAA and CUG. Identify the correct sequence of amino acid. Come on guys, get it up. So this question I did not solve until this particular date with you guys. But this is high time to do so. Any idea? Ma'am, your name? My name is Jasmine Macha. Okay. Guys, what do you think is the answer for this particular question? Come on. The codon on the mRNA. The codon on the mRNA basically is what? CAU, CCU, AAA and CUG. Identify the correct sequence of amino acid. That means you have to guess what tRNA comes over here and what tRNA carries what amino acid. For that, basically, you should know what this particular thing code for. That is the first question, right? So most of you guys are saying A. Is answer A only? Let me check it out. Histidine, proline, lysine and leucine. Is that the answer? Yes. But how did you get that answer? Everyone knows genetic code? Everyone knows genetic code? If at all you do not know, listen to me very carefully. This particular thing that you see over here is super important. And I'll tell you a way to remember it as well. Yeah, if at all you do not know. So the first one is you can arrest gangster. That means UCAG. Okay, UCAG. So first thing that you have to do is make a table where you write UCAG on one side. Similarly, you write UCAG on other side. Okay, and finally you put it over here also. UCAG, 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 UCAG. Cool. Now we are going to do the calculation. U from here, U. And then the first one is going to be U. So first one is going to be U. Then it comes UUC, UUA, UUG. That is how it is going to go. Okay, now listen. The problem comes where you have to remember the codes as such. So there are 20 amino acids out there. And there are few unique things that you have to remember that is generally there. And that goes like this. That is, for methionine, there is only one codon, and that codon is what we all know. It is AUG. It's doing a dual function. It is an initiator codon as well as it codes for methionine. Okay. Similarly, there is one more thing, tryptophan, UGG. UGG is for tryptophan. No other codons are there for tryptophan. So AUG is for methionine. UGG is for tryptophan. This is one unique thing out there. Apart from that, we know that there are three stop codons: UA, UGA, and UAC. UA, UAG and UGA. Basically, these three which are there, they are stop codons. This is also something that we know. But what do we do when we get a question like this, where we have no idea about histidine, proline, lysine and leucine? That's when we have to remember things. Okay, we have to remember things. And I am not saying that with this particular thing you will be able to remember it directly. No, you have to do your workout separately, but I can help you to remember it. Yeah, so are you ready for that? Are we all ready to learn how genetic code works? If yes, tell me yes right now. If yes, tell me yes. Assalamu alaikum Bashar. Okay, walaikum assalam is what you call this, what do you say, reply back, right? Yes. Yes, share shortcut. So let's go ahead and talk about it. Yes. Okay, so over here, the first one is phenylalanine, leucine, isoleucine, methionine and valine. This is how it goes, right? Phenylalanine, leucine and isoleucine, methionine and valine. You can remember it as fill leaf in my wallet. So this is fill leaf in my wallet. So wallet is separate, all four of them comes up at the end. Here methionine you already know, leucine has a bigger thing, phenylalanine just has two codons out there. Okay, and in between comes leucine. So fill leaf in my wallet. This is the first thing. Okay, now the next one is simple, very easy, and that is serine, proline, threonine and alanine. How do you remember it? Serious, professor, true, the alarm. Serious professor through the alarm. Serious professor through alarm. So that is serine, proline, threonine and alanine out there. Finally, you have this one also. Okay, I hope this is getting feeded somehow. We will be solving question. Do not worry. We will solve the same question with this knowledge. Okay, now you have tyrosine, stop codon, histidine, glutamine, asparagine, lysine, aspartic acid and glutamic acid. So if at all you see this particular track, everything is 2 in number. Everything is 2 number. Finally you have aspartic acid and glutamic acid. These are two acids out there. Okay, now you have to split them into 2 and that's only how you can remember. Tyrosine stopped his gun. So tyrosine was there, he stopped his gun. So he had a gun, he stopped it. Tyrosine stopped his gun. This is halfway. The next half is basically meaning nothing, but it is going to be of use. Asian-like aspartic glue. Now you can come up with something, obviously. Asian-like aspartic glue. Asian-like aspartic glue. So tyrosine stop his gun and Asian-like aspartic glue. Obviously that does not mean anything, but yeah. Okay, and finally you have this particular tract. So first one is fill leaf in my wallet. The second one is serious professor through alarm. The third one is tyrosine stop his gun and Asian-like aspartic glue. And finally the last one, if at all you see, is kind of little bit tricky out there, and it goes like this. You have cysteine and over here cysteine, over here you have your stop codon and you have tryptophan. Tryptophan and cysteine, I hope this part is going to be easy. Then you have arginine, then you have serine, then you have again arginine, and then you have glycine. So if at all you see, serine we have studied here also, please do not forget that. And we are going to divide this and we are going to divide this. So first let me divide this. Sister, stop tripping. Okay, what is it? Sister, stop tripping. That is the first part of it. The next part of it is like this: arginine, sir, arginine, glycine. It does not mean anything, but you have to remember like that: arginine, sir, arginine, glycine. So if at all you see both of them, two full columns are occupied by arginine and glycine, and in between that again serine and arginine is taking some space. So arginine, sir, arginine, glycine. This is the last thing which is out there. Yes. Yes. Now I know it can be tricky, I know it can be difficult, but still we need to solve it. And with this particular knowledge, if we go up over here and we try to solve it, CAU, which column will it come? Always remember, you have this thing, UCAG over here, UCAG over here. So we know that the first one, CAU means, for example, it should be coming somewhere in the second column. CAU, right? CAU comes in what? Actually, the third column. C and then A and then U from that particular side. So it comes in third column. That is the first thing that we need to remember. Okay, that is the first thing that we remember. And in the third column, what all things were there? Tyrosine, stop his gun. If at all you remember, tyrosine and stop were there, his gun was there. So HIS, his is for this particular thing itself. You got it? I know that it is not going to be very, very easy, but with this you are going to be learning it. This is present in the PDF. Yes, it is there in the PDF. JJ ma'am, love you too, dear. Okay, ma'am, did you have your breakfast? Yes. But dear, I don't think I am really fine. I'm having a, what do you say, headache for no reason. I think I slept late, that's why. But anyways, children, I hope that question is clear. I hope we can go ahead with the next question, and that is this. In the given figure of the lac operon, so this is the lac operon, identify the components and the enzyme. This is a brand new question. This is not a PYQ. So let's go ahead and try it up. Okay, let's try solving it. So X is this, Y is this, E1, E2, E3 is being mentioned over here. X is this, Y is this, E1, E2, E3 is there. You have to choose which one of them is correct. Prajul says, ma'am, what's the secret behind your beauty? Oh my God. The secret behind my beauty is you guys. Okay, and drink water. Yes, I drank water. Where can we get the PDF of this? PDF of this will be uploaded in the WhatsApp channel. Yes? Now, Shavani says, okay, Panvit says the answer for this question is 4, and Sanket says 4. Okay, what about others? Lakshmi says the answer for this question is C. Ajay, Black Ma'am, yes. Deadpool says the answer is D. Are you sure? Lavanya says B, Ramya says C. Okay. So B and C and D is the option that you are giving me. Tasmia, hello. Pooja ma'am, you're great and you're my fave. Oh, thank you so much. So D is what most of you guys are answering. Let me check what is D. D is this. So basic logic, first of all, before going ahead up and doing all this crap out there, we have to look at the picture, because we know what is easy to spot. The most easiest one to spot is what? E1, E2, E3. Right? E1, the first enzyme is beta-galactosidase, second enzyme is going to be permease, and the third enzyme is going to be trans-acetylase. Yes. So let's spot that, and let's see if at all we can find the answer with the help of that. So E1 is going to be beta-galactosidase. 3 of the options have that. No option left. Okay. So basically, I can just cut off this particular option. Dun, dun, dun. Okay. What about the next one? E2 is going to be permease. We just have these 2 options. So dun, 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 that also goes. Okay. Now we have trans-acetylase, and both the options are trans-acetylase. So we do not have any choice up there. Basically, we have concluded answer would be somewhere around this. Okay. Now come up over here. If you see, mRNA which is there is coding for X, and this X basically comes in contact with Y. So X is what? X is something that was already there. So the one that is already there, that is coded by the mRNA, is going to be what? Repressor protein. And what is the other thing that comes and binds to the repressor protein, Baba? Inducer, that is your lactose. So X is going to be your repressor, and inducer which is there is going to be Y, that is the lactose. If at all you remember the lac operon, you will be able to give the answer for this, and I think most of you did, because most of you gave me the answer as D, and you guys are super awesome. Okay, so perfect. Why I have marked this? Just a second. No, this is not the answer. Okay, this is the answer. D is the answer. Cool. Clear now? If yes, give me a thumbs up. Give me a thumbs up. Sudarshan, you use Snap, nothing, leave it here. What is happening over here? You're making me sad. Don't do that. Don't do that today. Today is not the day, dear children. Please reply me, cockroach. Dear, what's there in your NCERT is the only thing that is introduced in your what do you call examination. Okay. Next question, guys, question number 23, again brand new, but from the same concepts that have been asked before, so we have to try it up. Okay, now here you have to use your brain very much. So this picture was not for you, this picture was for me anyways. E. coli was grown in a culture medium supplemented with N15 NH4Cl, and total bacterial DNA was labeled with N15. The medium was changed containing normal N14 NH4Cl medium. The bacteria that show low density after 60 minutes are, how much percentage of them are going to show low density after 60 minutes? Any idea? Over here you have given the percentage. Solve this question properly. You tend to do a lot of mistakes. 75%. Who said that? Kyun, what are you crazy? That's the correct answer. And Panvit also said 1 in first go. What are you guys doing? What are you guys eating? Chaman Prash. Okay, Pooja, I want to ask something. Don't answer to him, Pooja. Okay, listen to me only. Okay, diagram is blood. Diagram is not for you, dear. Diagram was for me. I was thinking of explaining it, but anyways. So the answer for this question is A. Tasmi is saying, okay, A is what? 75 only. And for those of you who are not understanding, listen to me carefully. I'll explain this only once because this is a time-taking process. Okay, so listen to me. E. coli is there. Okay, E. coli is grown in a medium that is containing N15. You know, nitrogen is a component of DNA. So if at all I grow something in N15, basically the nitrogen that will be taken up. Okay, okay. So over here, the E. coli that was grown in N15 will be taking up the DNA. Will be taking up the nitrogen, and that nitrogen is going to be incorporated into DNA. Okay. I say that both the strands will be having N15 isotope of nitrogen. Okay, in that particular case, if at all you take the DNA and you do centrifugation, it will come and settle somewhere over here because it is a heavy band. N15 is a heavy isotope, which is why both of them are going to settle down somewhere over here. Okay, ma'am, done. Now, suddenly I am making a change. I am growing this. I am growing the E. coli in N14 medium now. What happens when I grow it in N14 medium? Listen to me. This time, the new DNA strands which are going to be formed will be taking what? N14 instead of N15. The older strands which will be there, it will be intact only. So, after every 20 minutes, my dear children, what is going to happen? After every 20 minutes, the DNA is going to divide and replicate. So, after 20 minutes, the DNA has divided, it replicated. Okay, it divided. Now the new strand which is there, if at all you see, it is green in color. Why? Because this time it has taken up N14 isotope. It has taken N14 because we have changed the medium, right? Okay. Now what type of strand do you get? When you do centrifugation, you get a strand which is there in the middle. You call this as hybrid strand because it is neither heavy. Why it is not heavy? Because both the strands are not red in color. Both the strands are not made up of N15. One is N14 and one is N15, which is why you get hybrid strands. Okay, okay. Now suppose again for next 20 minutes, I let it divide. Next 20 minutes also, I let it divide. What is going to happen? Basically, again when I divide this strand which is there, just focus on this, okay? This strand got separated, one red and one green. The new strand will be green only because why? We are growing in N14 medium only. Okay, so new strand is green. Similarly, Baba, what is going to happen? These two strands are going to divide. New strands are going to be green in color. Now when I do centrifugation, what is going to happen? I'll still see hybrid band and I'll see a band which has low density. That low density band is one that has N14 and 14. Okay, so this is the low density band. Cool. Now what happens? They're asking you what happens after 60 minutes. How much percentage of them will be showing low density? So you only use your brain and you think, okay, after 60 minutes, what is going to happen? These were 4 in number. They are going to replicate. This was 2. This became 4. Now 4 becomes what? 4 becomes 8. 4 becomes 8. And out of 8 of them, Baba, 8 of them, 6 of them are going to have light band only. 6 of them are going to have light band. Rest 2 of them which is there, it is going to remain as such. Because remember, Baba, all of them when they are going to divide, basically is the green one which they divides, Baba. And if I make it like this, because I know it will be difficult for you to imagine. If I make it like this, the DNA here, it gets divided into one green and another green. And another green, right? Okay. What happens? The new strand which is going to be formed is also going to be green in color. So you got 2 strands. What about this strand? This strand also, array. This strand also divides in the same manner. One green and one is going to be red in color. Okay, the new strand which is there, it is always going to be green, whatever you do. So in that particular case, here only you got 4 strands. Here also, Baba, you will get 4 strands such as that. But you will find only 2 of them are going to be like this. Only 2 of them are going to be hybrid. Rest everything is going to be green in color. So out of 8, if at all I see, out of 8, 6 of them are going to be hybrids. If at all you do the calculation, it comes out to be what? 75%. It comes out to be 75%. Okay. So which is why the answer for this question is going to be 75. Is this clear? Is this clear? Or is it not clear? Ma'am, this question can be repeated for KCET 2026? I think so. Okay, so this was not there in your PYQ. I mean, the concept was there, okay, but this same question was not there. You have been asked about 60 minutes. Always remember, after every 20 minutes, the DNA are going to get replicated. That is the first logic. Voice is incorrect. Once again explain and also correct your voice. Voice, what happened here to my voice? Let me check. The concept was there, okay, but voice is fine, da. Okay, but I still can. Okay, so yes, voice is clear like guys. Okay, so guys, I think for those of you who are not understanding, I need to explain it because that's my duty, and if at all someone wants to take a break of 2 minutes and come back, it's okay. I totally get you. Listen, first you were growing in N15 medium. When it was grown in N15 medium, all the DNA took N15 only. When you centrifuge, it came at the bottom and settled. This is known as high density. Okay. Now, after 20 minutes, basically while you are going to change over here, you're going to change the medium to N14 now. This is N14 medium, okay? And for 20 minutes, you are letting it divide. So what happens, the DNA strand that is over here, it is going to divide into two. They're going to divide into two. The new DNA strand which will be there, Baba, it is going to be N14. Right? The older one is going to be N15, but this is going to be N14. Now when you centrifuge, what is going to happen? When you centrifuge, you will see that you will not find a high density, you will find a medium density. You call this as hybrid DNA. This is somewhere in the middle. Okay. Again, what do? You divide for 20 minutes. 20 more minutes, that means you're talking about 40 minutes in general. 20 more, so 40 minutes. What is going to happen? You're dividing in N14 medium itself. What is going to happen? This particular strand has to divide into two. So one is the red strand and one is going to be what? The yellow strand. Similarly over here, yellow strand, and you're going to get the red strand. Now the new DNA which is being formed is also N14 because we are growing in N14 itself, right? So the new DNA strand is N14. Let's make it. So over here, if at all you centrifuge now, what do you find? When you centrifuge it now, you find that two of them are hybrid. Okay, and two of them are something that you never saw before. You never saw red before, right? It is low density. So two of them are going to be low density over here, and two of them are going to be hybrid. Again, when you divide for 20 more minutes, what is going to happen? 20 more minutes means 60 minutes. You will see that the same thing is going to be repeated. These hybrids remain as hybrid only because one parental strand will be there. Obviously, it is going to pair up with something new. So two hybrid strands are definitely going to be there. What about the rest of the strands? Whenever they divide, Baba, they are going to form N14 strand only. They do not have any other choice out there. So if at all it was 2, it became 4. Now 4 becomes 8. Now out of 8, always 2 of them are going to be hybrid and rest all of them are going to be having low density. So if I am saying out of 8, 2 of them are hybrid and rest all of them have low density, and you have to find what is the percent of low density. So 6 out of 8, so that means 75% would be the answer for this particular question. This is a very important concept. Do not mess it up. Please remember that. Cool? And okay, now just a second. Trust me, I do not know whether it's going to be tough or moderate. The second thing that I want to tell you guys is I will be blocking you guys. Okay, this is a friendly reminder. I really wish not to block anyone, but please remember, I have the power to block you guys. I'll do that if at all you talk amongst each other like this. Yeah, so please do not do that, and no chats apart from what is there in the studies. Okay, so don't do that. I know that you guys are friends, you want to be friends, but this is not the platform, Macha. What are you doing? I've also been in your age group and stuff like that. I also have friends. I'm also a young person out there, but I do not do it. Someone else is talking, respect them, right? It will be really good. Yeah, hello Ritika. Perfect. Ma'am, if again 20 minutes, what is going to happen? Same thing. If again 20 minutes, 2 of them are going to be hybrid and rest all of them are going to be what? What do you say? Low density only. Oh, every time 2 will be there. Cool? Yes, perfect. So the next question that we have is question number 24 and goes like this. Arrange the various steps of DNA fingerprinting technique in the correct order. So DNA fingerprinting is also a concept that is there which we need to know. So let's go ahead. Yeah. So you have to arrange the steps of the DNA fingerprinting in order. So go by this, see the options and tell me the answer for this question. Ma'am, if you are again keeping in N14 medium during... We are keeping in N14 medium only, dear. That is why it is going to take it up, right? Yes, perfect. Hello Raksha, hello Parvati, hello. Okay, so Kavita says C is the answer, Panvit says 1 is the answer. So whenever you say 1, dear Panvit, sorry I do not read out your name. This is because I am seeing ABCD options, right? So all of a sudden when 1 comes, yes. Ma'am, this much easy questions, yes, this much easy questions could also be asked. But during exam, no, things are going to be different under pressure. So not ZYT, and it's Vicky says A, Deadpool says A, Rabia says A, and it's Love, Hina Kausar, Norman, Chasemaker, and everyone. Okay, everyone says the answer for this question is going to be A, which is why I'm going to lock A, and let me see if at all it's A or not. So first thing first, in DNA fingerprinting, what do you need to do? You need to isolate the DNA first. That has to be the first thing. So isolation of DNA will be the first one. So that is basically either A or B. Other options, I am going to cut it off. Now, before I go ahead with doing all this drama, I want you to understand this. Okay, so this is my usual example, but I'm still giving the same example. Listen, there's a murder spot. Okay, there's a murder spot, and over there me and Tasmia were there. Okay, so I said that Tasmia did the crime. Tasmia said that I do not know who did the crime. So basically what happened, actually I did the crime. I am the killer. So from the murder spot, they got a sample of hair strand, and basically the hair has DNA anyways. The hair follicle will be having DNA. So they got the DNA from that particular place, and what they did is that they made a complementary strand of it. Okay, they got a DNA, they made a complementary strand of it, and basically they tagged it with something that is radioactive. What do you call this particular thing as? You call this as probe. Okay, that is there. Now they called both of us suspect. Okay, me, that is JJ, and Tasmia. We both were called. What was the first thing that was being done? Isolation of the DNA. DNA was taken from both of us. Okay, taken. After the DNA has been taken up, what is being done? The second process would be digestion. We are going to cut the DNA into small parts with the help of what? Restriction enzymes out there. So we say after isolation, we do digestion of DNA by restriction endonuclease. So this is going to be first, second is going to be this. Okay, what is the use of it? Basically our DNA has got separated. Okay, fine. After the DNA has been cut, my dear children, what we have to do? We have to run them to the gel electrophoresis chamber. That is how they are going to be separated from low to high density and stuff like that. Okay, or basically the smaller fragments are going to be separated very easily. So we do gel electrophoresis. That is going to be the third step. Now when we do gel electrophoresis, from this point onwards you should be more attentive. What exactly happens is that in gel electrophoresis, JNT, my DNA got separated like this. Tasmia's DNA got separated like this. Okay, and basically this particular DNA which I got from that particular area, that was also separated, and it was separated like this in the same pattern like mine. But I still do not know how, because I cannot clearly see it. If at all, I just see it through gel electrophoresis. Basically, I have to do the next thing. The next thing is what? I have to make sure that I transfer whatever is there on the electrophoresis chamber. I have to transfer it onto the membrane. So transfer the separated DNA fragments onto the membrane. This is going to be the fourth step. After transferring, what do? I want to clearly see things. So I am going to run this particular probe. This probe is only going to attach to the areas where it has the complementary DNA. So it will be attaching somewhere over here because we also share some things, but most of my DNA is going to be colored up. And basically, okay, so this is hybridization using VNTR probes. This is going to be the fifth step. And finally, Baba, you have to see it under the autoradiography machine. And when you come and see under the autoradiography machine, what has been seen? That Baba, Jasmine did the crime. It was not Tasmia, it was Jasmine, and Jasmine is a bad girl over here, right? So the answer for this question becomes what? Your answer would be is where is it? 1, 2, 4 and 2. Your answer is going to be A. It is going to be A. Is this particular thing clear? Is this particular thing clear, guys? Okay. Okay, next question. Guys, if it's clear, can you give me a thumbs up? Perfect. Okay, Jasmine is the killer. Yes, I am the killer. You are right. Okay, so next thing that we have is question number 25. Which of the following statement is not correct? That is being asked. Which of the following statement is not correct, guys? Structure with many ribosomes bound to mRNA is called polyribosome. Amino acylation of tRNA requires the presence of ATP. Okay, exons are the DNA sequences which are not expressed and do not appear in processed RNA. After transcription in prokaryotes, mRNA is used in the same compartment as there is no nucleus. So what do you think is the answer for this particular question? What do you think is the answer for this particular question, guys? So, Sumit dear, can you wait for some time? I'll be explaining it towards break because I think it will take too much time or else. Yeah? Please mark the question. I will be answering it to you. So C is what most of you guys are saying, which is SD and GUK. Okay, perfect. All of you are saying C. And C is what? C is this. Exons are DNA. Okay, let's go ahead and check it out. So structure, you have been asked what is not correct. Okay, what is not correct has been asked, not what is correct. Okay, a structure with many ribosomes bound to the mRNA is called polyribosomes. So basically you have mRNA. If at all you have many ribosomes bound to it, you actually call it polyribosome. This is correct. Okay, amino acylation of tRNA requires the presence of ATP. What do you mean by that? Your tRNA, Baba, it looks like this. There is 5' end, there is 3' end. On the 3' end, you have the amino acid being joined, and for this definitely you require ATP. So this process is known as amino acylation, and this requires ATP. This is also correct. Exons are the DNA sequences which are not expressed and do not appear in the processed RNA. Wrong. Exons are the expressed sequence. Introns are the non-expressed sequence out there, right? So this is going to be the wrong statement. Now after transcription in prokaryotes, mRNA is used in the same compartment as there is no nucleus. Exactly. In the case of prokaryotes, Baba, everything happens in the same region only. They do not have nucleus, they do not have anything out there. So everything is coupled reaction. We studied about it. So the only answer for this question is going to be C, and it's a very easy question. Yes? Okay. Tilak. Okay, Tilak and Mayura, Baba. Do you want to go out from this particular video? Live chat. If not, don't chat. Okay? Guys, is this clear? If yes, give me a thumbs up. Let's go ahead with the next question. Okay. Yes, exon, intron. Exactly, you are correct. Next question, I think it is going to be some question of your PU1. Guys, I'm asking you, is it okay? Give me a thumbs up. Please give me a thumbs up. Sumitra, I told you, dear, I will be explaining it to you. Give me some time. I'll be explaining to you a little bit. Okay? Otherwise, those of you who are asking the questions, who have been waiting for the next question, they'll be delayed a lot. So please wait for some time. Yes, perfect. Okay, so just give me a second. Okay, perfect. The given figure represents the process of transcription in bacteria. So this is the process of transcription in the case of bacteria, okay? And select the option which correctly labels as A, B and C. What is A, B and C? What do you think is the answer for this question? Guys, you have given me the answer also. Panvit says it's 2 the answer. Norman says it is B, that is the answer. Okay. Lavanya says B, Ritika and Anand and Sanket. Baba, I'll tell you, it will take some time. It's 26th question now. We have 103 questions to solve. Yes. And yes. Panvit, Alina, how are you? Are you asking me? Okay, I'm good. Thank you for asking. And Sagar, Savita, Nisha, and Rabia, Kyun, Tasmia, C only. Okay, so the answer for this question is going to be B. It is going to be B. Is it B? Let's check it out. Okay, so what is A? A is mRNA. If at all you see, Baba, when transcription happens, this is your DNA, a new strand is being formed. Yes, that is your mRNA. Okay, and you need an enzyme for doing all of that. That will be B. B is what? RNA polymerase. Now this RNA polymerase needs a support system during the end of transcription to end up the transcription, and that is known as Rho factor. Why? Because it is crying, it is breaking up with DNA out there, right? That's why it is crying. Rho factor is there to basically do the ending process, termination of it. So B is going to be RNA polymerase, and C, which is there, Baba, it is going to be the Rho factor, which is why the only answer for this question is going to be sigma factor. Just a second. This is wrong. Okay, I have marked it wrong. Don't worry. The answer is going to be B. It is going to be B. Okay? Cool? I hope that works. It's sigma. No. Sigma factor. Listen, over here, if at all it was right over here, we could have said that it is sigma factor. But this is on the end terminal, at the end, right? And your mRNA is dissociating. So it is termination. And in termination, what do you have? You would be having rho factor, no? You would be having rho factor. Sigma factor is for initiation process. It's for starting up the process, right? Sigma, it is too shy to start off. RNA polymerase is too shy to go and initiate the process, which is why it needs sigma factor. That is how it is going to go. Yeah? Got it? So the answer for this question is going to be B. 26th B. Cool. And now let's go ahead. Yes. Question number 27. This is from your PYQ. Come on. Let's try it out. And it goes like this. Find the sequence of binding of the following amino acid tRNA complex during translation to mRNA transcribed by a DNA segment having the base sequence blah 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 blah. So what do you have to find? You have to find the sequence of binding of following amino acyl tRNA complex. That means you have to find how the tRNA is going to be attached. But before that, during translation to mRNA transcribed by the DNA. So basically this is a DNA strand. You have to make an mRNA out of it. And then after making an mRNA, probably you can look for the anticodons out there. And you guys are giving me answers like this. Are you even trying? Is it like you all have ratified this particular questions? Now, Panvit says 2 is the answer. Prakash says, hello dear. Nauman says B. And Lachi says B. Okay. Bhavana, Sanket, and Akshaya, Prakash, Kyun. So the diagram is blurred. I can show it to you. Just a second. Okay. Check it out. Okay, this is the diagram that you have, and this is the strand that you have. You can use, I will go that side, and probably you can see it better. Yes. B. Most of you guys are saying B is the answer, which is why I have to go ahead and search for it. Listen, when these type of questions come, first of all, I already told you that this is a DNA that you have to make it into mRNA, but you have to use your brain there, okay? If at all you see T over here, T means what? It is going to be DNA. You have to make an mRNA. So how do we do that? Let's do it. So 5', when we make an mRNA, it starts from 5', T base pairs with A base pairs with U this time, not with T, because this is an RNA. Okay, C base pairs with G, and A base pairs with U, and T base pairs with A. Suppose this much is being done. With the help of this much, can I try to find my answer? Let me check. So this is what the mRNA strand. This is going to be the mRNA strand where I have AUG, and I have UA something. Anyways, so the first anticodon that should be coming, the tRNA that should be coming, should be having opposite to it. So instead of A over here, it should be having U. It goes with A, and finally C. So UAC will be the first one. UAC is going to the first one, and that is B. And where do you have B as the first option? There is B option. That is the B option right there, which is why the answer for this question is going to be BADC. So UAC joins first, then join the next one there, and this is how similarly you can get the whole answer. Once again, repeat this question. Yes, I can definitely repeat this question. Let's go ahead. Okay, let me try. So over here, the question, if at all we dissociate, find the sequence of binding of the following amino acyl tRNA complex during translation to mRNA transcribed by a DNA segment having this a sequence. This is a sequence that is given to you. First of all, what you have to do, since you find T over here, you have to make it an RNA. How do you make it an RNA? Listen, first 5' and 3' you write, and you make a complementary strand. T base pairs with what, Baba? T base pairs with A. What does A base pair with? It base pairs with U. C base pairs with what? G. A base pairs with what? A base pairs with again U. T base pairs with what? A. This is how it is going to go, right? We are going to get a strand something like this. Now what we have to do? We'll take the strand. So this is what? This strand which is there, it is known as the mRNA strand. Now onto this mRNA strand, the tRNA will be coming and binding, right? So the tRNA which is there, The first tRNA which is going to bind is what? We have to make again the complementary pair of it. So A base pairs with what? A base pairs with U. U base pairs with what? A. G base pairs with what? C. So basically UAG is the one. UAG is the first, sorry, UAC is going to be the first one. If at all you see over here in the option that is given to you, UAC is right over here. UAC will be right over here. Got it? Okay, and B is the first one. Where do you find B as the first one? Only in one option, that is this one, which is why this is the answer. If at all you would not want to do all this particular drama, you can do one very important thing, that is wherever T is there, just replace it by U. Okay, wherever T is there, just replace it by U. So it will be UAC, and then finally again, yeah. So UAC, if at all you do over here, if at all you replace, basically you can try to find the answers like this only. You do not have to do all the things. Why? Because you are making a complementary strand, then again making a complementary strand. That means at the end you are going to be redirected to this only, right? The only difference is that wherever T will be there, there U will be there. Yeah? So, yes. Okay. Okay, you're most welcome, Lachi. Question number 28. 28, what is the question? In structural gene, the template DNA strand has nucleotide sequence this. Find the correct and complementary nucleotide strands on the coding strand. On the coding strand. So this is a template strand that is given to you, Baba. This one, you have to find the coding one in here. What do you think is the answer for this particular question? Ma'am, when is math class? Math class is tomorrow, dear. Okay, Panvit says the answer for this question is 3, Lavanya says it is, okay, C, and Savita says it is C, a little bit fast, I'll try. Okay, Manjunath says C, okay. Lachi, C. Okay. So most of you guys are saying C, and C is what? C is going to be this. It's an easy question. So you have to, first of all, whenever you are going to find the complementary strand, look, it is in 5' to 3' direction only. Yeah. And then make sure that you're talking about what? You're talking about, yeah, nothing else. Okay. So basically make the complement of it. A goes with what? T goes with A. G goes with C. So if at all you see, you just have options, that is this, TAC, and that is C option, which is why your answer for this question is going to be C. It's an easy question. Okay, make sure you are not making any mistake when it comes in the case of RNA also. If it is mRNA, DNA that is given to you, so Baba, it's C. Yes, it is C. Okay, next question. RNA polymerase 2 is responsible for the transcription of. RNA polymerase 2, Baba, is responsible for the transcription of what? tRNA, rRNA, hnRNA, or snRNA. What do you think is the answer for this? Sam, no, Baba. How can you take laptop for the examination? You cannot. Yeah? C again, C again, Q and C again. So C again is what? hnRNA. RNA polymerase 2 is responsible for hnRNA. Yes, Macha, it is responsible for hnRNA. So always remember, we have RNA pol 1 and 2 and 3, and we need to remember each of their function. Okay. So first priority is given to formation of rRNA, because R, everything starts with R. So first priority to formation of rRNA. We have 3 type of RNA which is being formed: 28S, 18S, and 5.8S. If at all you see, all of them end with 8888. Okay. The second priority is given to formation of hnRNA. That is basically the one that is undergoing transcription, DNA to hnRNA, right? It's a precursor of the mRNA. So for that you have RNA polymerase 2. Now RNA polymerase 3 sounds like T. So 3 is for tRNA, and exclusively for some other things also. For example, you have snRNA, small nuclear RNA. Apart from that, you have different type of rRNAs as well. What are the different type of rRNA which is not included over here? Only one, that is 5S rRNA. Okay, 5S rRNA. Because it's not 5.8. Remember, 0.8, everything and 8 is starting up over here, and everything that is not, it's over here. Cool. Okay, so yes, ma'am, you told this before also. Exactly, I have told this before. Yes, guys, got it. Shall we move ahead with the next question? Ma'am, and your teaching is really good. Thank you so much. Thank you so much, Sam. Guys, Question is clear? If clear, can we go ahead with the next question? If yes, give me a thumbs up. Yes. Perfect. Thank you so much. Let's go ahead with the next chapter altogether, and that is what? Body fluids and circulation. Again, you will find few questions here and there from PU1, then we'll start with the PU2 chapter. Cool. Ma'am, it's helpful for NEET. I'll recommend watching NEET PYQs for NEET. That will be helpful. Okay. So the diagram given here is the standard ECG of a normal person. The P wave represents what? So Baba, in ECG you might have seen dhak, 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 dhak. So there is P, Q, R, S, and finally you have T wave. What does the P wave represent over here? Anyone? Any idea? Thumbs up. Thank you, Norman. Okay, clear. Panvit says 3 is the answer for this question. Hello. And Manjunath says 3 is the answer. Shavani, Manoj, Deadpool. Most of you guys are saying 3, 3, 3 or C is the answer for this question. What is C? Contraction of both of the atria. That is represented by P. Is that so? Is it represented by P? Yes, exactly. And you guys are absolutely correct. Listen, Macha. Everyone has a heart, right? Everyone check. Everyone has a heart. So basically left atrium and left ventricle, right atrium and right ventricle. So first of all, listen to me very carefully. Your atrium which is there, it is filled with blood. Okay. It is filled, yellow blood, sorry, just a second, okay. It is filled with blood. What exactly happens? This blood has to be transferred to the ventricles, because ventricle is something that is going to transfer to the rest of our body and to our lungs and things like that, right? So what do? First of all, atrium has to contract. That is known as systole. That is known as systole. Systole means contraction. And atrial systole means atrium is the one that is contracting. This is also known as, this is caused because of what? Depolarization. This is also caused because of depolarization of atria. So these terms are very important. You can get questions on the basis of that. Depolarization of atria causes atrial systole. And just a second, I stamped on something. And when that happens, basically what happens? The blood is going to flow down. So P wave represents over here, the P waves that you see over here, it represents what? Atrial contraction. Okay, ma'am, done, ma'am. Next thing, QRS. What do you mean by that? Now the blood which is there, it moved down. The heart is gone. Okay, anyways. The blood which is there, it moved down to the ventricles, right? Yes, it moved down to the ventricles. Now the ventricles also has to undergo systole. They also have to undergo contraction. This is known as ventricular contraction or ventricular systole. And it is caused because of what? Because of depolarization of ventricle. Same logic over here also. So QRS represents what? The contraction of the ventricles, or you call it as ventricular systole. Okay, and finally you have the T curve. T curve represents what? The ventricles after contracting, Baba, they are going to relax. They are going to relax, and that is known as that is caused because of repolarization of ventricles, and basically we call it as diastole of ventricles or ventricular diastole. You're not talking about anywhere, you do not talk about atrial relaxation, okay? You just say atrial contraction, then ventricular contraction and ventricular relaxation. That's all. Please remember, these two are depolarization, this is repolarization. Yes, so which is why the answer for this question is going to be C, because here you have been asked only about P. The reason I explained to you is that you can get questions on the basis of it and you should be able to answer it anyhow. Yeah, so I know it sometimes you might be irritated because I'm explaining extra, but I am not saying that this question is going to come. I am saying that concepts are going to be repeated. So I have the duty to explain you that, right? Hiya, my pet. Okay. Thank you, Norman. And hello, Raghavendra. Hello, Macha. Okay. So guys, let's go ahead with the next question out there. I know that you are running out of time. Question number 31. Select the incorrect statement. Come on, try it out. Select the incorrect statement. What do you think is the answer for this, guys? Second PUC Biology 100. Select the incorrect statement, guys. Specialized tissue is found in the heart called nodal tissue, which has property of auto-excitability. Does it have something like this? Yes. A tissue patch found on the left upper side of the right atrium is called SA node, okay? The bundle of fibers originate from AV node called Purkinje fiber, okay? The nodal tissue has the ability to generate action potential without any external tissue. Is that so? What do you think is the answer for this particular question? Hello, Sudarshan. Guys, what do you think? B, 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 B. Okay, Nauman says it is B. Raghavendra says it is 2, that is B. Okay, okay. Shravan says it is B. Lachi, and Madan, Shreya, Gagan, and Pragnya, and Maria, Shravami, and everyone. Most of Vijay is also there. Everyone, Bhavish. Bhavish has a different answer. Okay, so let me check it out. So over here, Baba, if at all you look. So I am going to directly tell the answer because I think that will be saving the time. It is going to be this. This is the answer. This is incorrect statement. Why? Because it is written left upper side of the right atrium. That is wrong. Now the reason I am explaining with the answer is that because I already have a picture over here. So listen, this is your atrium. This is your ventricle. Similarly, this is your atrium. This is your ventricles out there. Here other atrium is not being mentioned. Anyways, okay, other atrium is not being mentioned. But listen to me carefully. In the atrium, okay, the right atrium, at the right side, okay, in the right atrium, in the right side, you have SA node. Okay, always remember, I think with that particular picture, it is not easy to understand, so I have to make my picture. You have right atrium over here, you have right ventricle, left atrium, left ventricle, right? You have SA node over here. In the right side, not the left side. That is wrong. SA node is auto-excitable. It does not need any external stimuli. That is correct. Auto-excitability thing is also correct. Okay. Now, the SA node is going to send signals to the muscle fibers over here. When it sends signal to the muscle fibers, what is going to happen? The atrium is going to contract. And later, this signal is going to reach somewhere over here. What is there over here? In the left side, bottom of the right atrium. This is left side now. Okay. This is going to be the AV node. It's somewhere between the atrium and the ventricle. It is present more towards the left side. Yeah. Now it reaches the AV node. The AV node, Baba, it is going to send the signal to bundle of His. What do you call this? Bundle of His. And the bundle of His is going to send the signals to all the different parts with the help of some fibers, which is known as Purkinje fibers. What do you call these fibers as? Purkinje fibers. And finally, it is going to contract. The ventricles are also going to contract. So always remember, SA node in the right side, auto-excitable, it does not need anything. And AV node, it is more towards the left, in between the atrium and the ventricle. AV node comes between the atrioventricular. That is why it is known as AV. So do not make it at the top. Do not make any mistakes out there. Then bundle of His, then you have Purkinje fibers, right? So your answer for this particular question is going to be B. Cool? Yes? Perfect. Is it clear? And by the way, I am keeping on blocking children who are not attending to the class, because that was the only resort I had to. Okay, after probably a lot of talking about it and warning you guys. So I'm so sorry, but yes. Okay. Now you can be the next lucky person if at all you do keep on doing that. Guys, if it's clear, can you give me a thumbs up? Let's go ahead with the next question. Yes, perfect. Okay. Question number 33. This is also one of the repeated concept out there. Match the different type of leukocytes, as in the WBCs. Okay, and over here. Okay, so here you have the options and here you have the percentage. You have to match with it. The highest and the lowest is something that you have to make sure you do not miss out by any chance. Cool? Yes, Izesh, we are doing the next question. Come on. Okay. A is the answer. Okay, Nisha says A is the answer. Akshya, hello dear. Okay. What do you think is the answer for this particular? Vijay says B. Deadpool says A. Bhavish says B. Okay, you have given A, B, C, D option to me, huh? Okay. Most of you guys are saying it is B. Let's check it out. Listen, there is a very easy trick. Okay, when you talk about leukocyte, that is WBCs, the most famous ones that are found in the most abundant number is going to be neutrophils. They are going to be neutrophils. So if it is the largest number over here, it is going to be 60 to 65. So neutrophils is going to take that particular position. So A goes with 2. A goes with 2. How many options do I have? I have 3 options. I can cut off the middle one. Okay. And I also know one more very important logic, that basophils which are there, they are found in the lowest amount. So out of this, what is the lowest number that is given? It is going to be 0.5 to 1%. D goes with 3. D goes with 3. I just have one option, macha, and with that I can actually find my whole answer. I can actually find my whole answer. Got it? So basically neutrophils goes with this. Basophils which is there, it is the least one. Always remember, you can get questions, and this is going to be the highest one. The second highest position, if at all you are seeing the ranking, first rank is going to neutrophil. The second rank goes to what? The second rank goes to lymphocytes. It goes to lymphocytes. How much percentage? It is basically 20 to 25% out there. Do not miss out on this. These are very important things. And guys, can I ask you one question? Basophils which are there, what do they release? They release two very important things. If anyone has read NCR, if everyone is anyone is preparing for KCET really well, they might know the answer for this also. Anything? Any idea? Any idea? Hello, Manjunath. Basophil which is there, it releases two very important things. Ma'am, what about the dress code for KCET? Dear, I'll be making a video on it, okay? Officially, as soon as it gets published, I'll be making a video. Histamine and serotonin, exactly Shivani, exactly Tanu, Guk, everyone. Histamine and serotonin, okay? Histamine and serotonin, these are the two things, not heparin, okay? Histamine and serotonin are released by basophils. This is a very important thing that is also being asked. Cool. Let's go ahead with the next thing. Ding, ding. And the next chapter that we have is PU1 again. Three questions only from biomolecules. Okay, not more than that, don't worry. Shall we go ahead? Ma'am, break. We want break. Can we take a break once we reach 50 questions? Is that fine? These are the important questions, dear. Okay. So guys, the next chapter is biomolecules, and in biomolecules there are two charts with the help of which those two charts are something that gives usually the frequent questions out there, and we are going to deal with that as well. Yeah, and after this we are going to cover up the PU2 chapter which is there. So we are going to mix and match the PU1, PU2 chapter, and that way we will cover most of it. Refer to the given table and select the correct options from PQRS. So over here P and Q and R and S given over here, and Baba, you have to tell what is what. I'll tell you a hint, okay, I'll give you a hint. Oxygen which is there, it is found in the most amount in the Earth's crust. In the Earth's crust, it is found in most amount. Tell me. Okay, 34D, Lavanya is saying 34D is the answer. Okay. Hitesh and Shravan and Vijay, most of you guys are saying it is D, and D is what? Let me check it out. Some people are saying it is B. Okay, so oxygen, as I said, it occupies the most amount, that is Q. Q, the highest amount that you see is 46.6 and 46.6. Okay, so these two can be my answers. I am going to cut off the other options. Okay, okay. What about the next thing that is there? I also know that sulfur which is there. Okay, sulfur which is there, it is less in Earth's crust, and it is also less in the case of human body as well. Okay, it is less in Earth's crust, it is less in the case of human body, so R is also going to be less. So over here R is 0.03. It cannot be the same value, it has to be lesser than that, so it is 0.3. So basically my answer is going to be B for this. Sorry, it is going to be B for this. Listen to me very carefully. Okay, here O which is there, it is 46.6, that is the highest among everything in the Earth's crust. Okay, now when we talk about sulfur, sulfur is found in less amount in Earth's crust, and basically in human body it is also less. So here it is 0.03, here it is going to be 0.3. It is going to be 0.3. Cool? Cool. Now, not lesser, so I meant ulta. Anyways, okay. Now, carbon which is there, carbon is also same like this. Carbon is going to be exactly similar in the amount of sulfur. So, over here, if it is 0.03, it is also going to be 0.03. So, these are the few things that you need to remember. Please give a break for 10 minutes. Surely, I'll give a break for 10 minutes then. Okay, so please, once we are done with biomolecules chapter and when we are getting the next chapter, I'll give you a break. I hope that works. So over here, oxygen is the highest, always remember that. Carbon which is there, it is just 0.03, just like sulfur. These two things are very less in our Earth's crust. Okay, in our Earth's crust. So yes, that's all. Cool. I hope that's clear, guys. It's clear, right? If yes, can you, yeah, if you want to take a screenshot, you can. By the way, do not need to take a screenshot. It will be there in the, what do you call, PDF. You can take it directly from the PDF as well. Okay. Perfect. Now the next question answer I have already given. Ayyo. So it goes like this. Refer to the given passage and identify X and Y. And X is a type of protein that acts as a hormone, and Y is a protein which enables glucose transport into the cell. So X is a type of protein, is a hormone, and basically Y is a protein that enables glucose transport into the cell. So you know there is only one hormone mentioned over here, and that is insulin, because albumin, pepsin, trypsin, all of them are basically enzymes out there. And GLUT4 is something that helps in the transport of glucose inside the cell. Okay, it is the one that helps in the transport of glucose inside the cell. Yes? So basically the answer for this question is going to be D. It is going to be D. And this particular thing is also super important for you. Collagen, which is there, this table is important. Some proteins and their function. Collagen, which is there, it is the intercellular ground substance. It's in between the cells. Trypsin is an enzyme, insulin is a hormone, antibody fights against infectious agents, receptors are there on the surface of the cells, and GLUT4 is helping in the transport of glucose. This is a very important thing. Okay. Ma'am, say how much marks will you get after watching this video? The highest is what I'll say. Okay. It is definitely going to help you out if at all you utilize it well. Guys, I think now we are going to go ahead with PU2 chapter. These two concepts are very important. Along with it, if at all, you can have a look at enzymes also. Okay, the graph of enzymes. You please make sure you do that. That is also important. Now, do you want a break of 10 minutes? How many of you want a break of 10 minutes? Hello, Arshni. So do you want a break now, or should we take the break after a few questions? What's the take of it? KCET dress code, Alina will be surely making a video on that. Do not worry, okay? So just waiting for, what do you say, an official update to come through. Okay, some people are saying no ma'am, some people are saying break. Hi ma'am, hi. Okay, so we'll take a break then. Let's meet after your... Thank you so much, Manjunath. Okay, so I want a break. Okay, fine. So we'll take a break now. And yeah, and then we'll start fresh with the PU2 chapter, that is sexual reproduction in flowering plants. Come after 10 minutes. Cool? So as of now, the time is Kit Kat break. And when we come, we have to come with so much energy. Kit Kat break. And as of now, it's 12:27. Be back by 12:37. Okay? Yes. And guys, when you come back with energy, because my energy is going down, okay? As I said, I was preparing this PPT for like 3 o'clock I slept. Tomorrow morning I woke up and my energy is all going down. If at all you are not contributing to that, if at all you are not energetic, the class is going to be blah. Don't do that. Okay? So, yes. Hello. Hello, ma'am, time over, time up. Yes, I'm back. Okay. Hello, Kyun and Shavani and Shankar. It's Mr. Miku. Everyone, hello. Okay. So, children, I hope I'm audible as well as visible clearly. Hello, Nisha. Okay, perfect. So we are back with our live, and this time with more energy. Everyone had some food, okay, had anything? Please make sure you keep yourself hydrated, make sure you keep yourself healthy, okay? Your preparation should go along with mental health and also physical health, right? Yes. Okay, perfect. Welcome back. Perfect. So let's go ahead with the next question that we have, and this would be from the chapter Sexual Reproduction in Flowering Plants. More energy and more vigor. Let's say question number 26. A flower has 10 stamens, each having bilobed dithecous anther. If each microsporangium has 5 pollen mother cell, how many pollen grains would be produced by the flower? What do you think is the answer for this particular question? Come on. Hello, Chetan. Ma'am, breakfast now, Ada. Okay, so what do you think is the answer for this question? Please understand that a flower has 10 stamen, and each of them are bilobed and dithecous in nature. And you are being asked how many pollen grains are going to be produced. Any idea? Okay, Kyun says it is B. What about others? Nisha says it is B, and Chetan says it is B. How is my headache? Headache is still there. Somehow energy is a little less today. Okay, Lavi says it is B. Most of you guys are saying it is B. B is what? 800. Is it 800? Let's check it out. And everyone who is over here, make sure you try to give your answers by the wrong or right. Attempt the questions, okay? Attempt the questions. That is one very, very important thing. If at all you are not attempting, basically you never know where do you stand. Yeah? Yes. So, yeah. So over here, Baba, you're talking about 10 stamen. First of all, let's talk about only one stamen. One stamen, an anther is there. And each anther, how many microsporangia is there, Beba? Sir, Beba, yeah. Baba, 4 of them are there, right? It is going to be 4 of them. Right. Okay. Now, there will be pollen mother cell over here in each of them. And these pollen mother cells are going to give rise to the pollen grains. One pollen mother cell undergoes meiosis. At the end, how many microspores are going to be formed? 4 microspores are going to be formed. That means 4 pollen grains are going to be formed from one of them, from one pollen mother cell. Now here you have been a question given in the question itself. Each microsporangium has 5 pollen mother cells. Okay, so each of them, these are each microsporangium, and each of them has 5 of them. So 5, each of them are going to give rise to what? The 4 pollen grains. So 5 into 4 is equals to what? 5 into 4, 5 into 4, 5 into 4. So 20, 20, 20, 20. All of them will give me 20, 20, 20 pollen grains. So 20 into 4, basically 80 pollen grains I am getting from here itself, from this particular anther. Now how many similar anthers do I have? I have 10 stamens. That means 10 anther. So 80 into 10 is equals to what, Baba? It is equals to 800, which is why the answer for this particular question is going to be this. Okay? Yes, the answer for this question is going to be B. Is it okay? Is it clear to you? Everyone? Yes, tetrad. Exactly, Bhavish. Yeah? B, Chetan, yes. Cool, now. If you have got it, can you give me a thumbs up? Can you quickly give me a thumbs up? Question number 38. In apple, the chromosome number of gamete is 14. What is the chromosome number in the primary endosperm nucleus? Guys, come on. Try to find the answer for this question. In apple, the chromosome number, okay, of the gamete is 17. You're most welcome, Srinivas. Hello, ma'am. It's Amina. Hello, Amina. Okay. What is the chromosome number in the primary endosperm nucleus? What do you think is the answer for this? Shailaja says it is D, okay? And Vamsi, what help do you need here? Tell me. Chetan says A, and it's Mr. Miku. Ayo, what happened here? So I'm still testing my patience out there. I really do not want to block. That's the reason I'm not blocking. Okay, so A is the answer for this question, is what most of you guys are saying. Let's check it out then. Okay, we have to check it out together. Let's check it out. Listen, you first make this Okay, 3 antipodal cells, 2 synergid cells, 1 egg cell, and here you have the 2 polar nuclei out there. What is the logic? In apple, the chromosome number of gamete is what? 17. That means you're talking about the haploid number of chromosomes, because in the gametes you have haploid number of half the number of chromosomes. Okay, fine. Now what is the chromosome number in the primary endosperm nucleus? How is primary endosperm nucleus being formed? Your pollen grain, which has 2 male gametes, 1 male gamete comes and fuses with the egg cell. What happens to the next male gamete? Next male gamete, which is haploid in nature, comes and fuses with the 2 polar nuclei of the central cell. That's how it is going to make what? The primary endosperm nucleus, and this fusion is known as triple fusion. Why triple fusion? Because 3 nucleuses are fusing. 1 nucleus already has 17, so when you say the gamete has 17, that means the other nucleus also will have a 17 only. This is also haploid, this is also going to be haploid. Now, but 3 nucleus are fusing together, so 17 into 3, the answer is going to be what? 51. It is going to be 51. Got it? Got it? Yes? Hello, Sanjana. Yes, 3 into 17 or 3 and triploid, that is 51. Pallavi, you're actually correct. Okay, everyone who is giving me the answer, perfect. Super. Okay, so 1 male gamete fuses with this, another male gamete fuses with this. And if it's clear, can you give me a thumbs up? We'll move ahead to the next question. Guys, if it's clear, thumbs up, please. Yes. Thank you. And Maria, and everyone who feels stranded, who feels that I'm not answering their doubts and things like that, please stay back. I really want to answer your doubts, okay? Yes. Okay. Question number 39. Clistogamous flowers are self-pollinated because. Why do you think clistogamous flowers are going to be self-pollinated, Macha? They are bisexual flowers which do not open at all. They are bisexual and open flowers. They are unisexual. Their stigma mature before the anthodeiasis. What do you think is the answer? Why do you think clistogamous flowers are self-pollinated? Any answers? Come on. Okay. Shailaja says the answer for this question is A. What about the others? Okay, A. Okay. Next, Sahana, yes dear, tell. Silent Power says it is A. Shada says, Ramya says A. Ma'am, I have doubt. Tell me, Abdul. Okay, Chetan, Nandakishore, Gagan, Chase Master, Chandana, and Anne Maria, and everyone have different Navya. Okay, perfect. Guys, if I'm losing out any of your names, I'm so sorry. Okay, so the answer for this question is going to be A. Clistogamous flowers means, Baba, they are closed. Clisto, C-L for closed. Always remember. Now, they show self-pollination. Why? Because they are bisexual. They have both the male and the female part. And Baba, when they are closed, do they have an option out there? No. Which is why the gametes are going to land up over here, because they do not have any options. So they are bisexual flowers. This is what you call as bisexual, when you have both the sexes together, and they do not open at all, which is exactly why they are going to do only self-pollination. What do you call the process of self-pollination as? Anyone who remembers? They lie close together in a closed flower. Exactly, Navya. And what do you call this process of self-pollination as? The term for it? Geitonogamy, xenogamy, autogamy. What do you call that term? What do you call that term? 39A, exactly. Next, Sahana, Darshan, Kishu, Sajan, hello dear, and Shiva Prasad, Abdul Munira, Bhagyashri, Pallavi, Shivani. I'm reading out your names now, but yes. Okay. You call that syngamy, eh? Autogamy, okay, you call that as autogamy. We call that as autogamy. Self-pollination is also equivalent to autogamy. Please remember that, do not forget it. Okay, and also remember there are 3 flowers which shows it. Cow is what I remember it. Camelina, oxalis and viola. Camelina, oxalis and viola. They have both clistogamous as well as chasmogamous flowers, open flowers as well. The open flowers over here, they can do all type of thing. Autogamy, geitonogamy, xenogamy, whatever they want to do it. Okay, perfect. Question number 40. Select the plant species where emasculation is not required for artificial hybridization experiment. What do you think is the answer for this? One day I'll put note for you. Huh? What is happening over here? Note for you. I didn't get you, Nisha. Okay. So, Comalina, oxalis, viola. Exactly, Shivani. And perfectly, Suganya, Anil Kumar, and everyone, Ramya, who gave me the answer for the previous one. As in... Okay, children, you know what? You are doing great. You are doing great, and you are just concentrating on the class. You're giving me the answer. That itself is a big deal. So, Yes, perfect. Sajan, thank you for the thumbs up. Okay, Silent says C is the answer. Panvit says 3, that is C. Shivani says papaya. Most of you guys are saying Sanjana, Pragna, and Maitri is also there. Anil Kumar is there. Okay, children, for those of you getting your answers wrong, check it out. The answer for this question is what? Select the plant species where emasculation is not required. What do you mean by emasculation? Over here, suppose you have a very pretty flower and you have the female part and the male part, and you do not want the male part. So what do? You do kachak. Why do you not want the male part? Because the pollen grains from here can land up over here and can do pollination, and we do not want that to happen. That is known as self-pollination. We want to prevent it. So we do emasculation. Now in some plants, you do not have the male parts, right? You call them as unisexual flowers or unisexual plants out there. So in those plants, you will not need emasculation. So you have to spot the unisexual plant out there. And the only answer for this question is going to be papaya. You have read it in your NCERT. It's mentioned that papaya is what? Dioecious. Dioecious means what? One plant is going to be a female plant only having the female part, and one plant is going to be a male plant only having the male part of it. So that is why it is known as dioecious. So the answer for this question is going to be C, macha. C is unisexual. Exactly. Papaya. Papaya is papagai. You can remember papagai. It is what? Single. Papagai is single over here. Okay? Cool. Works. Let's go ahead with the next question then. Papaya has different male and female plant. Exactly, Pallavi, Shraddha and Hitesh. Perfect, perfect, perfect. Okay. So shall we move ahead with the next question? If yes, give me a thumbs up, everyone. Now I like the class. Okay, I see that the crowd is just studying. Perfect. Okay. Ma'am, that is not Darshan, that is Pavni. Okay, sorry. Okay, ma'am, you're teaching. Thank you so much, Damacha. Okay, guys, let's go ahead. And I think thumbs up are coming through the way. And the 41st question. The 41st question is a previous year question. Let's check it out. Which of this option is true in context of the above diagram of the pollen grain? So this time it's below diagram of the pollen grain. Okay, just check it out. So below diagram is what? Over here, A and B is mentioned. First of all, when you have got a question like this and when they're talking about A and B option, look at this and please mark what is A and what is B. You know this is a very big cell out there, and in this very big cell, Baba, a small cell is floating. What do you call this very big cell as? A is going to be vegetative cell of pollen grain, yeah? And B, which is there, it is going to be the generative cell out there. So A is vegetative, B is generative. What is B generating? B is generating what? Gametes. It is generating gametes. What is there? What is A doing there? A is providing nourishment to B. Macha, you go generate gametes. You go generate gametes. That is what the B is doing. Okay. So everyone is saying D is the answer. Everyone knows the answer. Okay. What is D? A is vegetative cell with abundant food reserve, and B is a generative cell which forms the male gamete. Exactly. If at all you tag it properly, you will be able to give the answer. And now, one among you have to tell me this also. Okay. Which among them, okay, this is A and the B one, which among them is responsible for the formation of pollen tube? Anyone? A or B? Which among them is responsible for the formation of pollen tube? Any idea? Is it vegetative cell or the generative cell? Is it vegetative cell or the generative cell that is responsible for formation of pollen tube? What do you think? Hey Sundari ma'am, Mohammad Safan, thank you. Kyun theek ho? Oh my God, listen, see over here. Okay. None of them are A. Yeah. So listen, always remember, vegetative cell which is there, it is there. Why it is not coming? Yeah, vegetative cell which is there, it is for the nourishment of the generative cell to form the gametes. Apart from that, this guy is also responsible for formation of pollen tube. Always remember, that is why it is also known as tube cell. Tube cell. So if at all that particular thing comes up, you should be making sure that you check it out. Yeah? Yes. Okay, perfect. So let's go back and do the question. You know what? We have went in, we have got into the next session now. Ma'am, awesome. I'm loving your session. That pedigree analysis also, I'll never forget. Thank you so much, dear. Okay. So, guys, can we move ahead with the next question? Can we move ahead with the next question out there? which is from the next chapter, Human Health and Diseases. Yeah, I told, and everyone who gave me the answer, I know that you guys are telling me the answer, but the answers are just going away and I'm really bad with this, okay? I'm so sorry. You know what? Every one of you has 1.5 acres in my heart, okay? Okay, let's go ahead with the next question out there, and that is from the next chapter, Human Health and Diseases, one of my favorite chapters, and probably could be yours also. Okay, let's go ahead. Which one of the following options gives the correct matching of a disease with its causative organism and the mode of infection? Come on, let's do it. So typhoid, pneumonia, elephantiasis and malaria is given over here, and here you have the causative organisms and here you have the mode of infection. You have to say which one is correctly matching. Okay, matching. For me 2 acre. Okay, Kyun, for you 2 acre. Okay, guys, come on, do this question. Norman says B and Silent Power says D and next Sahana. Okay, Kishan says D. Silent B, Panvit says B, Chandana says, what are you doing between B and D? Come on, solve it. It can be only one answer, either B or D. What do you think is the answer for this particular question? Either B or D is the answer. Ma'am, B. Okay, so most of you guys are going with B. Let's check it out. Okay, typhoid, pneumonia, elephantiasis, malaria is given. The easiest one that everyone knows, okay, if at all you wake them up and sleep from the sleep and check, is basically malaria. Malaria is caused by Plasmodium vivax. Okay, it is bite of male Anopheles mosquito. Papa, male guy is saying, how I didn't even bite anyone. Okay, I am just feeding on the plants up out there. Why? How will I cause malaria? So this is going to be wrong. Okay, it's male. That is why it is wrong. Okay, let's go with the B one now. Pneumonia is there since you guys said B and D. Pneumonia is caused by Streptococcus pneumoniae. Yes, it is. Droplet infection, is it? Yes, if I do a chi and basically that chi lands upon you, basically you can also have that particular disease. It's a droplet infection. So definitely this is also correct. So I will say B is the correct answer. Let's see why others are not the correct answer. We have to check that also, right? So let's check that. Typhoid is caused by Salmonella typhi. Perfect. With inspired air. Aha, if at all it was that case, then probably every one of us could have got typhoid. It is not through inspired air. It is basically through the fecal-oral route, through contaminated water and things like that, food like that. Okay, now why not elephantiasis? Elephantiasis is caused by Wuchereria bancrofti. Yes, with infected water, food and bite of male Culexa. Again, male mosquito is being framed. It is actually the female mosquito, no bacha. So this is also going to be wrong, and mostly it is basically transferred through the mosquito bite only, not through food and water and stuff like that. Okay, so the only answer for this question is going to be B, and everyone who gave me the answer as B, you know what? You guys are super awesome. And then comes the next question, that is 43rd question. Come on. Love you, ma'am. Okay, Vijay, Suttagati, Kyun, Kishan Gowda, Shraddha, Nauman, Radha, and Madiha, Shailaja, and Jyoti, Darshan, and Pix video, and Chetan, Savita, Shivani, all girls are there. Whoa, girl gang is there. Whoa, Manjunath is boy, Nivedita, and It's Love, everyone, Ashraf, Praveen Mullah, Muhammad Shawan, and if at all I'm missing out anyone's name, I'm so sorry. Yes, cool. 43rd question, guys. If at all you can give me the answer like this, 43 A, B, C, D, it will be really good. Ayo, question for you. Okay, Nauman says B, Pragna, Panvit, B, Manjunath, Jyoti, okay, Shivani, Ramya. B is what? Morphine is often given to poop. Okay, so what is the correct statement is being asked? Barbiturates when given to criminals make them tell the truth. Barbiturates are basically what? It causes sleep. Okay, so people who are suffering from insomnia, who is not able to sleep, they are given barbiturates. Apart from that, people who are suffering from depression are also given barbiturates. To treat depression and insomnia, you have barbiturates, amphetamines, and barbiturates amphetamines and benzodiazepines, BAB, okay, for treating insomnia as well as the depression. Okay, that is wrong, that's why. Morphine is often given to a person who have undergone surgery as a painkiller. Yes, because morphine is a sedative and a painkiller. So this option is going to be correct. Now, cannabinoids induce irritability and slows down the body function. Does it? Okay, cannabinoids which are there, they basically increases the cardiovascular activity. So it does nothing to do with slowing down of the body. Cocaine is given to patients after surgery. Now you tell me, you go to a doctor and doctor gives you cocaine after surgery for recovery, which will be very nice, no? So cocaine is a hallucinogen and it increases dopamine. It is not given after surgery. So this is going to be wrong. The only answer for this question is going to be B, matcha. Is this particular thing clear? Okay, ma'am, lemon juice you want? Give. Okay. Ma'am, both typhoid and pneumonia can cause fever and chills. So, okay, so in the case of what do you say, pneumonia, you are going to get cough and stuff like that also. In the case of typhoid, you are not going to get that. Okay, that is one thing, one very important thing. Okay, ma'am, mehendi is allowed in the examination. I have to check, dear. I don't think so. I have to check. I really want to check. Cool, guys, if this question is clear, let's go ahead with the next one. Yes, perfect. Let's go. Question number 44. Which of the following are the techniques for detection of cancer of internal organs? This is a PYQ. Which of the technique is used for detection of cancer of internal organs? That is the question. Radiography and MRI, MRI computer tomography, Vidal test and radiography, MRI Vidal test. What do you think is the answer for this particular question? What do you think is the answer for this question? Ma'am, D, cocaine function. Cocaine is there for, it's basically, it's going to affect your central nervous system, dopamine transport. Okay, it makes you very, very happy. Extreme happiness is known as euphoria. It causes that also. It causes hallucination. You will start seeing things which are not actually there. Yes. And now answers are coming up, flowing in and coming, and everyone is saying A, Lokesh, Panvit, Shivani, Chandana, Manjunath, Kishu, Ramya, Shardha. Everyone says A, and some people are saying A and B are correct. My 3. Pragnya. Okay, so let me check it out. If you want to find the cancer of internal organs, first of all, you have 3 things. Radiography, where you simply use X-rays, okay, for just understanding the structure and things like that. Then you have CT scan, where again you are going to use X-rays, but for what? But for understanding the 3-dimensional image of your internal organs, okay. Then you have MRI, where you are not going to use X-rays, you use non-ionizing radiation, that is safe radiation, and also you use magnetic fields for getting the understanding of living tissue. Anyway, Baba, with the help of these 3 things, we are getting understanding of the internal organs, which is why these 3 of them will be the answer. So radiography and MRI correct. MRI computer tomography correct, because computer tomography is CT only. Okay. Vidal test is for typhoid. Why is it over here? Again, Vidal test is over here. Wrong. Which is why the answer for this question is going to be A and B. The answer for this question is A. And everyone who gave me A as the correct answer. Okay, ma'am, please complete before 3:30. Match is there. My life is under risk. And you want to see match? You tell me. Okay, but I'll try my best, okay, so that you can go watch match also. Guys, can we go ahead to the next question? If yes, give me a thumbs up. Quickly, give me a thumbs up. Come on. And everyone who have not clicked on the subscribe button yet, click on the subscribe button, click on the like button, and yeah, that's all. Yes, give me a thumbs up. This question already done. Yes. So PYQs are also being repeated, okay? It's not like I'm just giving new questions. PYQs are also there because PYQs, you know, PYQs are important, right? Yes, only 2 thumbs up until now. Okay, I'm getting thumbs up. Okay, perfect. Let's go ahead. Identify the most infectious and fatal type of malarial parasite. Which is the fatal type of malarial parasite? Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, Plasmodium malariae. Which one of them? If at all you get it, you are going to like tring. Which is the thing? Yes, Deepa, you can definitely give more importance to the PYQs, okay? Okay, 45A is the answer, says Sanjay, Deepa, and Bhagyashri, Bhavish, and Shraddha, Kyun, Silent Power, Hitesh, Savitra, Panvit, Anu Patil, Jeevan Kumar, Ajay Kumar, and I can rock like this. Okay, Norman, perfect, perfect, perfect. So everyone is saying A, Plasmodium falciparum is the answer. There is no need for explanation. This is the one that causes the fatal or the malignant form of malaria. Very, very important. And everyone has to be pretty sure about the malarial life cycle. Very important concept out there. Do not miss out on it. Okay, do not miss out on it. Anyone wants to understand the malarial life cycle concept? If yes, give me a vote. If at all yes, give me heart. Give me heart as in the chat session itself. Okay, if you want malarial concept understanding or the important things that can come in. Ayyo. Only if I get heart, I'll explain that. Otherwise, I'll move ahead. Critical form of malaria. Explain this. Yes, I did. Oh, my dear children, I cannot explain. Okay, so I'm getting hearts. Some people are saying no. And okay, you see the number of hearts are more, right? So I have to explain. I'll explain fast. Okay, Pakka promise I'll explain within 2 minutes. I know that some people are there who have heard the story so many times. But yes, there might be new children. Yeah, and we have to cater them also. I'm so sorry. If for 2 minutes you drink water. Okay, so listen to me carefully. I will not repeat it for anyone. Plasmodium. Okay, Plasmodium. So there is mosquito. Key. Okay, and this mosquito, Baba, so this mosquito looks like something else. Anyways, in the salivary gland of this mosquito, you have Plasmodium. Chang-cha-chang. And this Plasmodium is in a very fatal stage. What do you call this stage as? Sporozoite stage. Sporozoite stage. Okay, sporozoite stage. It's there in the salivary gland. Now what happens? It comes and bites Papu. It comes and bites Papu. When it bites Papu, what is going to happen? The mosquito which is there, it is going to inject its saliva into our body. Why? Because the mosquito saliva has an anti-clotting enzyme that will let it drink the blood very, very easily. But along with saliva, who goes inside? The sporozoite which is there, it goes inside the body of Papu. Now, inside the body of the Papu, sporozoite is going to go where the first target is going to be liver cell. Okay, in the liver cell, they are going to replicate. Basically, they increase their amount. What are they undergoing? They are undergoing asexual reproduction. So in the human body, only asexual reproduction, no other type of reproduction is ongoing. Okay, so asexual reproduction is happening up over here. What will happen? What do you call this liver cell as? Liver cells are known as hepatocytes. Okay, hepatocytes. Anyways, once asexual reproduction happening, this cell is going to burst open. And when it bursts open, what is going to happen? These guys are going to be released. When these guys are going to be released, they go to the next place out there. What is the next place? The next place is the most famous place, that is RBC, red blood cells. What happens in the RBC? In the RBC, Baba, they have the most important thing which is known as hemoglobin. And they want to eat the hemoglobin, not the hemoglobin, exactly just the globin part. So what do? They cleave it. They cleave it into what? Heme and globin. Heme and globin. And what else is happening over there? These guys are still over here. What are they undergoing? They are undergoing asexual reproduction again over here. So they are eating, they are undergoing asexual reproduction also. Also remember, when they break hemoglobin into heme and globin, heme is very toxic for them, which is why they are going to convert the heme into something else. What do you call that as? You call this as hemozoin. They convert into hemozoin. Very, very important thing. Now listen, asexual reproduction is happening. Hemozoin is also made in the RBC. One day the RBC is going to burst open. Okay, again, because so many cells are there. When they burst open, what all things are going to be released? These cells and also the hemozoin. Hemozoin which is going to be released is toxic for us, which is why we get what? Fevers and chills in every alternate days, in a few days, in the case of malaria. What happens over here? What are these cells known as? Listen to me. Asexual reproduction happened over here. Asexual reproduction happens in the RBC, and when asexual reproduction happens over here in the RBC, the cells that you're going to get, you have a name for it. You call them as gametocytes. What do you call them? Gametocytes. Now, where are these gametocytes? They are now in our blood. Again, one mosquito will come. Zzz, will take our blood. When it takes our blood, When it takes our blood, it takes in what? The gametocytes. It takes in the gametocytes. Now the gametocytes which are there is there in the mosquito's gut. Just imagine that I am the gametocyte and I am inside the mosquito's gut. I am going to make gametes over here. I'll do sexual reproduction over here. Okay, the gametes which are there, they are going to sexually reproduce, or basically they are going to fuse in the mosquito's gut. In the mosquito's gut, this little peto there. And then finally, when the gametes are being fused, they form something that is known as ookinet, and finally they form a stage that is known as sporozoite, and this sporozoite moves to the salivary gland. So the question that is usually being asked is what? First thing that is being asked, in what stage does it affect us? Sporozoite. In what stage does it leave our body? Gametocytes. Where are the gametocytes being formed? RBC. Does asexual reproduction happens in our body? Yes. Does sexual reproduction happens in our body? No. Where does the sexual reproduction happens? Inside the mosquito's gut. Got it? These are the few questions that comes. And also, what is hemozoin? Hemozoin is something that is toxic for us. It causes fevers and chills, but it is made for by this particular guy itself. Ayyo. Is this clear? Is this clear to all of you? Okay. Baba, it's not just cellulose. There are a lot of things out there. Okay, it's not just the cellulose, it's just a lot of things out there in a normal plant cell. That's why it is very, very thick. Okay, we can digest to some extent, but not much. Yes, perfect. Ayyo. Now you give me glucose. Tomorrow you will find me with a drip. So instead of going for a trip, I will be on a drip of glucose. Okay, perfect. Thank you so much. And the next question. Thank you. Injection of an antidote injection against snake bite is an example of. So when we get antidote against snake bite, okay, what is this is the example of what? Autoimmunity, active immunity, innate immunity, passive immunity. What do you think is the answer for this? Ma'am, is cockroach and the families in KCET? Lata? Whatever is there in your NCERT, that is only there for your KCET examination. Rest is deleted portion. Okay? Okay. Let it be. I'll ask you later. Okay. D, Panvit, Silent, Power, and Savita, Sharda, Sidhu, and Lakshmi, Shailaja, Hina, Kausar. Everyone says D. Some people are saying C also. Minnu, hi. So the answer for this question, listen to me very carefully. Snake is there. Snake comes and bites us. Okay, snake comes and bites us. For the very first time it comes and bites us, our body does not know how to react. It will produce antibodies, but not much. Not much. The primary response is always slower, which is why we go to the doctor. Doctor gives us what? Antidote. Basically, he gives us preformed antibodies, ready-made antibodies against it. So when you're getting ready-made antibodies and when your body is not producing antibodies by itself and protecting you, what do you call this as? This is passive immunity. This is what? Passive immunity. If your body is producing antibodies and you're safe because of that, active immunity, but you're getting ready-made antibodies, it is passive. The other example for this is going to be tetanus toxoid. You go and have tetanus injection. No. What is tetanus injection? Again, the antibodies, ready-made antibodies. Got it? And this is artificial also. You're getting it from outside. Okay, got it? Natural is, for example, from the mother to the child and all, you get antibodies. No, that is natural passive immunity. Again, baby is not making itself. The mother is passing. That is why passive. Natural there because the mother is giving, right? Over here, we are artificially giving. Ma'am, when is chemistry class? Dada, chemistry class is already done. Now chemistry class will be there on Tuesday. Yes, you can watch the chemistry class in 2x speed, the previous ones. Ma'am, innate immunity is something that is there with us from birth. For example, their antibodies and things like that do not play any role. For example, your skin, your tears have some enzymes. There are some cells, for example, neutrophils, natural killer cells, macrophages. These are something that provides protection from the starting point onwards. They are non-specific. They can kill anything out there. Okay? So yes. Cool. Colostrum comes under passive immunity from mother to the baby. Okay, ready-made antibodies, but natural. Cool. I hope this is clear. If it's clear, can you give me some thumbs up? If it's clear, can you give me thumbs? Can you give me thumbs up? And Shailaja, drink some water. Thank you so much for reminding, dear. I drink very less water. Always. Very bad thing. Next. Okay, question number 47. A person shows symptoms like sneezing, watery eyes, running nose, difficulty in breathing, exposure to certain substances in air. Oh, this is common cold. No, this is something else. This is allergy, right? Which of the antibodies produced during such conditions? You have antibodies being mentioned over here, and you have to tell that which of them is being produced. Your teaching is fabulous. Thank you so much, Madira, and thank you so much, guys, for giving me the thumbs up. I love you a lot. Okay, so, and I owe you a lot. Come on, give me the answer for this. B. Lakshmi says B. Hitesh says B. Sheikh is saying B. Okay, Bhagyashree says B. Ashraf and Biru, Tarun, Sharda, if at all I'm missing out anyone's name, Viresh, Silent Power, Deepa. Okay, so B is what? IgE. Always remember, allergy. Allergy ends with E, so IgE. What does Ig stand for here? Everyone should know it. As antibody, we know it is basically immunoglobulin. It's a protein. Immunoglobulin, it's a protein. Do not forget it. Okay, allergy is an important topic. We can get questions. What happens in allergy? Antibodies are there on what? On mast cells. Mast cells not, it is known as mast cells. Okay, so IgE is on the mast cells. It comes in contact with some allergens out there. It sends signals. Mast cells is now going to produce two things, histamine. Okay, histamine and serotonin. What does the histamine and serotonin do? They lead to vasodilation. Your blood vessel, which was chutku like this, will become like this. Too much space will be there. Why? Because it thinks that it is emergent situation and you need to provide space so that all the other cells can come over here. This is over exaggeration actually, right? And always remember, what are the things that you give against allergy? You're going to give three things and you might forget it, which is why I'm reminding you once more. Steroids, adrenaline, and antihistamine. Histamine, antihistamine. So steroid and adrenaline is something that you might miss. Steroid, adrenaline and antihistamine is something that you give against this. And you guys are absolutely correct. Vasodilator, heparin, secreted by mast cells, immunoglobulins, perfect, macha. Okay, so the answer is correct. Can we go ahead with the next question? Yeah, if yes, give me a thumbs up. If yes, give me a thumbs up. Okay, perfect. Next question is this. Okay, this is from PU1. Come on. Okay, PU1 chapter, photosynthesis in higher plants. In the maize plant, CO2 fixation occurs in both mesophyll and bundle sheath cells. The enzymes involved in these cells for the process respectively are. I have taken those PU1 chapters which gives almost similar type of questions. So do not miss it up. Okay, do not miss these type of questions. In maize plant is what? Is a C4 plant. Okay, C4 plant. And always remember, in C4 plant, the arrangement is what? You would be having mesophyll cells and you will be having what? Bundle sheath cells. First mesophyll, then bundle sheath cells. Over here, this type of anatomy is known as Kranz anatomy. Why? Because, Baba, there is an enzyme that is known as rubisco. If at all it comes in contact with oxygen, it will make the bond really fast. It will do the process of photorespiration. And you want to avoid that. You want to avoid the rubisco to come in contact with oxygen, which is why you make sure rubisco is far away from the leaf surface out there. Okay. So what exactly happens is that the CO2 is initially fixed in some other cell that is known as mesophyll cell, and this fixation happens with the help of an enzyme that is known as PEP carboxylase, phosphoenol pyruvate carboxylase. Please remember it. So PEP carboxylase is over here. With the help of PEP carboxylase, initial fixation of CO2 happens. That means CO2 is being combined with phosphoenol pyruvate. You get oxaloacetate, and then later it forms malate. Anyways, the CO2, guys, is here itself. The CO2 is being fixed here in this particular format. Now what do? Malate, which is there, it is being sent inside. It produces CO2 now. Now the CO2 is being fixed again by rubisco in the bundle sheath cell. In the bundle sheath cell, it is going to be fixed by the rubisco. So always remember, mesophyll cell, it is PEP carboxylase. In the bundle sheath cell, it is going to be rubisco who is going to do the whole process. And that is the question. The enzyme involved in these cells for the process respectively are what? It is going to be PEP carboxylase and rubisco. The answer is going to be D. Please remember, it's Kranz anatomy, and this is to make sure photorespiration is not happening, and this is found in the case of C4 plants. This is a very important concept. Do not mess it up. So many questions have come from the same concepts. Yes, malic acid is transported. Exactly. Malate, malic acid is the same thing. Perfect. Got it now, macha? If yes, give me a thumbs up. If it's clear, give me a thumbs up. Everyone who gave me D as the answer, Tanu, Kalyan, Shivani, Manjunath, and Sheikh, and Kishu, Panvit, and yes, Praveen, Ajay, and Shivani, perfect. Hitesh, okay. Chase Master, Savita. Cool. Question number 49, this is, okay, so I've taken just one question from here because I think that was the only concept which was being repeated, rest everything was new things. Now from respiration in plants, this concept is something that is super important. Let's do it. Okay, let's do it. So I had tried to cover all the chapters, Deepthi, over here. Okay, at least one questions will be there from the PU1 also. Most of the chapters I've covered. Yeah? Okay, when tripalmitin is used as a respiratory substance in aerobic respiration, the process consumes 145 molecules of oxygen and releases 102 molecules of CO2. Then the RQ value would be respiratory coefficient. This is a question that is being asked, and please remember you have to know the formula. If you know the formula, Baba, you know everything. I do not know the question, it's okay, Sheikh, we'll solve it together. Okay, so this is how it is going to go. The formula is like this: the amount of CO2 which is going to be evolved, which is going to be produced, and over here the amount of oxygen which is going to be consumed is going to give you the respiratory coefficient. Over here, the amount of oxygen that is consumed is what? 145. And the amount of CO2 which is being released is what? CO2. In respiration, obviously, CO2 is released and oxygen is utilized, right? So you can remember like this, don't need to mug it up, okay? Now, Baba, if at all you do the formula and if at all you do the calculation, it comes out to be what? It comes out to be 0.7. But you do not need to do all this calculation. You can easily get your answer. How? Tripalmitin, which is there, is basically coming under fat, and fats have a respiratory quotient of 0.7. okay, carbs, which is there, it has a respiratory quotient of 1, that means unity, and fat is what? 0.7. what about proteins? It is 0.9. this is fixed, and this particular formula is also fixed. If you remember this, everything is sorted, and I told you a way to remember this as well, if at all you were there in the previous session. And it goes like this. Listen to me very carefully. When you write carb, okay, carb I cannot say, it's easy, 1, it's just 1, please remember it. When you talk about fat, you make the ulta of F, it looks like 7, right? So 0.7. Similarly, proteins, if at all you make ulta of P, it looks like 9, so 0.9. If you can remember this, it will be really good, it could be helpful. Yes, perfect. Ma'am, I do not know anything about first PU. It's okay, Macha. This is how we learn. Yeah? At least remember the important things that could be coming in. There are chances for this to come in. Yeah? So, give me a thumbs up if it works, if it's understood, everyone. I have given you the formula. You can get this particular PDF. This is going to be helpful for you. Do not mess it up. Okay? Perfect. You're most welcome, guys. Okay. So, question number 50, during the citric acid cycle, the various organic acids undergo decarboxylation, as in they lose the carbon dioxide. Okay, they lose the carbon. Which of the following organic acids of the above cycle has 4 carbon, 5 carbon, 6 carbon, respectively? And over here, you have got the options. You have to choose from those options. Okay, you have to choose from those options out there. Come on, tell me the answer for this. 4 carbon, 5 carbon. Citric acid cycle is also known as what? The Krebs cycle or the TCA cycle or whatever cycle you want to call it. And over here you have been asked 4 carbon, 5 carbon, 6 carbon respectively is what? Always remember oxaloacetate, malate, succinate, everything is going to be 4 carbon, 4 carbon, 4 carbon. Okay, citric acid, which is there, we know it's the first 6 carbon compound that is being formed. The first 6 carbon compound that is being formed is what? Citric acid. So over here I just have option this and option this. If at all you look at this, okay, citric acid is here, citric acid is over here. Probably my answer could be somewhere between this and this. Between this and this. Okay, you do have pyruvic acid? No, Baba. You just have succinic acid, which is known as succinate. Okay, and that has 4 carbons. Alpha-ketoglutarate acid is having 5 carbons. And yes, citric acid, which is there, it has 6 carbon. Now, if at all you use a bit of logic, it will be easy for you to understand. But still, if you want me to help you, listen to me very carefully, okay? Oxaloacetate is over here, 4 carbons. Okay, and one guy comes into the picture, that is acetyl-CoA, and says that, can you bind with me? Okay, so acetyl-CoA that having 2 carbon and oxaloacetate having 4 carbon, binds to produce the first thing, that is citrate. That is why you call it citric acid cycle, or it is also known as tricarboxylic acid cycle. Okay, you anyway got the citrate. Now what happens? The citrate which is there, it converts into isocitrate, and for the first time it is going to lose the carbon. If one carbon is lost, that means carbon dioxide is lost, that means one carbon is lost, right? So 6 which becomes what? 5, and that is found in alpha ketoglutarate, right? Again one more carbon is lost, and from this point onwards everything is going to be 4 only. So alpha ketoglutarate acid forms succinyl-CoA, then succinate, then fumarate, and then finally malate, and then again we reach up to oxaloacetate. So always remember, these guys are 6 in number, here you get 5, and from here you get everything to be 4. Cool. Is this particular thing clear to you? Very important. You might get questions on the basis of especially these part, acetyl-CoA, oxaloacetate. The first guy to accept the acetic acid over here would be oxaloacetate. All these things remember. Okay. I read those names, but I'm scared for the examination. You will do good. Trust me on that. Okay. Just when you get questions, think. Do not be scared. Think about it and reconfirm it. That's all. Guys, if this is clear, can you quickly give me a thumbs up? If it's clear, can you quickly give me a thumbs up? Yes, perfect. And thanks everyone who was saying CCC. Option given is wrong. No, it's correct only. Succinic acid is also known as succinate. It's the same thing. Succinic acid, succinate is the same thing, okay? So 4 carbon, 5 carbon and 6 carbon. Okay, perfect. Thank you guys for thumbs up. Let's go ahead with the next PU1 chapter, plant growth and development. Identify the incorrect statement from here. In plant growth and development, all the unique features about the hormones are something that is super important. Okay, ma'am, what about the rough work in examination? You have to do it in the rough column that is there in the question paper. Okay, in the question paper, you will find an area to do the rough work. You can do it there. Cool? Yes. Okay, let's do it. Identify the incorrect statement. Guys, kinetin is a derivative of adenine, which is a pyrimidine. Adenine, is it a pyrimidine? Adenine, with what I know, is what? Purine. Okay. Oxaloacetate is 4 carbon, ma'am. Yes, it is 4 carbon, dear. Did we do something wrong? Yeah. Succinic is also 4 carbon. No. You have to choose from this, right? Okay. Anyways, so where are we? Yeah, we are doing this question. D. Most of you are saying D is the answer. Nisha, Sadvika, Nisha, and yes, Panvit, everyone. So D is what? D is going to be 1 and 5. Let's see. Kinetin is a derivative of adenine, which is a pyrimidine. Wrong. Adenine is what? Purine. And kinetin is a derivative of adenine itself, but adenine is what? Purine. Kinetin is the one that is basically coming under cytokinin. Okay. The technique of decapitation is widely used in tea plantation. Decapitation means what? Suppose the tea is over here. The top portion of it, I cut it off. Why do I cut it off? Because, my dear children, in the top portion, you have auxin. And auxin is responsible for what? Apical dominance. That means it makes sure the plant grows like this. Not like this, like this. That is known as apical dominance. Very unique feature about it. Please remember it. Now, the tea plantation people want the plant to grow like this, laterally, not like this. So, what do? They cut it off such that it can go laterally. So, technique of decapitation is widely used in tea plantation. This is correct. Second statement is actually correct statement. You have to find which is incorrect, right? So first one was incorrect. Okay. Ethylene is a gaseous plant hormone. Yes, it is a gaseous plant hormone out there. The only gaseous plant hormone. So this is also correct. Use of GA3, that is gibberellin, hastens the malting process in brewing industry. Do you think gibberellin is responsible for malting process? Yes, it is responsible for malting and also bolting. Malting and bolting. Okay, malting basically means it allows the germination process to happen quickly. Not germination, yeah, the starch breakdown to glucose happen quickly. That is known as malting. And bolting means you might have seen that it forms what do you call rosette plants. Okay, you might have seen in the case of cabbage and stuff like that. Okay, so those are the two things done by G3. So that is also correct. ABA is a growth promoter. Wrong. ABA, abscisic acid, is a growth inhibitor, not a promoter. So basically, what can I say? 1 and 5 is the only one, which is why, as you guys said, D would be the answer for this particular question. If at all you want to know the functions of all of it, you can just have a look at this particular thing which is there when you get your PDF. Okay, so please do that. Clear? Guys, is this particular thing clear? Can we go ahead? If yes, give me a thumbs up. If we can go ahead, give me a thumbs up. Yes, perfect. Next one. Auxin leads to apical dominance. Juvelin leads to what? Adventitious root formation, accelerates abscission, closure of stomata, or bolting. What do you think is the answer for this? It's a very easy question. Come on. Do it. Thank you for giving me thumbs up, guys. What do you mean by this? Okay. Most of you saying Q. Q is what option? I think there's a typo. D. Most of you guys are saying the answer for this question is D. And D is what? Bolting. D is what? Bolting. Bolting means what? Premature flowering. It is allowing the plant to form flowers really fast. Okay, the process that leads to what? Premature flowering. That is known as bolting. And as I said, molting and bolting, both of them are being controlled by gibberellin. So the answer for this question is going to be D. Yes. Hooray. Okay. Sudden rapid elongation of the stem just before the flowering process, so that the flowering can be initiated really fast. Yeah, and it is seen in roseate plants such as cabbage and lettuce, and yeah. Okay, before I go ahead with the human reproduction chapter, I want to ask you a question. So this question we solved it previously in the previous life, but I want to ask you, see who were there and who all remember it as well. Okay, rice is there. Okay, rice is there, and rice is grown in paddy field. And flooding is happening over here. Which is that particular hormone which will save the rice plant from flooding and help it in growing up? What is that particular enzyme that is going to make sure that over here, if at all I say this is a node, it leads to internodal elongation in rice plants which are flooded in water? What do you think is the answer for this? I read only PU2, so I'm scared. Srinivas, I'll tell you one thing. Listen, with this particular point of time, you do not have much time to invest on your PU1. Solve the question, previous year question, you will get some idea about what in some chapters it will be repeated, in some chapters repeated concepts are not there, so obviously you cannot do anything about it then, right? Auxin, ethylene, who said ethylene? Nivedita said ethylene is the answer. Listen to me very carefully. Over here, your paddy field, okay, in the paddy field, it's full water. Now the plant wants to grow, stop away from it such that it can sustain, right? So for that, what exactly happens is that the plant is going to produce ethylene. It's a gaseous hormone, and since it is a gas, it cannot diffuse. It remains at the same position, and when it remains at the same position, ethylene does what it mostly does not, and that is internodal elongation. So it is ethylene as the answer for rice. This question we did it previously also, and Shivani and Pallu and everyone who gave me the answers along with Nivedita, perfect. And for those of you who do not know it, it's okay. It's okay, okay? We learned it today, and that's a new thing. Please remember it. Cool? Petiole elongation. Yes. Perfect. So guys, are we ready to solve some PU2 questions now? And I think when we go ahead and solve these questions, I will not be having any PU1 for some time now. I think so. If you're ready, give me a thumbs up. If we are ready, and we can go ahead with the next thing. Perfect. So let's go. Over here, 53rd question. Match the months listed in column 1 with the organogenesis of fetus in column 2. So over here, you have the months that is given, and you have to choose what is going to happen in which month. Okay, first month, second month, fifth month, sixth month. So everything is given to you. Come on, matcha, choose it. So these questions are coming for KCET. We expect that at least the concepts could be coming in. So yes, keep on doing it. Probably it can pop up there, right? Okay, Nauman says A is the answer. Savita says A. Sri Krishna says A. Shiv Prasad A. And Shardas A. Hitesh. Everyone. Shubhan. Everyone says A. Okay, some people are saying B also. Okay, Pallu A. So the answer for this question, let's check it out together. Okay. First month, when someone dies, we check the heartbeat. When someone is formed, we also check the heartbeat. So basically, first month, heart is going to be formed. A goes with 3. A goes with 3. I have 2 options. I can cut off these 2 options. Now, I try to find the answer from the options. Okay, D goes with what? D goes with 1 over here. 6th month, Baba, you know, eyelids are going to separate, right? Eyelids are going to separate. So D goes with 1, D goes with 1. And basically, I have got my answer. Answer is going to be A. Answer is going to be A. For everyone who are not very, very confident about organogenesis, listen to me carefully. I am going to talk about it right now. Okay, so this is my baby Pappu. Okay, this is baby Pappu over here. In the first month, Baba, what do you find? You find the heart. You find the heart. In the second month, which is there, so this is for the first month. In the second month, what is going to be there? Basically, Pappu will be having limbs and also digits, that means hands as well as the fingers are there in the second month. In the third month, what is going to happen? In the third month, basically, there will be major organs in the organ system being formed, and also apart from that, the external genital organ is also being formed. External genital organ, and also apart from that, as I said, major organ system is also going to be formed. Fourth month, skip Madi. Fourth month is not there in NCERT, so you do not need to be worried about it. First month, as I said, heart. Second month, limbs and digits. Third month, most of the organ system and also the external genital organ. In the fifth month, fourth month is skipped. Fifth month, baby says that I want hair on my head and I also want to move around. So basically, God says you want to move around then. So basically, in the fifth month, you will be finding the first movement. Okay, the first movement of the baby in the fifth month. Apart from that, the first hair on the head is also there in the fifth month. Okay, now what happens? Basically, one more thing happens, and that is, God said that you wanted hair, no? Have hair on your body also. So basically over here, hair will be there on the body. In the 6th month, hair on the body. Apart from that, what? Basically, eyelids are going to be separated. Eyelid separation, and apart from that, eyelashes are also going to be formed. Eyelashes are also going to be formed. Now I know it looks like a Dracula kid, but yes, this is the answer. This is one very, very important thing. Do not mess it up. Always remember, third month means 3 months means how many weeks? In 1 month we have 4 weeks. In 3 months, 3 into 4, basically we have 12 weeks. So no week by week data. In 1 month, 4 weeks. In 2 months, 2 into 4, basically 8 weeks, and that is how it goes. 3 months is known as first trimester, and over here 6 months is known as the second trimester. Cool? Yes? Guys, what is happening? Whom are you called creepy? I hope it's not me. Okay? Shankar, this is not your... Okay. Okay, guys, listen to me carefully. Do not fight amongst yourself. You are studying something, and please make sure you concentrate here. Yeah? And please tell me the name of the person who is disturbing you. I'll put them on timeout. Yes. Sagar, hello. Okay, so I hope this is clear. Yes, perfect. Okay. Next question. Which one of the following is incorrect matching of the events occurring during menstrual cycle? So menstrual cycle is given over here. You have to tell which one is incorrect. Incorrect is being asked. Okay, incorrect is being asked. So you have to choose the incorrect one. Okay. Proliferative phase, secretory phase, menstruation, ovulation is given. Proliferative, secretory phase, menstruation, ovulation is given over here. You have to choose the correct answer. Come on. Hello Sudarshan. Hello Saida. Hello. Okay, Nauman says D is the answer for this question. What about others? Okay, Shadas also says D, Nauman says D, Mahantesh also says, Mahantesh says a different answer, Panvit D. Okay, listen to me. Proliferative phase, maturation of the graphene follicle and regeneration of the endometrium. Yes, in proliferative phase, Baba, 2 things happen. Okay, one is what you call it proliferative. Why? Because the endometrium, which was just fallen down during menstrual cycle, it is regenerating. You also call this as maturation of graphene follicle, because in the same phase, Baba, you also call this as follicular phase. The graphene follicle is keeping on maturing, and the follicles are the one that releases estrogen, which helps in regeneration of the endometrium also, right? Yes. So this is correct. Secretory phase. Secretory phase is also known as luteal phase. Luteal phase. Okay. Development of corpus luteum happens over here. Yes, definitely it happens. Increased secretion of progesterone. Do you think? Yes. Why? Because the corpus luteum which is there, Baba, it releases progesterone. Okay, it releases progesterone, and that is why, and the progesterone which is there, it is going to act on the endometrium, and the endometrium is going to secrete certain very important things. That is why it is known as secretory phase also. This is also correct. Menstruation, breakdown of endometrium lining and its blood vessels. Yes, you know in menses what is going to happen? Basically the blood is going to be broken down, endometrium is going to be broken down. Finally you have what? You have ovulation. In ovulation what happens? LH and progesterone attain the peak level. Wrong. Ovulation means what? The graphene follicle which is there, it is going to rupture. It is under the influence of LH and also estrogen, not progesterone. Progesterone is not in the picture until luteal phase. Okay, so it is going to be estrogen and not progesterone, which is why over here the option is going to be wrong. The answer for this question is going to be D. Is it clear? Is it clear? Clear, guys? If it's clear, just give me a thumbs up. And who is disturbing whom? Oh my God. No one disturb my bacha log, okay? If at all you disturb my machas and machis out there, probably I will have to time out you. Okay, anyways, okay, perfect. So you have given me the thumbs up, so I believe that you have understood the concept, which is why I'll go ahead with the next question out there, and that is this. Choose the correct sequence of sperm transport during ejaculation. What is the correct sequence of sperm transport during the ejaculation? Come on, come on, come on. A, B, C, D is given to you, and you have to choose your answer. When we were kids, me and my brother, we used to complain like this to our parents. So, just like that I'm feeling as of now, guys. Okay. D, 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 A, D, 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 D. So, most of you are giving A and D. Let me check it out. Seminiferous tubules, rete testis, vasa efferentia, epididymis, vas deferens, and ejaculatory duct. That is correct, no? Rete testis is there, it sends to vasa efferentia. From the vasa efferentia, it go to epididymis. From the epididymis to vas deferens. From the vas deferens to ejaculatory duct. A is your answer for this question. Why did you say D? Let me check out. Other options are wrong. Okay, why they are wrong? Seminiferous tubule, rete testis, then epididymis. Where does vasa efferentia go? Wrong. First rete testis, then vasa efferentia, then epididymis. Okay. Seminiferous tubules, vasa efferentia, rete testis. Rete testis has to come first. Okay, rete testis, epididymis wrong. So always remember, guys, the answer for this particular question is going to be A. For those of you who do not remember, listen to me. Here you have the testicular lobules. Yeah, inside that you have the seminiferous tubules. These tubules join up together to form the rete testis. This is the rete testis, right? This guy says that I want to move out, I want to exit, I want to exit. So E for exit, E for vasa efferentia. Yeah, and then finally they are trapped inside the very long epididymis, that is 6 meters long. Finally they are going taken to distance with the help of vas deferens. And lastly you have the ejaculatory duct, right? So the answer for this particular question is going to be A. Okay, is this clear to all of you? Yes? If yes, just give me a quick thumbs up. If this is super clear to all my dear children, give me a quick thumbs up. And Shankar eats love and picks. Guys, what is happening over here? You are grown up. Yeah? And do we need to fight like this? That too in a live session with thousands of children. Not thousands, 76, don't worry. But still you should not be fighting, okay? So, yeah, perfect. Let's go ahead. Question number 56. Match the hormone with the sites of production. So, over here you have HCG and HPL, progesterone, androgens, and relaxin. And over here you just have to match it with the hormones. The reason why I have put this question is that, Baba, you might get hormone-based questions coming from this particular chapter. Sanjeevani, why half up and half down, da? Okay, I hope it's fine. Okay. Hitesh, I cannot go faster than this much. Please understand. Okay, I'll die standing over here. A. Okay, Hitesh says A is the answer. Nauman says A. Shraddha says A. Pallu, silent power. Okay. Everyone says the answer for this question is A. Bio with Divya. Hello. Okay. Someone with bio name. Nice. Pavitra, Asfia, everyone. Deepthi, what happened? Okay. So, okay, fine. A is what most of you guys are saying. Let's check it out. Whenever match the following question comes, I always tell match A and D first, and before doing all those things, read the options carefully. So you have ovary, you have placenta, you have corpus luteum and Leydig cells. I'll say go the other sideways. Okay, rather than solving this way, solve the other way. So placenta over here. Placenta, you know, basically it is going to go with HCG and HPL. We all know that, yes. Leydig cell which is there, we all know it is responsible for only one thing, and that is formation of testosterone. And testosterone come under androgens. With this, can we tell the answer? A goes with 2. A goes with 2, A goes with 2. I just have one option like that, and basically 4 which is there, it goes with C. 4 goes with C. This is going to be my answer. For those of you who are not able to get it, listen to me out carefully. How does it go? So HCG and HPL, human chorionic gonadotropin, human placental lactogen, are placental hormones. Progesterone is over here, and progesterone, Baba, is released by ovary also. But you have got another option, corpus luteum. And corpus luteum releases only one thing, and that is this. Whereas ovary can release multiple things, so we can give that option later priority. Right? Androgens, the male sex hormones, are going to go with Leydig cells, and relaxin which is there, it is going to go with ovary. Ovary is releasing relaxin during the later phase of pregnancy, such that the ligaments can relax and the baby can easily move out. Okay? Got it? Understood? Hi ma'am, lunch matcha? No lunch matcha today. Okay, so is it clear? Is this particular question clear? If yes, give me a thumbs up. Most of you started giving me thumbs up. Now you guys know. Don't waste any time. You started giving thumbs up before only. Okay. Next question. Question number 57. If 2 primary spermatocytes and 2 primary oocytes undergo meiosis simultaneously, what will be the ratio of spermatozoa and the ova produced at the end of gametogenesis? What do you think is the answer for this? How many spermatozoa and ova are going to be produced? Come on. Thank you guys for thumbs up. Lots of love to all of you. Yes. And I miss talking to you, but cannot do it anyways. Okay. So one spermatocyte which is there, Maba, okay, it is 2N in nature. It undergoes division. You get 2 secondary spermatocyte, right? It is going to be haploid in nature. It undergoes division. What do you get, Baba? At the end, you get 4 cells. What do you call these cells as? Spermatids. So basically, from one cell you got 4. From one cell you got 4. What about primary oocyte? Listen, primary oocyte is there. 2N was there. It underwent division, but it is unfair division. One cell is small, one cell is big. Both of them are haploid. This is known as the first polar body. It goes off. It degenerates. What happens to this? This is the only secondary oocyte which is present. Okay. Again, when it divides, meiosis 2 happens. Unequal division, unfair division. Here N and here also N. But this is the only one that survives, and that is the ovum. So from one cell, you got only 1 egg. Okay. From here, but one cell, you got 4. So what is the question? What will be the ratio? Spermatozoa and ova produced. Spermatozoa first, that is 4, and ova second, that is 1. So 4:1 ratio is equals to, your answer would be B. And everyone who gave me the answer as B, okay, you guys are super awesome. Baba, drink water. Thank you. Yes, clear? Clear na for everyone. Thank you so much, Deepthi. And by the way, everyone who is watching this video, don't show conjusi while clicking on the like button and clicking on the subscribe button. Do that. Okay, let's go ahead with the next question out there. Question number 58. Select the correct path of flow of milk during the breastfeeding. During the breastfeeding, what is the flow of milk? Come on. A, B, C, and D. And those who remember my formulas, remember it very well. Because we are a student. Oh my God, thank you. Okay. Backbone pain, exactly. Too much of standing, that's why. Probably when I reach around 40 years, I'll be like 80 if this goes on. Okay, so C, Bhagyashree says C is the answer, Panvit says C is the answer, Kavita says D, Lakshmi says D, and okay, so C and D, C and D. Why there's a confusion between C and D? C and D is what? Let's check it out. So first, remember my formula. Dal fell on mat. Oh, you. Where are we? Just a second. I did something. Oh, you saw the answer, right? But anyways, dal fell on mat. It goes like this. Okay. M stands for mammary tubule. Mammary lobes. Okay, lobes has what? Lobes basically form alveoli. Alveoli basically opens into tubules. Tubules forms duct. Duct form ampulla. Ampulla form lactiferous duct. So that is the dal fell on mat. Okay, so by that particular logic, if at all we see mammary tubules are there, so obviously this cannot be the one, because either lobes should be there or alveoli should be there. Now alveoli, mammary tubules. Okay, mammary ampulla, mammary duct. Array, mammary duct should be coming first. No, how can mammary ampulla come first? So this is wrong. What is D? Alveoli, mammary tubules. Okay, mammary duct, then mammary ampulla, then finally lactiferous duct, which is why D is your answer for this particular question. Do not get confused between alveoli and ampulla over here. Alveoli comes first, ampulla and la for lactiferous. So you can remember like that. Yeah, so guys, the answer for this question is going to be D. Is it clear? Is it clear to everyone out there? If yes, give me a quick thumbs up. We'll move on to the next question. We are correct. Yes, you guys are absolutely correct. You guys are doing so great. Yes, cool. Okay, so okay, perfect. Let's go ahead. Question number 59. Guys, we have completed human reproduction chapter also, and we are getting into the next chapter, that is reproductive health. And Macha, we are doing super good. Okay, I'm super proud of you. Question number 59. Fill up the blanks in the following paragraph by selecting the correct options. Okay, so obviously you have to spend some time over here. Just imagine it's a KCET question and solve this question within 1 minute. Dash methods work on the principle of avoiding chances of ovum and the sperm meeting. I do not know this option, cannot tell the answer fast. So I will go with the next option and see the answer. Dash is one of such method in which the couples avoid or abstain from coitus from day 10 to day 17 of the menstrual cycle. This is known as periodic abstinence. During certain period, there is no sex. Okay, there is no sexual intercourse. This is known as what? This is periodic abstinence. So 2 is going to be periodic abstinence, and you have got 2 options like that. Cut off these 2 options. Cut off these 2 options. Okay. Dash is another method in which the male partner withdraws his penis from the vagina just before the ejaculation to avoid insemination. Again, interruption between the coitus. This is known as coitus interruptus. So basically you have got 2 options again. Okay. Now if at all you see, these 3 options are same. So that means we have to choose from here itself. So 1 can be either natural or surgical. Let me see again, go back to 1. Dash method work on the principle of avoiding chances of ovum and the sperm meeting. It can be surgical, because in surgical, basically with the help of surgical, the ovum and the sperm do not meet at all, right? The help of surgical method. So the answer for this question is going to be D. It is going to be D. And dash method is based on the fact that ovulation and thereby the cycle does not occur during the period of intense lactation, that is lactational amenorrhea. Cool, guys? So the answer for this question is going to be D. Who said C? Who is the one who is saying C? It is D, no baba. Macha, it's D, no. Why there's a confusion? Prakash, thank you so much, da. Okay. Okay. So, I have also marked as C. No, it is D. D is the answer. Okay. Just give me a second, okay? Methods work on the principle of avoiding chances of ovum and the sperm. It can be C also. but it can be D also. Let's just put this question on hold. Why? Because there's a confusion. Yes, natural methods avoid by the principle of chances of ovum and the sperm meeting. Okay, in lactation and amenorrhea, there is no ovulation itself. Okay, but in the case of surgical methods, you make sure the gamete transport is not taking place. Again, you are not allowing the ovum and the sperm to meet. Right, so which is why it could be A, it could be C, or sorry, it could be A or it could be D also. So actually, in the book, C is marked as the answer, which is why I think C is being put over here. Let's put this question on hold, okay? I'll be making sure I recheck it once more, put the logic once more. Anyway, it's not a PYQ, it's a new question, okay? I hope that works. Yes? Cool? It should be somewhere between C and D. Okay, anyways, let's see that. Question number 60. I will be coming back with that question, okay? Don't worry. Guys, tell me the answer for this question. A childless couple visited Assisted Reproductive Technologies Art Center to get assistance to have a child. Okay, on diagnosis, it was noticed that there was a low sperm count in the male partner. Which of the following strategy of art is more suitable in this particular case? Baba, remember. Okay, over here, what is the question? You have been asked where the sperm count is very, very low. What is the process that is going to be applied over here? It is applied over here. Kavita says it is B, AI. Okay, artificial insemination. Bhagyashree says A. Okay, Norman says B, Adya says B, no, and Prakash, hello dear, tell me. And Maria says B, Sharda, okay, Jeevan, Panvit, most of you guys, Asfia, Shivani, Vinay, Gates, B is what? Artificial insemination. Exactly. Artificial insemination means you either collect the sperm from the donor or from the partner itself and then put it inside the reproductive tract. If you're putting inside the vagina, you do not have any specific name for it in your NCERT. But if at all you're specifically putting inside the uterus, you call this as intrauterine insemination. Intrauterine insemination that comes under artificial insemination. It's not gamete intrafallopian transfer. It's not zygote intrafallopian transfer. It's not in vitro fertilization as well. That works by a different logic altogether. So B is your answer for this particular question. Is this thing clear to all? Can we get can I go ahead? Ma'am, and a favorite bio teacher. Oh my God. Thank you so much. And that was Malayalam. Okay, thank you, Reid Stormer. Okay, guys, can we move ahead? Yes. Prajwal, you scored 100 in biology. Okay, so super good. Just a second, 4 are done. Yes. Okay, cool. So next question. The government of India legalized MTP with some strict regulation in the year what? 1951, 1961, 1971, 2001. What do you think is the answer for this particular question? Any idea? B. Okay, Shankar says B. Vamsi says B. Okay, what about others? Noha Kazi is also here. Hello, dear. Okay, say C is the answer for this question. Panvit says C. Okay, what about others? Come on, guys. Come on. Manjunath says C. Sharda, C. Okay. Silent Power, Adya, A. Asfia, Kyun. Okay, and Maria. C is what? 1971. Dear, in the year 1971, MTP was legalized. Always remember that. And if at all you add +1, it becomes 20. And if you do ulta, it becomes 17. 2017 MTP Amendment Act was being put. Okay, so legalized in 1971, Amendment Act in 2017. These are the two important things. And always remember, always one more very important question. MTP is considered to be safe until which month? Is considered to be safe until which month? Everyone, come on. Ma'am, your number 01934356. Guys, tell me this. MTP, which is there, it's safe until which month? What amendment? MTP Amendment Act, according to which legalization was done over here, but people were doing it randomly. There were so many deaths being recorded, which is why an amendment act was put, according to which, yes, exactly, first trimester, according to which it was said that for the first 3 months it is considered to be safe. Okay, first 3 months means 12 weeks, or basically the first trimester, it is considered to be safe. You just have to take permission from one medical practitioner according to this particular act. But you can do MTP until 6th month, that is the second trimester, or basically 24 weeks. But in this particular case, you have to take permission from 2 medical practitioners. That comes under Medical Termination Act. So please remember, it can be done until 6th, but it is safe only until 3rd. Okay, this is very, very, very important. How many questions will be, how many marks will be there? I do not know. I cannot tell how many marks will be there, but I can surely tell this will definitely help you well. Yeah? Okay. Let's go. You want Kit Kat, ma'am? Yes, once we meet, you can give me Kit Kat. Question number 62. Can we go ahead with question number 62, guys? If yes, can you give me thumbs up? I'm so sorry, I moved forward. It's okay, right? Can you give me? Risk is there in the 6th month. Exactly. There is too much amount of risk in the 6th month. Shall we go ahead? Yes, perfect. 12 week. Okay, I think so. Okay. Sri Krishna says A, Panvit says B. Okay, Sinchana is a hello dear. Ganesh Rao, thank you, thank you. Nisha says B. Madiha says B. Panvit, Shivani, Sri Krishna, Anne Maria, Venkatesh, Gulala, cute name. Shubhan, Shivani, Ramesh, Asfia, I'm trying to read out. Saida, okay, Prajwal. You're saying B is the answer for this. What is the question? During IVF, IVF embryo transfer, after fertilization, if embryo is having 8 or less number of blastomeres, then it is transferred to A. Okay, so if at all, when you're fusing and you get the zygote, and basically the zygote divided and it has 8 cells now, it has formed an embryo having 8 cells or lesser than that. Where will you put it? You will put it in the fallopian tube. So A is going to be fallopian tube for sure, so you can cut off these options. Okay, now what is there? And if the number of blastomeres is more than 8, where do you put it? It is going to be inside the uterus. Why? Because if at all it is more than 8 and you're still putting in the fallopian tube, it can lead to ectopic pregnancy in other areas, and that can lead to death of the mother and the child. That is known as ectopic. Okay, it is known as ectopic pregnancy. So always remember, this is going to be your, B is going to be your answer for this particular question. Guys, I'll tell you one secret funda. Okay, I'll tell you one secret funda. Once this live has been done, spend some time on this particular PDF, use this PDF and revise the concepts that are being asked. I'm not asking you to just do the question, the concepts, whatever there's there in the art, go revise it. So from each topic I have taken one question. That means these topics are important. Got it? Obviously I cannot give multiple questions, or otherwise it won't be 100. It would be more than that, right? So yes. And by the way, are we ready for the next chapter? If yes, give me a thumbs up, everyone out there. And you guys are going really good. You guys are super awesome. I am super proud of you, of being a teacher. Yes, and do not forget me after your KCET examination. If at all you meet me somewhere in Bangalore, say hi. Yeah? Example for ectopic pregnancy, Baba, ectopic pregnancy means pregnancy that happens in any other area. It can be in fallopian tube. There are so many people who die because of that. There's no example. It's a situation that happens, right? It can happen to anyone. Okay, cool. Adya, lunch break. So guys, if at all we take a break now, it might take a long time. Yeah, so you guys, do you want break? Yes, Prajwal, tell me, what's the doubt? Okay, so those of you who want a break at this particular point, I do agree that you might be, you might have not had your lunch. Please go ahead and have your lunch, okay? And you can come back and watch this video. Will that work? Otherwise, we will not be able to complete this chapter. How much time will you need? Chandana, thank you so much, dear. Ma'am, after 75 you can give a break. Yes, after 75 can we take a break? That will be working. First year biology we are doing, dear, we are doing in between. Okay. Yes, I'll finish all the chapters. Okay, guys. Okay, so guys, we will take the break after 75th question. Okay, I hope that works. Okay. Yes, continue. And if you're so hungry, please go and have, please do not wait for me. Okay, go and have your food. That will be really good for you. Okay, biotechnology principle and procedure, 63rd question. Let's reach out to 75th very fast. Okay, select the option that correctly states the working action of restriction endonuclease EcoR1 on DNA sequence GAA-TTC. So this is a sequence that is given to you and you have to tell how or exactly EcoR1 will be working at which particular location. So A, B, C, D options are given to you. Make sure you go through this very carefully. Okay, make sure you go through this very carefully. Song, what are you asking me? That's the only thing that is left. Okay. A. Most of you guys are saying A is the answer. A is what? Are you sure A is the answer? Check out. Very important question. Come on. You cannot do mistake over here. Check. You know the answer. You're just doing a very small blunder. Most of you guys are saying A is the answer. Guys, we're doing something wrong. If at all you are, since as everyone is saying A, I have to come over here, tell what is the correct answer. You know the site for EcoR1 is GAA-TTC, right? Yes. Is from 5' to 3' direction? Yes. Now you see over here, the cut is made between GA and the other A, between A and A in the case of option A. Do you think that is correct? No. The cut is usually made between G and A. G and A, not between A and A. So the answer for this question is going to be B, no. 5' to 3', 5' to 3', GAA-TTC, cut between G and A. Got it? Which is why the answer for this question is going to be B. It is not A. If at all you gave the answer as A, that means that you are not reading the questions carefully. You have to be more attentive with reading questions. You know the answer, but you are being, what do you say, because of your speed, you're ruining it up. Don't do that. Okay, you will miss your marks. Cool. So is it clear? Is it clear to everyone? If this question is clear, yep, I said Q. Perfect. So if this question is perfectly clear to all of you, give me a quick thumbs up. Come on. We told right answer, ma'am. Kavita, kudos to you, dear. Okay, so initially I saw a lot of you gave me A as the answer. That is why I said. But now it is saying B. Good. Deepthi, I'm trying to cover as much as I can. Yes, PU2 I'll definitely cover. PU1 most of the chapters I'll be covering except 2 or 3. That's all. Okay. Perfect. Let's go ahead with the next question. Study the given figure carefully and select the incorrect statements regarding this. Okay, so given figure is this. I hope you can see it. If not, I'll just mark it up also. This is going to be A. This is going to be B. And these are the wells that are there. Over here you have 1, 2, 3, 4. Okay, you have 1. You have 1, 2, 3, 4. Now, it represents, you have to find the incorrect one. Okay, you have to find the incorrect one out of this. So, it represents a typical agarose gel electrophoresis in which lane 1 contains undigested DNA. So, over here, if at all you see, thus the lane 1 which is there, it contains the undigested DNA. Check it out. Do like this. Okay, do like this. Silent power, I'll tell you the answer. Wait, Damacha. Okay? Okay, guys, tell me. Undigested DNA? Yes. Since it is undigested, which is why you will not be able to see any bands out there. So I'll say statement 1 is correct. Lane 1, statement 1 is correct. Okay. Next one. Smallest DNA bands are formed at A and largest DNA bands are formed at B. Smallest at A and largest at B. Let's check it out then. Smallest at A and largest at B. Wrong, no. Large should be there in A and small should be there in B, because you have seen the DNA wells are over here, from here to there it goes. The smallest one will be in the B side. So this is going to be incorrect. Here ulta is given over here. So which is why I'll say this particular statement is going to be wrong. 2 is going to be wrong. 2, I have got 2 options only. I have got 2, so basically I can cut off these 2 options. Okay. Now, according to this, either 4 or 3. Let's see. Separated DNA fragments can be visualized after staining in the visible light. What do you think? For this, I do not have to show any picture. The separated DNA fragments can be visualized after staining in the visible light. Is that the answer? Most of you guys are saying C is the answer. Okay. Some people are saying D is also the answer. Let's check it out. We do have to check it out, right? B. Okay, let me check it out. Okay, so separated DNA fragments can be visualized after staining, yes, with ethidium bromide, but not in visible light. It is in UV light. So this visible light and UV light is different, which is why this particular statement is also going to be wrong. So 3 is wrong. What about the 4th one? The separated DNA strands are cut out of the agarose gel and extracted from the gel piece. This step is known as elution. Yes, this is known as elution, where basically the separated strands are being cut from here and being taken out. I wanted you guys to know all these terms, which is why I have put this question, where everything is included, so do not miss out on these concepts. So your only incorrect options are what? B. It is only B. This one and this one. Smallest are towards this side and largest should be that side, and also it is not viewed under visible light, under UV light only. Ma'am, what should we want to wear for the examination? I will be making a video on dress code, Macha, don't worry. Okay, I will be making a video for sure. Everything that is important for your KCET, I'll be making. Ma'am, instead of visible light, it will be UV light. Okay, it will be UV light. Cool. I hope this is clear. If it's clear, can you give me a thumbs up? Everyone who is watching this video, and if this question is clear, give me a thumbs up. Also, one more thing, they can be asking, who separates first, the larger one or smaller one? We do not know that. Who separates first? We do not know that. But who moves the faster or the farthest? We know that, and that is with a smaller band. Okay, so for all it says that smaller ones move first. That will be wrong. You cannot know that. That can be anything. Cool. And yes, thank you so much, Adya. Okay, perfect. Let's go ahead with the next question out there. Question number 65. From the following tools, techniques of genetic engineering, identify those which are required for cloning a bacterial gene in animal cell and choose the correct option. Bacterial gene in an animal cell. So you have to think, you have to cut open a bacteria, get inside, take the DNA, come back, get inside an animal cell and do all the process. So what are the things that you require over here? And thank you so much for giving all the thumbs up. Lots of things. Dear Sayada, I have to check, dear. Okay. Ramesh Reddy, biology. Amma, big thing. Thank you so much. Okay, guys. Nauman says A is the answer for this question. Okay, what about others? Check out. Listen, so first of all, you have bacterial gene. You are putting in the animal cell. So that means you have to go inside the bacteria. Bacteria has a cell wall made up of what? Peptidoglycan. So you need enzyme to break open the cell wall. What is that enzyme that you need? Lysozyme. Lysozyme is there in our tears also, which is why crying is also really, really good. So first thing that you would need is lysozyme. Lysozyme is there or not? Yes, it is there. So mark it up. Do you need lysozyme? Yes, ma'am, I need it. Okay. Next thing, Baba, which you need is what? Okay. So we have taken the DNA. Now the DNA is taken outside. The DNA has to be that means it has to be cut. For that you need restriction endonucleases. So this is also needed. Okay. After that, Baba, after you have cut the DNA, you put it in electrophoresis chamber, right? So you need gel electrophoresis also. Okay, done. After electrophoresis, what do you need? Anything else apart from this? Okay. Apart from this, basically you have taken the DNA. Okay, suppose you have taken the DNA, you have to transfer the DNA into animal cell. Listen, when you're saying you're putting inside the animal cell, 2 things should be coming into your mind. First thing, either you need a retrovirus, that is a vector, or probably you need a vectorless method. In vectorless method, what is there for you? Micro injection, not gene gun. Gene gun method is for inserting things in the plant because they have a cell wall, that is why you need a gun. So this is not for us. Similarly, for opening a plant cell wall, you need cellulase. Over here, there is no plant cell wall. Agrobacterium tumefaciens is the vector that you require for putting things again in the plant cell. If at all you need a vector, you require Agrobacterium tumefaciens. That is also not there for animal cells. So what is only thing that is left? It is micro-injection. With the help of a syringe, we are going to inject what? The nucleus directly inside, DNA directly inside the nucleus. Right? Finally, the DNA has to be attached to each other. For that, you need ligase. So what all do you need? 1, 2, 4, and 6, and 8. 1, 2, 4, 6, and 8. The only answer for this question is going to be A. Is it clear to all of you? And I see that most of you, Hina Kase, Kyun, all Kreji Games, Asfia, Saida, Tanu, Ramesh, and Chandana, Lakshmi, Rangappa, and Karthik, and everyone, Shankri, perfect, Chinchana, Sidhu. Okay, so if at all you have got the wrong answer also, I hope it is clear now. Yeah, if it's clear, guys, give me a quick thumbs up. Let's move on to the next question out there, and please drink water. Yes. Okay. Question number 66. Identify A, B, C, D in the given diagram of E. coli vector PBR 2022 and select the correct option. So this is a diagram that is given to you. Okay, I think you can clearly see it. Over here you have A starting from this point, you have B over here, you have C over here, and you have D over here. You have to identify what is what. You have to identify what is what. Thank you guys for the thumbs up. Okay, cool. Tell me. Okay, Norman says D is the answer, is it? Okay, Vinay says D and Shravani says zoom. Okay, zoom. I'll zoom. Just a second. Okay, this is A. Okay, so everyone says D is the answer. D. Preeti is also there. Hello, dear. So D is what? Let's check it out. So the most easiest one to spot out will be this one. We know in PBR 322, Baba, D is always going to be origin of replication, right? It is going to be origin of replication. Then you have ROP site, replication protein that is required, right? So wherever you see D is origin of replication, you have only two options like that. Either A or it is going to be D. So basically cut off the other options, which is not making any sense to you. Okay. Now what do you know already? That C which is there, if this is tetracycline resistant, C is going to be ampicillin resistant, right? Ampicillin resistant gene over here. So C is ampicillin, both of the places it's like that. Okay. The next option that we have is in tetracycline you have BAM-H1 and SAL1. So B is going to be BAM-H1, B is BAM-H1, B is BAM-H1, and the answer for this question is going to be this. It is going to be this. Okay, it is going to be D. Always remember it. So basically, if at all I see the plasmid, I have ampicillin, I have tetracycline, here I have origin of replication, here I have ROP site, and that's all. So one pista shake, one vada pav. Okay, with that we thought it's with that we went to eco class, eco R1, eco class, but we realized actually it's Hindi period, Hind 3. Okay, and then there we bammed into Salma Miss, Sal1, and then we gave Salma Miss to Vada Pav. Okay, so please remember, this is very, very important. Please do not forget it. ROP is a site which is basically responsible for production of a protein that is super important for replication to be starting off. Okay, so it's an important thing for replication to start off. Cool? Very, 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 very important. Cool, Macha. Let's go ahead then. Yes. Guys, give me a thumbs up. If everything is clear, we can go ahead then. Give me a thumbs up, everyone. Hiya. Heart is coming on. Thank you so much, guys. ROP maintains the copy number. Wrong. ORI maintains the copy number. Don't go and write ROP maintains the copy number. Who said that? It is ORI who maintains the copy number. Okay. Origin of replication maintains the copy number and everything. Okay. Ma'am, you're teaching. Thank you so much, Macha. Okay. Question number 67. Suppose DNA samples collected for DNA fingerprinting analysis are less than required quantity. Okay. It is very less. Which of the following techniques is helpful to make the sample sufficient for the above analysis? If at all the DNA sample is less and if you want more and very less sample is there, what do? DNA probing, electrophoresis, chromatography or PCR? This is a very straight question. The reason I have put is that I have seen questions like that. Comes in board exam. Yes. Yes. Tanu says D is the answer for this. Okay. Norman says D is the answer for this. Okay. Tanu and Vijay and Bhagyashri D. Silent power. Kavita and Maria. Norman, Shreya. Yes. Shravani, Sadhvi, Lakshmi, Pragnya and Shashik. And everyone, GS Suni is also there. Everyone says D is the answer. D is what? PCR. Polymerase chain reaction is something that is super important for amplifying the little amount of things that is less. For example, if at all you are not able to find the disease which is there and very less pathogen is there, you can amplify it and write it with the help of PCR. PCR. And it is also there for finding any mutant gene in the case of cancer. So always remember, for amplification of little things that is there and little amount, and also for finding mutant genes, because you know what, in the case of this we create probes and things like that which can basically replicate. We make the probe, right, the primer out there. So if at all it is mutant gene, it is not going to attach up there, and we are able to identify, oh, this is a mutant gene out there, right? So please remember that also, always. Okay, I hope we can go with question number 68, and we have reached microbes in human welfare. Okay, ready for this question? Come on, Macha, solve this. Question number 68. Match the microbes in column 1 with the commercial industrial products in column 2 and choose the correct option. So over here you have got the things, you have got the option, choose it. Do not do it fast. Please understand and do it. Okay, Pix video. Don't do, Macha, don't do. Okay, ma'am, doubt. Yes, Panvi, tell me. Fast, ma'am, too late. How do you think I can go faster than this? Hi, Shreya. Okay. B, C. Okay. Hitesh says C is the answer for this. Shraddha says B and 68 C. Lakshmi says C. Venkatesh says C. C is what? C is going to be. Okay, let's check it out. So, Baba. Trap the easy one. What is the most easy thing that you can see over here? Saccharomyces cerevisiae. Saccharomyces cerevisiae is going to go with ethanol. Right? Yes, fermentation. So C goes with 1. C goes with 1. I just have 2 options. I'm going to cut off these 2 options. Okay. Now we have Aspergillus niger, Clostridium butylicum, Trichoderma polysporum and Monascus purpuris. Again with the name Clostridium butylicum, butyric acid. B goes with 4. B goes with 4. Again the same options. Okay, fine. Aspergillus niger which is there, it's a fungi, and what does it form? It helps in the formation of citric acid. So A goes with 3. A goes with 3, and I've got my answer. It is going to be this. Aspergillus niger is a fungi that forms citric acid. Very, very important. Trichoderma polysporum, it is the one that forms cyclosporin, and Monascus purpuris is the one that forms statin. This and this, all of them fungus. Saccharomyces cerevisiae yeast also comes under fungus. Only Clostridium butylicum is a bacteria. Okay? Yes? Cool? If this question is clear to all of you, and I know that you have given me the answer as C, give me a thumbs up. We'll move on to the next question out there, Macha. We'll move on to the next question. Come on, let's do it. Yeah? Jeffrey Epstein, eh? Okay, perfect. Thank you. Let's go ahead with the next question out there, and it goes like this. Identify A, B, and C and D. Okay, so you have A, B, C, and D over here, and you have to choose from the options over here. So this is 20 mg per liter, 5 mg per liter. This is what? BOD, biochemical oxygen demand. This is 300 mg per liter, and this is 400 mg per liter. And over here you have got options. You have to choose from this. Always remember, what do you mean by, ma'am, I want your option. What is this known? Ma'am, this is known as goat teaching. I hope that goat is not for some other goats. Yeah, I hear this question. Come on, Do this question. This is very nice question. I like this question. C is the answer. Lavanya says C is the answer for this question. Okay, so fast. Okay, let me check. Vijay says B is the answer and Lavanya says Sri. Shraddha says D. Guys, check it out. Listen to me carefully. What do you mean by biochemical oxygen demand? Suppose this is an aerobic bacteria. Tan-ta-dan. Aerobic bacteria says that I need too much amount of oxygen to basically act on what? To basically act on the organic acids and organic molecules and break it down. I need too much amount of oxygen. That is known as BOD. So basically, if at all I say pollution is more, if pollution is more, the demand for oxygen is also going to be more. If pollution is more, demand for oxygen is going to be more because aerobic bacteria will be needing that much amount of oxygen to break it down, right? So wherever you find highest amount of pollution, that's where you find highest amount of BOD. And where you find least amount of pollution, that's where you will find least amount of BOD. Use that logic and try to find out your answer. So if at all you're able to see D is 400 mg, right? If you see D goes with the thing that has highest amount of problem, it will be the answer. So if at all you see primary effluent, secondary effluent. Secondary effluent will be having less BOD. Why? Because it is being treated. Primary effluent is still going to be treated. Untreated sewage pattern, that is even bad than primary effluent, right? Because it is not treated at all. Obviously here the BOD will be very much. River water, it can be clean also. Sometimes not too clean, but yes, still better, on the better side. So untreated sewage water is going to be D. So that is my answer. It is going to be C option. C will be my answer for this question with only one thing that I have said. But again, I have to reconfirm it. So 5 mg, B is 5 mg per liter. B is 5 mg from liter. Again, secondary effluent, it is undergoing treatment, right? Primary effluent, river water, untreated sewage water. What is the one that will have least BOD? Obviously, in comparison, it will be river water only. Okay, it will be river water. That is why the answer for this question is going to be CE. I hope this is clear. Everyone who gave me the answer as B and D and things like that, A, B, C, D, we have played. Baba, you have got it? Have you got the answer? Yeah, we are good to go. More bad, more polluted, more BOD, more polluted water. Exactly. Yes, children, if you're good to go, if you're liking these questions, do not forget to hit the like button. And if you're good to go, please put up the thumbs up. We'll move ahead with the next question. Got it. Cool. So next question that is there, question number 70. A farmer has applied chemical fertilizers in his crop field for many successive seasons, okay? In the next season, the crop growth was poor. As soil lost its fertility. Suggest the suitable microorganism that replenishes the fertility of the soil in his fields. This is 2019 question. Thank you for the thumbs up. Okay. Untreated water, sewage water will have obviously high BOD. It will be too much polluted, right? Yes. So what do you use? If at all, you fertilizers is something that you have used, okay? And basically that is making sure that your basically soil fertility is lost. So obviously what are you going to use? The microorganisms that you're going to use. B, everyone says B. Lavanya, Silent Power, Kuhn, Sinchana, and everyone I see. B is what? Nostoc, exactly. Spirulina, chlorella, as well as Spirogera has nothing to play over here. Nostoc is what? A nitrogen fixing bacteria. So nitrogen is a fire fertilizer, obviously. It's a very important thing that you need. So Nostoc would be the answer for this particular question. Nostoc is a cyanobacteria or a bacteria? Guys, Nostoc is a cyanobacteria or is it a bacteria? What do you think? Cyanobacteria or bacteria? B. Nostoc, cyanobacteria or a bacteria? Come on, I'm waiting for you guys. I think it's coming very slow. Nostoc and Annabena also fixes nitrogen, exactly. Nostoc, Annabena also fixes nitrogen. It's a cyanobacteria, exactly. So always remember, instead of it's ano. Okay, instead of uno you play, right? Annabena, Nostoc, Oscillatoria. These three of them, they come under bacteria, but under specific bacteria, that is cyanobacteria. They can prepare their own food as well. They are autotrophic in nature. Cool. So B is the answer for this question. Now, how can I show you? You have already seen that particular answer now. That's so sad. But still, which among the following products of microbes is not obtained from fungi? Which is not obtained from fungi? What do you think is the answer for this particular question? It's a cyanobacteria, okay? Children, I think it's a bit lagging behind because net is coming, going over here. So C, everyone give me the answer. C, Swiss cheese. Listen to me. Okay, you have been asked which one is not obtained from fungi. Now, even if you have seen the answer, obviously I will ask you another question, that where do you get Swiss cheese from? If not a fungi, then which is the bacteria or what is the organism that is going to produce Swiss cheese? Tell me the answer for that. Okay, by the way, penicillin is produced by fungi, Penicillium notatum, and this is the first antibiotic that you made. Statin you get from Monascus purpureus, again a fungus. Cyclosporin A, it is produced from Trichoderma polysporum, again a fungus. Swiss cheese is produced from what? You have to tell me. Swiss cheese, statin esta, statin is produced from Monascus purpureus. Okay? Someone is saying yeast. Swiss cheese is produced from what? Always remember, yeast is a fungi. Yeast comes under fungus, so that is not the answer for this. Swiss cheese is produced with the help of Propionibacterium shermani. It's shermani, okay? It's shy. Why? Because it is producing CO2. That is why you see big holes over here in the cheese. That is the reason you see big holes in the cheese. So what is this? Propionibacterium shermani. Propionibacterium shermani. This is very important thing. Do not get it wrong. Roquefort cheese, on the other hand, is produced by a fungi. Roquefort cheese is produced by Penicillium roqueforti, but anyway, you do not have to remember the name, but please remember it. Okay? Yes. Okay, guys, just give me a second. Just give me a second, okay? Akash, there is update, free up storage space is coming on the screen. The live might end if at all you do not delete it. Can you come here first?
Live won't end.
Are you sure? Then,
nothing will come.
Because previous time it got off.
No, live won't end.
Sure?
अरे स्टोरेज का कोई जरूरत नहीं है लाइव.
ठीक है भाई. Okay. Okay, so yes. Okay, guys, 72nd question. That is, enzyme which is useful to remove the oily stains in laundry is. Enzyme which is there to remove the oily stains in laundry is what? I'll keep the promise. 75th question, we'll take a break, okay? Enzyme which is useful to remove the oily stains in laundry is what? Amylase, renin, lipase or protease? What do you think is the answer for this particular question? C. Savita says C is the answer. Bhagyashree says C. Hitesh and Vijay said, and Lavanya, Spurti, everyone. MD Sulaiman, Biru, okay, JSNE. C is what? Lipase. Always remember, lipase for lipids, and oil is coming under lipid. They are fats, right? And for removing it, you need lipase. Protease, where do you need it? For example, you have juice, and in juice you have so many pieces. You want to clear that up. Okay, you want to clear that up. For that, you will need protease. That is something different. Okay, in bottled juices, you will be needing it. Amylase is for breaking down the carbs out there. That is not needed over here. So the only answer for this particular question is going to be 2. So it is going to be C. Okay, perfectly clear. Very important things. Do not forget it. Okay. Next question. Roquefort cheese is ripened by fungi, virus, yeast, bacterium. Roquefort cheese which is there, it is ripened by fungi or virus or yeast or bacterium. What do you think is the answer for this particular question? Clarify agent, protease and pectinase. Exactly, Shivani. For juices, right? Yes. Savita says D is the answer for this question. Is it? Okay. Jyoti says A is the answer. Okay. MD Sulaiman says A. And Bhavish says 73C is the answer. Okay. Different answer. Lavinia says A, and Silent Power says A. Praveen A. Shru A. Biru C. Okay. Rishika says A. Sujata says A. Shraddha, Lavanya, Komali, and Sasisa. Okay, most of you guys are saying the answer for this question is A. It is A. I told you right now. Roquefort cheese is a fungi. Swiss cheese, bacteria. Okay, Propionibacterium Shermani. S for Swiss cheese, S for Shermani. You can remember like that. There's a bacteria. This is a fungi. The name of the fungi is not given in your NCERT, so you need not worry about it, but please know about it. Guys, also one more very important thing. I just want to ask you one thing, that if at all, okay, I am asking you, what is the function of statin? Can you tell me the function of statin? What is the function of statin? Come on, fast. This I need fast, before I end this chapter. The function of statin. Uta aitla. Blood cholesterol lowering agent. Exactly. Please remember, it is what? Blood cholesterol lowering agent. Okay, it makes sure it stabilizes. So I remember statin for stabilizing. Statin for stabilizing the blood cholesterol lower, and basically this is produced by Monascus purpureus. It is a fungi. Next thing that is there, it is cyclosporin A. Cyclosporin A. What is it? Cyclosporin A, which is there. What is it? Everyone, Shamsuddin, every one of you who are giving me answers, super. Okay. Cyclosporin A, what does it do? Lower LDL. Perfect, Kishan, Hina Kausar, everyone, Shravani, Sujata, Spurti, guys. Next question that is there. Cyclosporin A, what does it do? It is a clot buster. Who said it is a clot buster? C for clot buster. No, don't remember like that. It is an immunosuppressive agent. Suppose I take a kidney from Shamsuddin, and basically what happens, my kidney is not working well, I take the kidney from him, my body will reject it. Why? Because it is foreign. That is why I need immunosuppressive agent during this particular case, so that I can sustain with a new kidney. Okay, it is produced by Trichoderma polysporum. Okay, Trichoderma polysporum, which Trichoderma polysporum, which is very important, right? This is also fungus. Now finally you have the last one, that is streptokinase. Streptokinase, what does it do? This is a clot buster. This is going to be a clot buster, and this is produced by a bacteria. The only one that is produced by a bacteria, that is Streptococcus. Now coccus term, when it comes, it means round bacteria, coccus. So streptokinase is produced by Streptococcus, and it is the clot buster in the case of patients who are having myocardial infarction, or basically heart attack. Okay, so do not get it wrong. These three things are very important. You can get questions based on this also. Cool? Yes. You will thank me later when you see some questions from here. Okay, 74th question. Come on, guys. I think it's coming very slow. In between, our light went off, probably because of which. Yes. Yeah. Let's solve this question. Consider the following setup at Miller's experiment and select the correct options regarding it. So over here you have got A. This is A. Okay, this is going to be B. This is going to be D, and this is going to be C. You have to choose A, B, C, D. What is happening over here? Is A boiling water and NaCl? Is B CH4, NH3, H2O and N2? And is C water containing organic compounds? And is D 900 degrees Celsius? What do you think is the answer for this particular question? Most of you are saying it is B. Is it B? B. B is what? B is going to be this: CH4, NH3, H2O and N2. That is correct. Wrong, no macha. Listen over here. See correctly. A is boiling water, NaCl. NaCl has no play over here. A is definitely boiling water. It produces water vapor. That is correct. But NaCl will be wrong, so this option is wrong. Now B is CH4, methane, ammonia, H2O, and not N2. It should be H2. It should be H2. So this is also wrong. Okay, so that is also wrong. What about C? C is this water containing the organic amino acid. Sorry, organic compounds are there. Yes. So basically, when Stanley Miller did his experiment, he was able to see water that is containing the amino acids. That was the first organic acid that he found, the organic compound that he found. So this is going to be correct. What about D? 900 degrees Celsius is wrong because it happens in 800 degrees Celsius. It happens in 800 degrees Celsius, which is why the answer for this question is going to be C. It is going to be C. Cool? So everyone who gave me the wrong answer, I hope it is sorted now. And everyone who gave me the correct answer, kudos to you. Yeah? And now the last question before the break, it does this. If the frequency of dominant allele is 60%, find out the percentage of heterozygous individuals in the population. Very important question. Come on, do this and go. If the frequency of dominant allele which is there is 60%, find out the percentage of heterozygous individual in the population. Come on guys, you can do it. This is an easy question and very important question also. Shrayan says it is C. Norman says it is A. Okay. What about others? Bhagyashree, Parnavit, most of you guys are saying it is A. Listen to me very carefully. Okay. Dominant allele, we are saying it is represented by P always, and the recessive allele is represented by Q always. Here we are saying the frequency of it is 60%. When it is giving in percentage, you can count it in other way also, divide by 10 and you will get 0.6. That will be the frequency of allele. This is in percentage. If at all you want to know the frequency of allele, just divide it by 10, it becomes 0.6. Okay. Now you have to find what is Q first, because you need to know have an understanding of heterozygous individuals, as in the one which has allelic combination like this, not like this, not even like this, a combination like this. For that we need to know about the dominant allele, that is 0.6, about the recessive allele and a little bit more. So basically what do? We put the equation P plus Q is equals to 1. What is P over here? P is going to be 0.6 plus Q is equals to 1. What is Q going to be? 1 minus 0.6. Q is going to be 0.4. It is going to be what? 0.4. Now we have got it. Now you are going to replace it where? In the formula P plus Q whole square, where P square plus 2PQ plus Q square is equals to 1 will be coming. So anyways, this is the formula. P square means you're talking about AA. Q square means you're talking about small aa. But here you have to find the percent of heterozygous. That means this. So you replace 2 into P is what? P is 0.6 into Q is what? 0.4. What will be your answer? 0.6 into 0.4 is equals to 0.24. Into 2 is equals to 0.48. So always multiply it with by 2 if at all you want to find the heterozygous number. If you're not multiplying by 2, you are getting your answers wrong. Now you have got 0.48. If at all you transfer it into percentage, convert into percentage, it becomes 48%. Usually whenever the term 60 or 0.6 and things like that come and you're talking about heterozygotes, it will be 48%, 4.8 and stuff like that. Okay, so the answer for this particular question is going to be A. If everyone got it, give me a thumbs up. If everyone got it, give me a thumbs up. Clear? Now for you. Yes, I can explain again, and those of you who want to take a break, you can start off your break, okay? Until when do you, 10 minutes break works for you? Does 10-minute break work for you? If yes, come back by 2:55, okay? Come back by 2:55, and for those of you who do not understand this question, stay back over here. Okay, we'll take the new questions from 2:55 only. Okay, so listen to me very carefully. Over here, children, please go for the break, okay? Those of you are going. Yeah. Dominant allele percentage is given 60% and dominant allele is represented by P over here. So we assume that A is dominant and small a is recessive. This is dominant and this is recessive. We say 60%. So basically if at all you want to calculate the frequency, you have to convert them into points. Okay, decimal points. It becomes 0.6. If at all, you just calculate like this, 10. Not into 10, basically into 10 by 100, whatever. Whenever you convert into percentage, you get 0.6. Okay. Now, over here, you have to find the value of Q. How do you find it? There's a formula, P plus Q is equals to 1. P is what? 0.6 plus Q is equals to 1. Q will be what? You shift this particular 0.6 there, 0.6 goes there, Q is equals to 1 minus 0.6, which is going to be 0.4. This is the frequency of what? Of the allele Q. Right? Now you have been asked about what? You need to find the percentage of heterozygous individuals. How do you do that? You have another formula, P plus Q whole square is the formula. So P square is like A plus B whole square, right? So P square plus 2PQ plus Q square. Over here, when you talk about heterozygotes, we basically just generally understand it's capital A and small a, right? So you multiply 0.6 into 0.4, but that would not be enough. You have to multiply it with 2 also, because in the formula it's there. So you have to multiply it with that 2. So 0.6 into 0.4 is equal to 0.24. And you multiply it by 2, it becomes what? 0.48. And if you talk about it in percentage, it becomes what? 48%. Similarly, if at all you have been asked about what? The homozygous allele, so it will be what? 0.6 whole square. That will be AA, because 0.6 into 0.6 will be 0.36. That means 36%. Similarly, homozygous recessive, how do you find it out? You have got the recessive allele, that is 0.4. You do a whole square, it becomes 0.16. That means what? 16%. This is how things are going to go. Cool? Works? I hope now it is understood. Yes. So guys, time for a Kit Kat break and let's meet. Nitya, it's not understood, dear. Someone was there who didn't understand. Okay. For homozygous condition, how can we find? So same thing as I said, Baba. For homozygous condition, if at all you're talking about capital A, dominant homozygous, so you just replace 0.6 into 0.6, P square, right? So 0.6 into 0.6, that is this one. If for recessive, you calculate the recessive allele. 0.4 is given for recessive, 0.4 into 0.4, it becomes 0.16. Okay? Cool it, Vittal. Madiha, top 100 question in the PYQ series. Yes, dear, it will be really good for you. Okay? If you can, just watch this video carefully and also, yes. Thank you, Nitya. Okay, tell how to eliminate the wrong answer. In this way, there is no way to eliminate usually. So it would not come in 47 and stuff like that. Never it will be coming in that particular range if it is 6 being mentioned. So there is ma'am kaichila. Okay. So there is no elimination that can be applied over here, dear. But yes. Okay, guys. So I think I also deserve a break. I'll go take a break and come back and then let's do the other questions. Kit Kat break. Meet at works. You're most welcome, Madiha. Okay, ma'am is MS Biotechnology Ramaiah University. Dear, I do have to check it out. Without checking, I cannot tell you, Kavita. I'm so sorry. Malayali, Anu. Whom are you asking? Yes, I am Malayali. Okay, so guys, I'll take a break and I'll come back. 2:55. By then, I think those of you who went for lunch break will also be coming back, right? Yeah. Meet you really soon. Baba, where are you? Which Baba? Is it me, Baba? Hello, hello. Am I audible, by the way? Yes. Guys, we are back. And don't spam, please. Okay. So, let's just wait for 1 minute so that everyone else can also join. Ayyo, where is it? Let them join, let them join. Let's complete it together. What's up? Had your food? Had your lunch? Had some tasty, yummy, yummy food? Or were just like eating something else? Oh my God, what am I? Oh no, it's there. Hello, 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 hello. Ma'am, looking beautiful, is it? Thank you so much. Good afternoon, and yes, creative talents, it will be helpful. Okay. I think children, we can go ahead, and children who want to, ma'am, are you there? Please come. I'm there. What happened? Am I not visible? Okay, let's do it now. KitKat break is over. 76th question. Come on. Match column 1 with column 2 and select the correct options from the codes given below. So you have got the options over here, okay, and you have to choose the correct one. Genetic equilibrium, gene flow, saltation, and genetic drift is there, and basically you have to choose from these options which are being given to you. These are the things that affects Hardy-Weinberg's principle. Most of them are the factors that affects the Hardy-Weinberg principle. Creative, thank you, ma'am. Because of you, I scored 100 in bio. Is it? You scored 100. Congratulations to you. Okay. Come on, children. MD Suleiman says the answer is B. Ma'am, your wife is so beautiful. Oh my God. Okay. Jyoti says it is D. Okay. Why there's an option between D and B? Okay. Vijay says it is D. Okay. Lavanya says D. Most of you guys are saying D. Norman also says now it is D. D is what? D is what? Let me check. D is this. A goes with 4. So guys, genetic equilibrium means all the gene pool that is there, it is going to remain constant. For example, I am saying that this place had these many tall alleles out there and these many small t alleles out there. It remains the same. I am saying that. This is the frequency. So gene pool remains constant forever. Is that true? Obviously, that cannot be true. But basically, genetic equilibrium says that. Okay. So A goes with 4. A goes with 4. That is definitely the option. So you can cancel these two options. Now, D is what? Genetic drift. Genetic drift means what? Genetic drift means change in the allelic frequency only by chance. So I have given you the example out there before also. Suppose there are green and black beetles. Okay. Suppose there are green beetles over here and there are some white beetles over here. Only few white beetles. The green one says that, hey Macha, you stay away. This is our area. We are going to deal with this particular area. You stay outside. So what happens? Basically, one day, one big elephant comes and stamps on it and all the green ones die. So what happens? The allelic frequency, do you think it has changed? Yes. Now the white ones would be more in number. And is it occurring by chance? Yes, Baba, it is occurring by chance also. So can I say genetic drift goes with change in population frequency due to chance alone? Yes. D goes with 1, D goes with 1. All the options are same only. Okay. The other option which is there is gene flow. Gene flow means what? Some organisms from here. For example, from America, someone is coming to India. From India, people are going there. Emigration, that leads to the flow. The allelic frequency is going to be disturbed, right? So obviously, gene flow goes with 3. B goes with 3, B goes with 3, and I have got my answer right over here. Saltation is what? Single step large mutation. Single step large mutation which is there, that is known as saltation, which is why the answer for this question is going to be this. Cool. Clear? Everyone? Is the answer clear? The answer is D for this particular question. I hope this is clear to all. If it is clear to all, give me a thumbs up, and everyone who have went for break, ask them to come back. Hello, Marti. Hello, hello. Let's assume that the exam is going to be good for all of you. Guys, if it's clear, just give me a thumbs up. We'll move on to the next question out there. Yeah? Yes, perfect. Thank you so much. Okay, question number 77. The image below is a diagrammatic representation of the operation of natural selection on different traits marked as A, B and C. Which of the following options correctly identifies the correct labeling for A, B and C? You have to give me the answer from here. Okay, so A, B and C is given. The picture is also given to you, and if at all you want to have a look at it, A says that the peak is getting high and narrower, it's like this. Okay, B says that it is moving in one direction, peak is shifting to one direction. C says that there are two peaks being formed. What do you think is the answer for this particular question? What do you think? Ma'am, take the bio, NEET bio also. So for NEET bio there are different teachers and they're doing it pretty well, right? Yes. 77A, Lavanya says it is A. Okay, she changed the answer. Okay, so Sheil says it is B, Kavita says it is B, Prajwal says it is D. Okay, what is happening over here? Guys, what is the answer? Prajwal, creative talent says B, most of you. Okay, Komali, Rushda and Shivani, B, Mart. Okay, so B, Hitesh is also there. Okay, guys, is the answer B for this particular question? Let's check it out. Right? B is what? B is this. Ayyo, everything changed. So always remember, suppose I am saying that there is tall character and there is dwarf character, but the middle character is being selected. That means it is going to show stabilizing. It comes in the middle. The peak is going to be there in the middle and it is going to narrow down. It is going to go up like this. Now suppose I am saying it is tall, it is dwarf, and dwarf ones are the ones which are being selected over here. So I say that peak is moving towards one direction, and that is being mentioned over here. That is known as directional selection. So first one is going to be stabilizing, other one is going to be directional. What about this? Where two peaks are there, that means both the extreme characters are being selected over here. Both of them would be found in a population. That is going to be disruptive. It is going to disrupt and you get two peaks out there, which is why the answer for this question is going to be B. It is going to be B. So everyone who gave me the answer as B, Aftab, I have taken your name. Vijay, option B is correct, exactly. So everyone who have given me the answer as B. Okay, perfect. Cool. Shall we go ahead? If yes, can you give me a thumbs up? If yes, can you give me a thumbs up, everyone? Oh my God. Sai, hello. Okay, perfect. Let's go ahead then with the next question. Question number 78. The brain capacity of Homo habilis is? The brain capacity of Homo habilis is 1800 cc, between 650 to 800 cc, 900 cc, 1400 cc. What do you think is the answer for this particular question? Come on. Chumps, come back to form. Come on. Brain capacity of Homo habilis is what? Okay, MD Sulaiman says it is B, Kavita says B, and Prajwal, Lavanya, and Shankar, silent, Bhagyashri, and Shambhu, what happened here? Tell me. Ask me the question. Okay, Sheikh and Chandana, most of you guys are saying B is the answer. What is B? B is between 650 to 800 cc. Is that the answer? Yes. Please know about all of it. Okay, first remember you had two guys, Dryopithecus and Ramapithecus. Dryopithecus was more ape-like. What about Ramapithecus? Ram is human being. So more human-like or more man-like. From Ramapithecus you got Australopithecus. Australopithecus, please remember it. And about them, it's only written that they were found in East African grasslands, and also they had made stone weapons, not to kill animals but to hunt fruits and stuff like that and eat it. Okay, they did not kill animals by any chance. Now from Australopithecus, Baba, you got the first human-like features, organisms, that is Homo habilis. Habibi, the first Habibi you got over here, and from this point onwards they had started mentioning about the cranial capacity. The first cranial capacity that was mentioned was 650 to 800 cc, that is Homo habilis. From Homo habilis, you got the first person to stand erect, and that is known as Homo erectus. Homo erectus. Okay, and their cranial capacity increased to what? 900 cc out there, 900 cc. And they were the ones who started eating meat and stuff like that. Until now, they were all vegetarians, but from then, it was non-vegetarians. And from Homo erectus, Baba, you got two things. One is Neanderthal man, and the second one was Homo sapiens, that is us. First, they evolved, then us. Here, the cranial capacity was the highest. It is 1400 cc. And basically, they were burying their dead, and they also covered their body. Okay, and Homo sapiens, they are the modern human beings out there. Who is known as Java man? Java man is basically Homo erectus. Okay, so please remember these few things are there. This is very, very, very important. Okay. Yes. Dr. Ahen is cardiosurgeon. Dr. Ahen, okay, is cardiosurgeon. Good. Ma'am should be English teacher, is it? Okay. Kavita, good, 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 good. So I hope that is clear. Do I need to explain that once more? No, right? Okay. Neanderthal man, Neanderthal man. Okay, I'll just say it. Listen to me carefully. Dryopithecus, Ramapithecus. Dryopithecus were ape-like, Ramapithecus more man-like. From Ramapithecus, you got Australopithecus. They were found in East African grasslands. They started having a stone and all. With that, they used to take fruits and basically eat the food. Okay. From them, you got the first habibi, that is Homo habilis. 650 to 800 cc is there. They also eat vegetarian food only. From them, okay, from them you got Homo erectus, the first guys to stand erect, and basically they are also known as Java men because their skull and everything were found in Java, Indonesia as well. Their cranial capacity increased to 900 cc and they started eating non-veg or basically meat and stuff like that. And from Homo erectus you got two things. Neanderthal man first, 1400 cc, they started burying their dead and they started covering their body also. And also from Homo erectus you got one more thing, and that is us, Homo sapiens. Okay. Yes. So that is the answer. There is nothing like 1800 cc. Also, please know about the years. This is 15 million years ago, Ramapithecus and all. Australopithecus 2 million years ago. Homo habilis, no year is mentioned. Over here 1.5 million years ago. Neanderthal 1 lakh to 40,000 years ago. So please remember their year gap as well. That is also very, very important. Now coming to the next chapter, and that is biotechnology and its application. Guys, is it okay? Are you understanding this? I hope it's clear and you've given me a thumbs up as well. So I assume I can go to the next question. Okay, it goes like this. Read the following statement and select the correct ones. BT cotton is a genetically modified crop. Pomato is somatic hybrid. Golden rice is transgenic variety of Oryza sativa, which is rich in vitamin A. What do you think is the answer for this? You have to find which ones are the correct ones, not the wrong ones, the correct ones. Come on. Shambhu, ma'am, tell me, dear Shambhu. Okay, Lavanya says 79D is the answer. Okay, Lakshmi says B, Prajwal says D, Silent Power says D, Bhagyashri says D. Okay, Bhavana, Shivani, most of you guys are saying D is the answer. Let me check whether D is the answer. BT cotton is genetically modified crop. Yes, what do? You put BT gene, cry gene inside the plant, and that is how you genetically modify it. That is correct. Pomato is a somatic hybrid. Yes, it is a result of somatic hybridization, where what do? You take a plant cell, you take another plant cell, this is of potato, this is of tomato, you remove their cell wall. If the cell wall is removed, Baba, what do you get? The thing that is left behind, the thing that is left behind is known as what? The protoplast. So then you fuse the protoplast and you get the somatic hybrids. Pomato is one of them, correct. Golden rice is transgenic variety of Oryza sativa. Oryza sativa is what? Rice only, scientific name of that is Oryza sativa. Golden rice is a transgenic variety? Yes. Why? Because we enrich it with vitamin A. And since we are putting vitamin A, that is why it is golden in color. Okay, and yeah, so basically I'll say that all these options are correct. 1, 2, 3 is correct, which is why my dear Chutku champion said D is the answer, and Chambu, Kishu, Ambariya, Hitesh, Shweta, Shivani, Yankanna, Vijay, everyone, Sashi, and everyone who gave me the correct answer. Before also I spelled some name out there. GS Suni is also there, I suppose. Yes, perfect. Cool, na? Shall I go with the next question? If yes, give me a thumbs up. If yes, give me a thumbs up. And by the way, if at all I am missing out any names, please forgive me. Okay, you know my condition. There are so many comments coming up, right? It will be difficult to read out all the names, yeah? So shall I go ahead? By the way, everyone have hit on the subscribe button, hit on the like button. If not, do that right now. Okay, perfect. Next question, question number 80. Eli Lilly company produced genetically engineered human insulin. Find the correct statement of the following about the process product, okay, about the product which is there, which is being produced. You're forgetting take my Aftab. I'm not forgetting there because the names are scrolling up, that's why I'm not able to check. Okay, so genetically engineered, you have to find what is correct statement. Okay, genetically engineered human insulin was prepared in 1993. Is that so? I do not know the year. Let's check the other options. Two chains A and B were produced by E. coli already joined by disulfide bonds. Two chains A and B were produced by E. coli already joined by disulfide? No, it is not already joined. We joined it later on, right? So this is wrong. Two DNA sequence corresponding to chain A and chain B were integrated with main genome. Peptide chains extracted, joined by disulfide bonds, and human insulin was being produced. So what are they saying? That basically A and B, which was being introduced inside the main genome, and peptide chains were being produced, and these peptide chains were joined using disulfide bonds. Okay, this is the next option that is given to you. Now, the last option, corresponding to chain A and chain B, two DNAs were integrated into E. coli plasmid, A and B chains were extracted and joined together. So there is no mention about disulfide bonds. What do you think is the answer for this question? And I really want to see who will give me the answer for this. The correct statement. The correct statement, dun 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 dun. Jeevan says 1987. Okay, that is the year. Shraddha says A is the answer. Okay, you have to find what is correct. Bhavish says C is the answer. I want to spot the C ones first. Kavita says C, Bhavna, C, Vinod, C, okay, C, Lavanya, C, Bhagyashree, C. C is the answer, no? C is the answer. Or dig it up. Is C the answer? Check the answer. Guys, you know what? I'll tell you a secret. If at all you promise me, you'll tell no one. Okay? The secret is that I also went with C first. But then I realized that's a problem. That's the wrong question. Why? Because over here, two DNA sequences corresponding to chain A and B were integrated with the main genome. Why are you messing with the main genome? You put it in the plasmid only, no? How can you put it in the main genome of the E. coli? E. coli has a separate genome. You just put things in the plasmid, not in the main genome, which is why the statement is going to be wrong. That means, what is the answer? This is going to be the answer. Corresponding to chain A and B, two DNAs were integrated in E. coli plasmid. Then A and B chains were extracted and they were joined. Now they are not mentioning anything wrong. They were joined. They have just not mentioned disulfide bonds, but it's still okay, which is why the answer for this question is going to be D. It's going to be D. Is it clear, everyone? Yes? Okay. So everyone who gave me the wrong answer, I hope it is clear, and everyone who gave me the correct answer, kudos to you. If it was a guesswork also, still kudos to you. Your guesses are working nice. Okay, so if it's clear, give me a thumbs up. Let's go ahead with the next question. By the way, we have reached 80th question. Oh my God, updates of life. Don't do. Okay, let's go with the next question out there. Ayyo. Question number 81. Match column 1 with column 2 and select the correct option. So over here you have column 1, you have column 2, and you have to choose the correct options. Lepidopterans, coleopterans, dipterans, and over here beetles, flies, tobacco budworm, mosquitoes, armyworm is given over here. So basically you have to choose. Okay, you have to choose from the following which class, who belongs to what all things. Come on, come on, come on. Still 23. Yes, we'll do it fast. Come on. We got Adigittum from Jasmine Ma'am before RCB match. Yes. Yes. Okay. B. Okay. B. Everyone is saying B. What is BB? Let me check it out. B is this. A goes with 3 and 5. Yes, lepidopterans, in that comes tobacco budworm and armyworm. Worm comes under this. Okay. Coleopterans, in that comes beetles. Dipterans are the easiest one. Flies and mosquitoes come there. Flies and mosquitoes are cousins. They come under dipterans. Lepidopterans, you have worm, and basically you have coleopterans for beetles. So which is why the answer for this question is going to be B. And you guys are absolutely correct. Okay, so there is no much explanation needed. This is something that you might have to do rectification for, right? I do not have an option to correct you over here. Will that work? Yes, B is the answer. Okay. Guys, can I go ahead with the next question? Can I go ahead with the next question? Good afternoon, Noor. I'm doing good. Thank you for asking. I hope you're also doing good. Yes? Yes. Question number 82. Come on. Guys, select the correct match regarding them. A, B, C, D. Okay, so you have alpha-1 antitrypsin, ROSI, mice, and GAAC. Okay, so you have to choose the correct match regarding them. Come on. So alpha-1 antitrypsin is there for treatment of phenylketonuria. Yes, no? It is there for treatment of phenylketonuria. Is that the answer for this particular question? "15 minutes, 20 question challenge." Come on, come on, come on. Do this. Bhagyashree says C, Shraddha says B, Bhavish says B, Ganesh says A, Bhavna says C, and Sai ratification, Lavanya says B. Okay, most of you guys are saying B, 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 B. Okay, let me check it out. Okay, alpha-1 antitrypsin is there for treatment of emphysema. It's a respiratory disorder, not phenylketonuria. So this is going to be wrong. Please remember that. That's why I put this question on all. ROSI, which is there, ROSI is not your neighbor. ROSI is a cow. Okay, ROSI is a cow who's producing what? Protein enriched milk. What is the name of this protein? Alpha lactalbumin. It is known as alpha lactalbumin. So this is correct. No, it is written protein enriched milk. So B is going to be correct. Mice, testing of polio vaccine safety. Mice is there for testing of polio vaccine safety. What do you think so? What do you think so? It is also correct. Mice, Papa mice, it is used for all this testing. GAAC, Gene Engineering Approval Committee. What is Gene Engineering Approval Committee? Genetic Engineering Approval Committee should be there. No. So B and C is the answer, which is why your answer for this question is going to be B and C only. The answer is B. All of you who gave B as the answer, put a tap on your back. You guys are absolutely killing it. Okay. Works? Yes? Shall we move ahead with the next question? I think whatever I'm saying comes a bit later. That is why I am just waiting over here for you to give me the answer. My neighbor is Daisy, not Rosie. Yes, okay. So all the Daisies and Rosies are out there. Genetic Engineering Approval Committee. Exactly, creative talents. Okay, PK also it is mentioned in NCERT too. No, Baba, alpha-1 antitrypsin. No, alpha-1 antitrypsin is used for treatment of what? Emphysema, respiratory disorder, not for treatment of this. No. Okay, yes. Macha, let's go ahead with the next question. Question number 83. Now here I want everyone to pay attention. Okay. Read the given statements and select the correct option. Statement number 1. Nematode-specific genes were introduced into the host plant using Agrobacterium tumefaciens vector. Okay. Statement number 2. Host plant generated double-stranded DNA triggers protection against nematode infestation. So everyone who knows what do you call RNA interference will be able to answer this. And by the way, RNA interference gave the most amount of questions throughout these many years. So this is a very important question. Do not mess it up. Okay, do not mess it up. Come on, do it. You can do it. Ma'am, once I've completed the bio chapter during writing mock test, I'll start to forget. It's okay. Revise the concept once and go attempt the mock test then. Okay, it will be coming back to you. Sometimes you feel that. Yeah? Okay, Bhavna says the answer for this question is C. Prajwal says it is B. Silent Power says A. What about others? Praveen says B, 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 B. This is very important. Ma'am, please say. Okay, let me check. So over here, Dikshit is also there. Hello. So guys, the answer for this particular question is going to be nematode-specific genes were introduced into the host plants using Agrobacterium vector. Yes. So I hope you know that there is a nematode that is known as Meloidigoin incognitia. This is going to affect what? Your tobacco plant, the roots of the tobacco plant, right? How does it do that? Basically, it gets into the cells of the tobacco plant and produces its mRNA. When you have mRNA, basically it gives the information to form the proteins, and these proteins are something that is toxic for the plants. So what is our method? This mRNA, which is single, we'll try to make it mingle. Okay, we'll try to make it mingle by giving a complementary strand to it. If at all we give a complementary strand, what is going to happen? Translation will not happen. Why? Because only single-stranded mRNA can undergo translation, not double-stranded. So that is the logic. So what do? We are going to take the gene over here. Okay, we are going to take the gene of Meloidigoin incognitia. Listen to me carefully, otherwise you will not be able to understand. We are going to take the gene of Meloidigoin incognitia. Okay, suppose this is a gene. And I want to put it in the plant. Okay. But how can I do that? If at all I want to insert anything into the plant, I need a vector. Now it can be vectorless method, that's such as you have gene gun and stuff like that. Otherwise, if at all you're needing a vector, it has to be what? Agrobacterium tumefaciens. Agrobacterium tumefaciens, you know, it has TI plasmid, and the tumor inducing plasmid, if at all we remove the transfer DNA and we put a gene of interest, our thing is being done. So we are doing that with the help of Agrobacterium tumefaciens. We are introducing this particular gene to the plant. Now the plant will be producing what? Double-stranded RNA. Double-stranded RNA is going to be produced by the plant, right? Now what happens? You have the single matcha over here, that is your mRNA. This double-stranded RNA which is there, it comes and one strand of it, okay, one strand of it remains as such. The next strand which is there comes and joins with the mRNA. It becomes mingled. Do you think it will be able to undergo translation now? No, Baba, it does not undergo the translation now. And basically, if there is no translation, will it make the protein? No. So that is the logic. So nematode specific genes were introduced into the host plant. Yes. Using Agrobacterium vector. Yes, you need a vector and you did that. Okay. Host plant generated double-stranded DNA? No, it produces double-stranded RNA, not the DNA, which triggers protection against nematode infestation. So it is double-stranded RNA, not the DNA, which is why this statement is going to be wrong. Statement 1 is going to be correct. So statement 1 is correct, but statement 2 is incorrect, which is why the answer for this question is going to be C, matcha. So everyone who gave me some other answer, which most of you give. Okay, I hope this is clear now. I hope this is clear now. Is it clear? Is it clear to all of you? Yes, Swati, dsRNA so C. Yes, exactly. So everyone, dsRNA so B. What is happening? Okay, I am correct. C, perfect. Cool. Okay. Let's go with question number 84. This is from the chapter Organisms and Population. By the way, one more very important thing from this chapter, cry genes. Okay, cry 1AB is there and cry 1AC is there. Oh, yes, and 2AB is there. What is this for? This is for cotton bollworms. Always remember, cotton bollworms. How do I remember it? First letter starts from C. First word starts from C. C, okay. Second word starts from B. Second word starts from B. Cotton bollworm. Ma'am, corn borer is also starting from C and B only. How do you remember that then? So basically for that you have cry 1AB. So only one word is there. Only one particular gene is there for corn borer since there's a smaller name. Okay, you can remember it like that. First letter is C and last letter is B. So cry 1AB for this. Please remember this. This is also very, very important. Cool? Ma'am, do you know Malayalam? Yes, dear Flabby, I know Malayalam. I'm a Malayali. Yes, works. Works for everyone. If yes, give me a thumbs up. Children, if you have understood, if it's all clear, give me a thumbs up. By the way, for everyone who is worried about IPL match, you tell me, okay, you tell me one very important thing. A teacher who strictly felt that she has to make PPT for children who probably could be watching her live tomorrow and are dependent on that particular live for some reason. If at all I can put efforts into doing that, can't you just put some efforts into making sure that your, what do you call, the game and stuff like that can be shifted a bit aside? Yes, for some time, at least during exam season. We are trying a lot and you cannot even imagine. I have been standing from 10 o'clock in the morning, okay? And with just 10 minutes of break, that is 2 breaks out there, I have not had my lunch as well. Please understand the importance of this particular live and take it seriously. I have never told it before, but do not do it, okay? If at all, I definitely understand your craze for match, and please go and watch the match when you have enough time. As of now, there are a few questions. Let's do it. Yes, let's do it. This is very, very important. I feel very sad when you do this just during examination and then say that, oh my God, I have not studied anything. That would not work. You have to be accountable for your actions. Yeah? Okay, let's go ahead. 84th question. Many parasites do not enter in the host body and live as ectoparasites. Find the incorrect match for ectoparasites and related hosts. So over here, you have to find which one is not matching at all. Okay, which one is not matching at all? Come on. Lice is there. Lice is an ectoparasite. Ectoparasite means the one that stays outside our body. Is the lice one that is there on the humans? Obviously, yes. Okay, lice is ectoparasite for humans. This is correct. Copepods. This is something that we forget. Okay, copepods are there as an ectoparasite for marine fish. Is that correct? Yes, ma'am. That is also correct. Okay. What about female mosquitoes? Female mosquitoes, they are for mammals. They basically go and attack the mammals. But are they considered to be parasites, Baba? Yes, everyone is saying C is the answer. Okay, C is the answer. Yes, Baba, C is the answer. Female mosquitoes basically are not parasites, and that is why it's an incorrect match. Cuscuta, which is there, it's a plant parasite, and they are found on hake plants. That is also correct, which is why the only option that is left with is going to be C. Please remember, mosquitoes are not parasites. Yeah, always loyal to both RCB and studies. Perfect. Okay, so yes, I hope that is clear, guys. If it is clear, just give me a thumbs up. Great going, Ganesh, Creative, Komali, Amreya, Kavita, Bhavish, Shweta, Vinu, and Shraddha, Hina, Anne Maria, Shivani, Narasimha, everyone. Okay, Hitesh. If at all I missed anyone's name, please forgive me. Kyun. Okay. Ma'am, this question is of PYQ. No, this is not of PYQ. There are PYQ questions also, but yeah, I'm correct again. Perfect. Let's go. Question number 85. Match the columns and select the correct option. So you have got, okay, options over here, and you have to choose which one is correct. So basically, you have to check like this. You have to match like this. 1, parasitism. 2, what will be there for this? And 3, what is going to be taking place out there. So, Baba, the easiest one to spot, I'll say, would be this. If at all you look at this particular question carefully. Thank you for giving thumbs up. Flabi, can you suggest any video for preparation? I had only watched your video. You're talking about subject, dear. There are 100 questions, top 100 questions being done for each subject. You can check that video out. It could be helpful. Yeah, it could be really, really helpful. Say anything, Malayalam. And now everybody. Okay, come on. Guys, the easiest way to spot over here would be minus, because minus relation is seen only in the case of competition. No, Baba, you are going to see in competition itself. In competition, what is going to happen? Both of them are going to compete with each other, which is why both of them are going to be harmed. Both get harmed. So I say 1. Okay, just a second. If I say like that, then 3, it's kind of confusing, is it? Just a second. Yes. So 3, 2, C. 3, 2, C. This is what I find. 3, 2, C is over here. 3, 2 is over here. So I can cut this option, and I can cut this 2 option also. Okay. Now what is there? Mutualism is where both of them are going to be, benefited. So 4, and both of them are benefited. That means which one will be there? Both get benefited. A. So 4, and A. 4, and A, which is why your answer would be A for this particular question. Yes, exactly. Okay, it is going to be A. Now someone has a complaint. Cool. Is this question clear to all of you? Ganesh Rao. Sorry, dear. Okay, so the names are coming up. That's why I'm saying Preetu, Pooja. Perfect. Cool. So please spot the easier ones and try to find out. This is not a PYQ, but yeah. Okay, let's go ahead with next question. Guys, I hope it is getting cleared. Okay. Can we go ahead? Give me a thumbs up. I'm speeding it up because you have been asking for that, but I hope you're understanding. Yeah? Yes. Cool. Let's go. Let me just explain. Amensalism means one is going to be, one is going to be negatively affected, other one is going to be neither harmed nor benefited. So it is a -0 relationship that is very uncommon, and basically no examples for this is also given in your NCERT. Okay. Parasitism means one is going to be affected, other one is going to be benefited. So plus-minus relationship. Okay. Now, question number 86. Question number 86. From the intertidal area of Scotland, sea coast, Balanus barnacle was removed. The effect was. So, Baba, from the intertidal area, Balanus was being removed. Okay, so Balanus was the one that was dominating, and Thalamus was the papa guy out there. Okay, it was recessive over here. Okay, when Balanus was removed, what was the thing that you were able to see? What were you able to see? A, B, C, D. What is the option that you can choose from here? What is the option that you can choose from here? Come on, come on, come on. Drink water. Thank you. Vijay says it is D, Bhavna says D, Silent Power says D, Sumitra says it is D, Shweta, Nandini, everyone, okay. Ambria says D. D is what all of those are. Let me see. Let me see. That is the effect. Thalamus barnacle population reduced in the absence of prey Balanus. No, Baba. How can that be true? Listen to me. Prey. It's a predator. Balanus is a predator, right? And Thalamus population reduced in the absence. How can it go? Reduce. Thalamus is going to increase when Balanus is not there. You are removing Balanus, no? So Thalamus is going to increase. And first of all, Balanus is not the prey, it is the predator. So this is going to be wrong. Thalamus population decreased because of other predators. There is no mention about other predators that we know about, right? So this is also going to be wrong. Thalamus population flourished because of absence of superior Balanus barnacle. Exactly. So these guys, whenever they found that, okay, the superior guy is not there, basically they are going to make sure that they increase their amount in too much number and they are going to flourish, right? So basically the answer for this particular question is going to be C. No, it is going to be C, right? It maximizes. Just a second. Yes, I was correct. So those of you who gave me the answer as D, it's not all of the above. You have to read the question very, very carefully. Okay, so I've selected this question because I knew that there could be something coming up. It's example of competition. It's an example of competitive release also. Competitive release money, for example, Balanus is there dominating. Okay, when Balanus comes, it's like Thalamus, you move out, bacha. And basically, Thalamus will be moving out, giving the whole area for Balanus. That is known as competitive release. Okay. Will this question come in examination? These concepts have been repeated and I'm trying my best to stay as close as the actual amount of questions that could be coming in. So yes, this could be helping you well. Ma'am, Thalamus population reduces after due to interspecific competition. No, ma'am. It's after that, right? Interspecific, that comes later on. But that particular situation is not given to you at all, right? And obviously all of the above option is not correct. What is the correct and the straight option that is given to you? It is C, which is why C is the answer. Cool? Now, question number 87. Come on, solve it, everyone. In the following table, the individuals of two species are given. Which of the pairs do not show commensalism? Which one do not show commensalism? That is the question. Commensalism means one is going to get benefited, other one is going to neither get harmed nor benefited. So orchid and mango. Do they show commensalism? Yes. Mango is a very big tree that is out there. Orchid says that, Macha, can I be right a little bit over here? I will not harm you at all. I just want sun rays. That's all. So this guy is getting benefit. Other guy is neither getting harmed nor benefited. So this is correct. Cattle egret and grazing cattle. You might have seen cow, they move around. When they move around, small birds are there, cattle egret, they also move around. Why? Because whenever the cattle moves, they are going to stamp the soil such that all the insects and everything comes up. And the cattle egret can go eat that particular insect. So this is also commensalism. Why? Because this guy is getting benefited, that guy is neither getting benefited nor getting harmed. What about sea anemone and clownfish? You might have seen Finding Nemo. In that a fish is there, right? The fish which is there, it is in the tentacles, in between the tentacles of the sea anemone. It provides protection. Sea anemone is providing protection to the clownfish. So this guy is getting benefited. What about this guy? Neither getting benefited nor getting harmed. Glomus fungi and the plant root. Here, what is there? Mutualism is there. Yes, all of you are saying D, Hina, Kausa, Sumitra, Bharat, and Mamta, Praveen, and Creative, everyone. Sayada, Vinay, if at all I'm missing out your names, so sorry. Glomus and fungi, basically, that is seen that they show mutualism. Both of them are helping each other. Glomus fungi is providing phosphorus from the soil, providing overall growth and development, providing resistance to pathogens, providing tolerance to all these different conditions out there. And the plant, what is it providing? Food. The food is being provided. Okay, this is known as mycorrhiza association, and this is an example of mutualism, which is why D is also correct. So D is going to be the wrong one because you have been asked about commensalism, not mutualism, right? And all of you who gave me what did I do? I put this question without the answer on. Okay, anyways. Okay, so demutualism, my dear children, all of you who gave me the answer, you guys are going great. All of you who do not know the answer, you are studying something. Okay, so world's pearl logistic growth is described by the equation dn by dt is equals to rn k minus n, where r and k represents what? This was a question. This is a PYQ question. Unfortunately, I've given you the answer already, but yes. So other three are commensalism, exactly. So the flow goes like this. Baba, listen to me very carefully, okay? Over here, the population is growing. In natural condition, do you think it can grow? No, it will be having a carrying capacity. Every environment has a carrying capacity that is denoted by K. Okay, yes. So it will grow until that particular level. Yeah. If at all there is no carrying capacity, basically it will grow like this. That is known as J-shaped curve, exponential growth. This is known as logistic growth or Pearl Hell's logistic growth. For this, you have a formula dn by dt is equals to Rn. That is a formula where R is intrinsic rate of natural increase denoted by basically birth minus death. So if at all birth is more, we say that R is more in amount. Okay, cool. That is not usually found in the nature unless and until you are providing situations like that. Now what about this logistic growth? The formula is dn by dt is equals to Rn, and you put k minus n divided by k. K is what? The carrying capacity. It's what? The carrying capacity. Cool. And over here, that is the question that is being asked. What is R? Intrinsic rate of natural increase, and K is the carrying capacity. But please know about this is an important thing. Okay, I think double 88 has come, so extra question will be there. Anyways, read the statement and choose the correct answer. You have to read the statement and choose the correct answer. Come on guys, try out this question. Question number 88. Statement number 1, the monarch butterfly feeds on poisonous weeds during its caterpillar stage. Statement number 2, it helps butterfly to become distasteful to its predator. What do you think is the answer for this particular question? What do you think is the answer for this particular question? B. Lavanya says it is B. Okay. Sujata, Shraddha says B. Nandini says B. Hitesh is also saying B. Okay. Vinay says B. Shraddha, Pallu and Lavanya and Saida, Fuddukaichilla. Okay, and everyone. Asfia, Vinay. Perfect. Pragnya, everyone. B is the answer. B is what? Let's check it out. The monarch butterfly feeds on poisonous weeds during its caterpillar stage. Yes. Why? So that when it becomes grown up, it can be distasteful. So anyone who comes eats it like, what is this? It will not come and eat it again. Okay, so this is correct. So both the statement 1 and 2 is correct, and it's a correct explanation as well, which is why this is a thumbs up. Yeah? Shall we go ahead with the next question? If yes, give me a thumbs up. I have been not asking for thumbs up. Give a thumbs up if at all it's all clear. If at all, we can go ahead with the next question. Nale Live is for math. Okay, come on. Thank you so much. Question number 89. The shape of the pyramids reflects the growth status of the population. Identify the type of age pyramid represented for human population. So this is the pyramid that is there, and you have to find the one that is there for human population. This is for human population. This is given over here. What do you think? Is it declining, expanding, or ascending, or stable? What do you think is the answer for this particular question? Any idea? A. Everyone is saying A is the answer. A is what? Declining. Is it declining? Is it declining? Let's check it out. Listen to me carefully. Okay. here this is the pre-reproductive age group organism, here is the reproductive, and this is post-reproductive. If at all the lower area is less, if at all the lower group of organisms are less, that means what? In future there are less organisms to reproduce. If there are less organisms to reproduce, my dear children, what is going to happen? Basically it is going to decline, which is why the answer for this question is actually A. Okay, if at all it was stable, both the pre-reproductive and reproductive would have been the same. Okay, if at all it was expanding, the lower portion will be very much big in nature. That is how it goes. So this is going to be declining because the lower portion is less, and I hope you already know the answer, which is why we move on to the next question, and that is question number 90. Guys, come on. And this is from the chapter ecosystem. So everyone who gave me the correct answer, super awesome. Okay, let's go. Match column 1 with column 2 and select the correct options from the given codes. So these are the options, that is A, B, C, D, and you have to choose from here. Column 1 and column 2 is rightly being mentioned over here. A. Okay. Gross primary productivity, net primary productivity, pond, crop field, and decomposition is given to you. Ma'am, you're beautiful. Thank you so much, Ratvik. Okay. Guys, come on, tell me the answer for this. Baba, yes, Lakshmi. Okay, Vinay says B is the answer. Bhagyashri says C is the answer. Hitesh says B is the answer. And B. Okay, Ambariya, Shraddha, Lakshmi, and Tata. Okay, IPLG, oh my God. They've come here. Okay, C, B. Okay. Most of you guys are saying B. Some people are also saying A. Let me check the answer. Listen, the most easiest one to find out is what? Probably pond or crop field. Or basically you have decomposition. Right. Crop field could be more easy. Crop field is something that we make. Man-made ecosystem is there. So D goes with 2. I just have one option and I've already got my answer. The answer is going to be B for this. It is going to be B for this. Is the easiest way to go with this. Okay, so crop field goes with man-made ecosystem. Pond is a self-sustaining ecosystem. And what do you mean by decomposition? Decomposition is a process where you require oxygen because aerobic bacteria is doing it. It says that I need oxygen, right? And what about net primary productivity? The amount that is available for the next group of organisms to take in. The productivity that is available for the next organism to take in, that is known as net primary productivity. So, so that goes over here. Where is it? Available to the secondary consumers. It goes like that. Now, photosynthetic production, how much amount of photosynthesis is being done and how much amount of productivity is being produced, that is known as the rate of production of organic matter during photosynthesis is known as what? Gross primary productivity. So this goes with this, which is why your answer for this question is going to be B. Why am I talking about all of it? Because this is very, very, very important. All the terms that you see over here, you have to revise by yourself. Very important. Ma'am, what is your name? Guys, don't spam. Yes, Shraddha gave me my name. It's Jasmine. Okay, my name is Jasmine. Ammalu is also there. Okay, so Ganesh is also saying my name. Perfect. Thank you so much, guys. You at least know my name. Okay, question number 91. Shall we move on to it? Okay. NPP equals GPP minus R. Exactly, Shivani. Pooja, the math class would be there tomorrow at 10 a.m. Okay, come on. Identify the incorrect statement regarding the flow of energy between various components of the food chain. This is a PYQ question and probably we can do it. Yeah? So, energy flow is unidirectional. Energy flow, is it unidirectional? We all know energy flow is always unidirectional. The lower one will have high energy, the upper one will have low energy. Okay. Green plants capture about 10% of the solar energy that falls on the leaves. You tell me the answer for that. Is that true or false? Yeah. Next one, each trophic level loses some energy as heat to the environment. Obviously, not all energy transfer is going to be 100% efficient. Whenever you pass the energy, some of energy is going to be lost. Okay, the amount of energy available at each trophic level is 10% of the previous. We know 10% law. So what are we left with? We are just left with this option, that is green plants capture about 10% of energy. That is a thing, right? That is a thing, and everyone have given me the answer as B. Lavanya, Shraddha, Ratvik, okay, no, not Prajwal, Shivani, Creative. Listen to me. The answer for this question is B only. Why? Because green plants capture we cannot say 10%, it can be anywhere between 2 to 10%. It can be anywhere between 2 to 10% of photosynthetically active radiation, or you call this as PAR. Remember, sun emits too much radiation, but only a little bit of it can be taken by plants, and that is 2 to 10%. Cool? Ma'am, how to score good score? You have to watch this video. Cool? So green plant basically capture 50% of it. That is known as photosynthetically active radiation. Out of that, only 50% of energy light is available. Out of that, only 2 to 10% of it can be utilized by the plants. That is what I meant. Okay? Yes. So B is the answer for this particular question. Is this thing clear to you? Is this thing clear to all of you? Ma'am, when is your next class? On Monday. Ayyo. Is this clear to everyone? If yes, give me a quick thumbs up. If yes, give me a quick thumbs up. We'll move on to the next question. Hitesh, love you too, da. Ma'am, what's your age? How aged I look? My age is 55. Okay. Next one. Question number 92. Choose the correct sequence of steps involved in decomposition. Come on, we are on 92 question. Come on, come on, come on. Choose the correct sequence of steps involved in decomposition. Thank you for giving me thumbs up. Okay. Ayyo, madto. Okay. Come on. Baba, cool. B. Sagar says B is the answer. Chandana says A. Shivani says D. A, B, D given. C is only thing that is left. Give me that also. Bhavish says D. Suprit says A. Okay. Shivani D. Asfia says D. What is the answer for this? Listen. Okay. FLCHM. FLCHM, fragmentation, leaching, catabolism, humidification, mineralization. Your answer for this question is going to be D. Do you have any formula for that? I actually tell formulas, I usually tell tricks, and I forget it. I usually forget it. D is the answer for this. Fragmentation first, then whatever water soluble inorganic components are there, they will move into the soil, and whatever is left, they are going to be broken down. That is known as catabolism. And while you do catabolism, some of it will be available as humus. It is dark amorphous substance which is there. It's not crystalline, only amorphous. And basically it is going to break down slowly into inorganic things. That is known as mineralization. Okay. And everything about decomposition, at what temperature? Warm temperature, aerobic bacteria, all of this thing is very, very important. Okay, so please remember it. FLCHM, and there was a formula to remember it. I mean, there was something to remember it, but anyway, got it right. Let's move on to the next question. It's an easy question, right? Thumbs up. Funny lion can hunt mice. Funny lion can hunt mice. Yeah, good one. Who said that? Narmada. Funny lion can hunt mice. Yes, that is our time. Funny lion can hunt mice. Okay. Fatal Club of Headmasters. Oh my God. Okay, Fatal Club of Headmasters. Cool. Perfect. Let's go ahead. Question number 93, biodiversity and conservation. By the way, over here, please remember, lignin and chitin, if at all is there, decomposition is going to be difficult. In the case of water, aquatic ecosystem, detritus food chain is going to go low. Why? Because their temperature is temperature is very low inside water. Very low temperature means what? Basically, it's cold. In cold temperature, decomposition cannot take place. So their grazing food chain is something that is going to be dominant in water. Okay, perfect, Sagar. Okay, thank you. And yes, let's go with the next question. Question number 93. Match the column with and choose the correct options. Endemism, hotspot of India, sacred groups, cryopreservation, ex situ conservation. From this chapter, you can get whatever questions that is there, but still, let's try it up. Okay, let's try it up. Shivanand. Ma'am, what's my full name? Jasmine Joy. Okay. Endemism, hotspot of India, sacred groves, cryopreservation, ex situ conservation, come on. Over here, Khasi and Gential is there, advanced ex situ conservation is there, species found in particular areas, zoological parks and botanical gardens, Western Ghats. So the easiest for me is what? Ex situ conservation. Conservation outside their natural resource, right? And everyone has started giving me the answer also. Hitesh, Shraddha, Ganesh, okay, A, Asfias, okay, Amlu, A, okay, fine. So let me go with A, but let me check. E goes with 4. E goes with 4 because zoo and botanical gardens are ex situ form of conservation. E goes with 4. I do not have any other option, which is why I think A is the answer for this. Endemism means what? It is very unique. Organisms are not going to be found elsewhere apart from this area. So species found in a specific area, in a particular area only, is known as endemism. Okay. Now you have hotspot of India. Okay, hotspot of India is basically, I can say it is going to be Western Ghats. Western Ghats, basically it comes under one of the biodiversity hotspot. There are 34 in total, but this is one among them. Yeah, sacred groves you can found in Khasi and Gential hills of Meghalaya. So that is the one that is there. And cryopreservation, which is left behind, it is an advanced ex situ conservation along with seed banks, IVF and all of it, tissue culture. Okay, so the answer for this question is A, and if at all you have got it, give me a thumbs up, and everyone who have not hit on the like button yet, click on the like button. Let's make it 300 and let's increase the subscribers out there. Yeah, and guys, stop spamming. Yes, do not disturb my little children. Let's go. Let's go ahead. Thumbs up, please. Fast. Yes. Question number 94. Match for M and N with species A relationship shown in graphic representation below. Okay, what is M, what is N? Over here you have species richness, and here you have area. So you can let me know. What is this? So I'll tell the story. Thank you guys for giving the thumbs up and listen to me, the story time. Story goes like this. This is Alexander von Humboldt. Okay, I am Alexander von Humboldt and I'm walking around and I see, oh my God, with more area that I'm exploring, I see more organisms out there. So he came to a conclusion that basically, if I'm talking about area, and this is the species richness of the species, okay? With more explored area, it is increasing. But after I reached at one particular point, I realized that it's not increasing further. So this is what we say, that it has reached some level up there. It has reached a level up there. Okay, it is not going to increase any further. Right? So how did he come up with a formula for that? He said that species area, which is there, species richness which is there, we can say it is proportional to the area, but not all the area, to some extent, and that some extent is represented by Z, that is Z word. Okay. Now, if at all we remove this particular thing, we replace it with C. It becomes, the formula becomes this. So don't do ratification, understand this, I always say this. Okay. So this is the formula for this particular curve that you see. Now, if at all you put log value, the curve comes like this, and you put the log formula, it goes like this. If it was S there, I will call it log S now. Is equals to, if it was C there, I'll say log C. It was A there, so I'll say log A. But you know Z value is also there, so Z value comes before log. So this is the formula for log. This would be the formula for log. So over here, if at all you had to choose, what option do you have? M is equals to C, S is equals to C, A, Z. Yes. Here also the same. These two options are anyway going to be wrong. Now, what is N? N is equals to log S is equals to log C plus Z log A, which is why the answer for this question is going to be A. And everyone who gave me the answer as A, yes, it is going to be A. Very, 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 very important. Whew. Cool? Also, one very important thing, this is known as the slope of regression. Yeah, it is denoted by Z, if at all. Yeah, and there is C is nothing but the y-intercept. Yeah, guys, if at all you can just give me thumbs up. There is no leaderboard. There's no leaderboard here. Yes, perfect. So let's go ahead with the next question out there, and that is question number 95. Select the options from the following which is not major characteristic feature of biodiversity hotspot. So you have to say which is not a major characteristic feature of biodiversity hotspot. Come on. Come on guys, come on, do this question. Which is not the major characteristic feature. You have first one, large number of species, second one is destruction of habitats, third one is abundance of endemic species, and D1 is large number of exotic species. Which one is not is being asked? Sigmoid curve, yeah, that was sigmoid curve, dear Flippy. Ma'am, please make a video on veterinary science. Yes, dear, most probably I would be able to do it only after your KCET examination. I'm not giving any false hope over here, okay? So B, everyone is saying B is the answer for this. What is B over here? Let me check. Destruction of, I knew that you will do this mistake. And you know what? I have done this question before. Listen to me carefully. Biodiversity hotspot is there. What are you saving are there? Large number of species are being saved. Abundance of endemic species, as in species which are very unique. They are only found in those areas. Now, destruction of habitat, this is all something that you see over there. Okay, biodiversity hotspots, you usually see destruction of habitat out there. There are so many, because, you know, yeah. Because it's in a less explored area, people go and destruct the area out there, create buildings out there. So yes, it is under a lot of trouble because of this. What is the answer? Large number of exotic species. This is wrong. Exotic means something that you have brought from outside. In biodiversity hotspot, they are naturally replenished with so many endemic species, not exotic ones. Okay, so D is going to be your answer for this particular question. So all of you who have given me other answers, wrong. Okay, it is going to be D. I hope that works. 95, few more questions are there, bus. Okay, guys, thumbs up. Let's go ahead. Question number 96. In the given pie diagram, the vertebrate groups are marked as 1, 2, 3, 4, and 5. Find the correct proportion of vertebrate groups over here. Can you tell me what is the answer for this particular question? Ambaria, tomorrow class will be there for mathematics at 10 a.m. D. Parvi Mullah says, oh my god, D is the answer. Vinay says, thank you for the thumbs up. Sujat, okay. Saida, thank you for thumbs up. Okay. Hitesh says C, Bhavish says C, Pooja says D is the answer. Snag Wines says D, yeah, okay. Manjula, Flabby John, and Deepa C, Bhagishri, Asfia, okay, Gagan. Okay, listen to me. The answer for this question is going to be, always when you get something like this, just understand, remember the formula. Farm. It is what? Farm. Always. So, over here, just a second. Yeah, it is this. It is going to be this. So what is C over here? C is going to be, just a second. I did a mistake. First one is going to be fishes for sure. First one is going to be fishes. And when you do farm, basically here comes aves also. So in that particular case, first is fishes and 2 is going to be mammals. This is going to be your answer for the question. C is going to be the answer for the question. It is going to be C. But just let me, okay, FB Ram. Ah, that is nice. Okay, just give me a second, okay? And conservation, CRT PDF. Just a second. Yeah. Okay, so it is going to be C. It is not something else, it is going to be C. Ma'am, RCB match now. Oh my god, I'm ending it. Okay. So in the farm one, in between A comes, A is for aves. Okay, A is for aves. That is also there. Basically, that means birds. So the answer for this question is going to be C. I hope that is clear. Is this question clear to all of you? Is this thing clear to all of you? If yes, can you give me thumbs up? If yes, can you give me thumbs up? And I can go ahead with the next question. Very, very slow. Okay, perfect. So question number here, this is marked wrong, okay? This is the answer. Okay. Question number 97. Again, now only PU1 questions, few questions, 6 questions are there, we are done, okay? Epigynous flower, not even 6. Epigynous flower is one in which, what is being seen? Ovary is superior and other floral parts are inferior. Ovary is inferior, other floral parts are superior. All the floral parts are at the same level, none of the above. What do you think is the answer for this question? Come on, first. What do you think is the answer for this? Epigynous flower is the one which. What are you able to see over here? Manjunath says B, Amalu says B is the answer, okay. Lavanya says A, Hitesh says B is the answer, Karthik also says B is the answer. What is B? What is B? Ovary. Ovary is inferior and other floral parts are superior. Is that the answer? Exactly. Ovary is going to be inferior and other parts are going to be superior. So if at all you see this is epigynous. Ovary is right over here. On the top of it you have the sepals, the petals and the male parts. So this is inferior ovary, that is epigynous. If at all the ovary is superior above everything, you call it hypogynous condition. If at all it is in somewhere in the middle range, all of them are in the same level, we say it is perigynous. So the answer for this question is going to be B, and all of you who gave me the correct answer, kudos to you. Yes? Okay, shall we move ahead with the next question? Question number 98. Identify the floral formula of plant belonging to the potato family. Potato means Solanaceae family. Potato means what, Baba? Solanaceae family. And in Solanaceae, what do you have? Let's check it out. In Solanaceae, what is there? So, if you can clearly see over here, bisexuality is there. Male part and female part is clearly been mentioned. So, obviously it is going to be bisexual, as said in all. Okay, now what does K stand for? K stands for the calyx. K stands for calyx. That means the petals which, sepals which are there. How many sepals are there, Baba, over here? You check it out. Sepals, how many of them are there? 1, 2, 3, 4, 5. 5 of them are there. And they are attached to each other? Yes, ma'am, they are attached to each other. So, what do? I put K and then I put 5 and then I put it in the bracket. Why? Because they are attached to each other. Okay. Okay. What about the next thing? C. C is what? Corolla. Corolla means your petals. Okay. All the petals together. Now the next thing that you see inside is petals. 1, 2, 3, 4, 5. Again, all of them are joined to each other, which is why 5 will also be inside the bracket over here. So again, 5 is inside the bracket. Okay. What about androecium? The male sex organ over here, androecium, how many of them are there? 1, 2, 3, 4, 5. Are they close to each other? Are they arranged together? No. They are fused with what? They are fused with the petals. This is known as epipetalous condition. Okay, they are not fused amongst us themselves. So we say they are 5 in number, but we do not put in bracket. Rather, we put something like this. This shows the connection between the petals and them. Okay, then what do we have? We have the gynoecium. What about the gynoecium? Baba, gynoecium, whenever it is present in the center, that means it is going to be superior. You make a line below it, that means superior is there. Okay, and one more very important thing which is there, and that is how many gynoecium is there? Basically 2 of them is there, and 2 of them are fused within the similar placenta. So I'll say 2 and I'll put it inside the bracket, which is why the answer for this question is going to be D, because everything is being maintained. Also please understand there is a line below it. Okay, and all of you, okay, cool. Please remember 2 family, Brassicaceae and Solanaceae, both of them very important. Question number 99. In which type of vascular bundles are xylem and phloem present at the same radius? In which type of vascular bundles are xylem and the phloem present at the same radius? So phloem will be there and similarly xylem will be here itself. In the same radius, what do you call this arrangement? Any idea? And in some cases, if at all the phloem is here, the xylem would be here. That is not similar arrangement. That is known as alternate arrangement. Where do you see here? C. C is the answer. Most of you guys are saying C, 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 C. Sorry, I'm not reading out your names because your time is running out. C is what? Conjoint. Yes, it is known as conjoint. And conjoint is something that you find in the case of what, Baba? Stem. In the case of stem, you find it monocot stem and monocot stem, dicot stem. Everywhere you find conjoint. Okay. But there are 2 things. Conjoint open, conjoint closed. Open means it will be having a layer of cambium in between. That will be leading to the secondary growth, secondary xylem phylum, secondary phloem. That is a feature of dicot. Okay, and if at all it is not there, okay, so I will call this as open to change. That is why, that means there is cambium. In the case of monocot stem, what is going to happen? There is no cambium in between, so I'll say it is closed. But always remember, if it is a stem, it is going to be conjoined. What about this? This arrangement is seen in the case of what? Radial arrangement is this, and radial R for root. All the root, dicot or monocot, they will be having this. If, how do you understand whether it is dicot? If it is having cambium there, it will be dicot root. If it is not having cambium, it is going to be monocot. Please understand the logic. You can get questions based on that also. We have solved the question in the mock test, the full length mock test, right? So the answer for this question is going to be C, Baba. It is going to be C. Someone says C, Baba. Yes, exactly, Gagan. Gaganesh. Grinod is there, Umbria. Perfect, children. Radial roots, yeah. So please remember that particular thing, radial roots, RR. This question could be coming in, it could be helpful for you for our class. Okay, I have given the whole answer over here. You can check it out using the PDF. And next thing, 100th question, 3 more are there, then we'll finish it up, don't worry, okay? And it will be mixed, Mahalakshmi, it is going to be mixed. The functional unit of contraction is, contraction, the functional unit of contraction is what, Baba? Portion of myofibril between two successive Z lines, portion of myofibrils between two successive M lines, and center of the edge zone or center of the I band. What do you call this? The functional unit of contraction. You call this as sarcomere. You call this as sarcomere. And where is it? Between the two Z lines. Please remember, so many questions have come from this chapter. The same topic. There are two Z lines over here. This portion that you see is known as sarcomere. In between this band that you see is A band. Okay, A band is formed of just myosin, the dark one. The dark one. Okay, it is the darker band that is taken up, and over here, if at all you see these red bands which are there, which are on the Z line, they are the actin filaments, and they form the I band, and the actin one is the one that has light bands. Okay, they have the light bands. Well, usually what happens whenever contraction takes place, both the Z lines come here. When the Z lines come here, what is going to happen? The A band, the size of it remains the same. What about the I band? It gets reduced because it gets inverse. That is why it gets reduced out there. So always remember, when contraction happens, I band changes, but the A band remains as such. And basically, this is just having myosin. Over here, you have actin also. And yes, that's all. Okay, so the answer for this particular question is going to be portion of myofibril between two successive Z lines. This is something that is super important that you should not be missing from locomotion and movement. Yes. 3 more questions, guys. We're gonna finish it. We are gonna do it fast. I'm not asking you the question, I'm explaining it to you for making sure the time is being utilized. Identify the incorrect statement with respect to the rules of binomial nomenclature. Binomial nomenclature, you have to find which is incorrect statement. Okay, the first word represents the genus while the second component denotes the specific epithet. Is it? Yes. The first word represents the genus, the second word is basically the species name. Homo sapiens, Homo is basically genus, sapiens is basically specific name. Biological names are generally in Latin or Latinized irrespective of their origin. Yeah, if at all it is not in Latin, it is converted to Latin. That is also correct. Now, biological names are underlined separately when handwritten. Are they? Yes, whenever you are writing it through hand, you have to underline it. And my dear children are giving me the answer D, D, D. I'm super proud of you. And what is D? Biological names are printed in italics to indicate their non-Latin origin. They are definitely printed in italics, but it is to indicate their Latin origin, not the non-Latin origin. So this is going to be a wrong statement, and you had to find that. So D1 would be this. Cool? Okay, so again I have written everything about it. Question number 102 from plant kingdom. Phycoerythrin and floridine starch are found in. Phycoerythrin and floridine starch are found in what? Brown algae, blue-green algae, red algae, or green algae? What do you think is the answer for this? 102C is the answer. Yes. Red algae, Rhodophyceae, is the one where you have phycoerythrin and fluoridine starch. This is one very important thing. Do not mess it up by any chance. This is very, very, very important. Okay. Rhodophyceae, they are the ones which produce non-motile spores. There are some unique features about it. Okay. So do not mess it up. It's an important topic. And yes, all of you are correct. Okay. And that's all. It was 102 questions. Okay. So my dear children, we have finished all the questions out there. Obviously there is more to it. Okay, and we cannot put that over here, all the questions. But today I'll be giving you a video of most important concepts of KCET Biology, chapter by chapter. You can watch that video in YouTube and make sure you make the best use of it. I have done a lot of research for that. And will be Banal Sangeeta, it will be more than that. It will be 30 marks, you will get more than that. Yes. So, dear children, first of all, thank you so much for being throughout. There were some children who were there throughout the live and some who joined in between. I believe that this live was useful for you. Yeah? Amlu, that's so sweet of you. Prajwal, thank you so much. Gagan, thank you so much, dear. No man, okay, thank you, Shivani, everyone. Guys, I'm so sorry I could not read out your names today, but I really mean it. You guys are wonderful, and yeah, please take care of yourself. Yes, and do not forget to hit the subscribe button before going. Lots of love to all of you, and thank you so much, Preeti, everyone. Ma'am, I scored 92%, but being compared to 98, you cannot do anything. Just give your best. That's all. Okay, and forget about the people who said that. And yes, Polichu. Thank you so much, Muhammad Sawan. Okay, guys, Vidya, Hema, love you too, dear. Yes, I'll drink some water. Thanks everyone, Shraddha, Sharanya, everyone. I'm just here. If at all you have any doubts, you can ask me. Saida, love you too. You're most welcome, Hina. Okay, so I think, and Maria, thank you so much. Okay guys, so I'll take rest. I'll go sit somewhere, and thank you so much for being over here, and make sure you study well. We are putting in our hard works for you guys. You have to put in your hard works too. And ma'am, will I get 50 plus and score 100 in bio? You will be getting it. Okay, so difference between homozygotic and monozygotic twins. Where did you see this question, dear? Homo is never been asked. Monozygotic is still being considered. Monozygotic basically means one zygote which is there, it is going to divide into two and it is going to form two embryos. So basically all the genetic matter, everything is going to be similar. But homozygotic is something that you never, I mean, that is, are you sure that is a correct term for whatever you are asking? Love you too, Chetan. Yes. Guys, I think I'll wind up, otherwise I'll fall down now. So wishing you all the best. Please take care of yourself. If you have any doubts, put it down in the comment section. We'll be checking that there. And yes, ma'am, is it a last live? No, I'll be coming live more towards examination. Yeah? Bye-bye. Take care.